Good morning, everyone. Let's get started so we can keep the schedule more or less on time while people are trickling in. So thank you all for being here. Thank you for spending your Saturday talking about eye and vision research. We are delighted that you all are here. And welcome all of you to the 10th annual Bench to Bedside Symposium organized by the Gavin Herbert Eye Institute. It's a bittersweet event for us. Obviously, many of you know Chris Kenny was the person who initiated this event and had been running it for a long, long time. Unfortunately, she passed away late last year, and we have instituted a new Chris Kenny Memorial Lecture, which will be, the inaugural lecture will be given by Deborah Farrington, who collaborated with Chris and was a good friend. And so we are very grateful and excited that Deb is here to do that. And I think that Chris would have been happy and proud of this event. We have a record turn up. We have, let's see, look at my notes. We have 212 registrants this year, which is about 50 more than last year. We have a record number of poster submissions. We have 58 posters compared to 30 last year. And if you walked around, you saw that we had to put posters all over the place. So it will be a bit of a treasure hunt to find all the posters, but some of them are in the corridor, some of them are in the main hallway. There are some in the adjacent rooms as well. So please try to see as many of them as possible. I want to acknowledge, of course, the sponsors for this event, people who, companies and entities who provided financial support, including AbbVie, Genentech, Sun Pharma, and the Discovery Center for Eye Research. The department is also supported by Research to Prevent Blindness and CTVR. I also want to acknowledge the people who worked very hard to make this event possible. My co-organizer, Ken Lin, who worked with me to put the agenda together and to invite all of the speakers. Of course, none of this would have been possible before without the hard work of Dana Collinson and Angelica Arias, who worked very hard to bring all of you together, put the agenda, and just organize the logistics of the event. I also want to thank the people who are here on Saturday from our staff working and helping with the event. Luis Delgado, Thomas Cross, and Angel Nalbandian, and Ezra Rumke as well. I want to thank the poster judges. Again, I said we had a record number of posters this year. So it was hard work for the judges to have pre-screened the posters, and there will be a poster award ceremony at the end of the day. And so the 6 people who worked very hard to review the posters, and we'll be reviewing them more today, are Kapil Mishra, Austin Fox, Andrew Smith, Philip Kaiser, Dorota Skronovska-Kravchik, and Gulab Zodeh. If you are presenting a poster, please make sure that you are around your poster during the poster sessions and try to talk to the judges and to everybody else as well. So again, thank you all for being here. We are very excited. We have 5 very diverse sessions ahead of us, with poster sessions in between and a lunch break and a dinner and a celebration at the end of the day. Thank you all for being here. And again, let's hope we have a great day together about vision research.
Hi, everybody. My name's Sam Spiegel. I'm one of the neuro-ophthalmologists here. I'll be moderating our first session. We're going to start our morning off with Dr. Vivek Patel. He's a professor of clinical ophthalmology here at UC Irvine and also the chief of the neuro-ophthalmology division. In his free time, aside from shuttling his kids to and from their sports practices, he also has a very busy clinical neuro-ophthalmology and adult strabismus practice. On top of that, his research here focuses on advanced imaging of the visual pathways. He's going to be talking on advanced imaging of the visual pathways, structure, and function correlations.
Thank you, Sam, and thank you, Vladimir, for inviting me to be part of this symposium. It's a true Southern California affair, given all the people that are here speaking with us. But I'd like to just start off with ensuring that I know how to advance. This is actually kind of an interesting area that I've been working on, been interested in, even prior to being a neuro-ophthalmologist. The neuroscience revolution in terms of imaging has been incredible, and it expands well beyond the retina and optic nerve all the way back to the brain. So some of the stuff that I've been working on, we'll talk about. So this is actually the very first clinically used CT scan in 1971 at Atkinson Morley Hospital in London. And you can see this frontal lobe tumor that was there for a 41-year-old woman who had presented with some frontal lobe signs, some disinhibition, memory changes, behavioral changes. And then this first CT scan, which took 3.5 hours to process, was what it showed. And then the surgeon that took out this tumor confirmed it was a cystic astrocytoma. So we've come a fair way since that time, and now you can see this is a modern MRI on the right that you can see every single day in the clinic. And then we can even assign colors to the orientation of the white matter in so-called diffusion tensor imaging. So that colorful map there you can see, in the green is essentially fibers that are moving front to back or back to front. The red are fibers moving left to right or right left. And you can see that the purple are fibers that are moving from the dorsal to ventral, ventral dorsal. So you can actually assign what's called a color map by orientation. And then if we put them together, each of these individual diffusion tensor images into a tract, you can get what's called tractography. And you can see on the right this really exquisite mapping of the circuitry within the brain. You can see the slice on the left is essentially a nice, beautiful, gross dissection of how you can see the visual pathways. There's a chiasm in the middle, the tracts, the lateral geniculate nucleus, and all the way back to the radiations. And you can approximate these images beautifully using this tractography. So these techniques are available. And of course, we're no strangers to seeing near histological detail in the retina every day. You can see on top, there's obviously a amount of histopathologically the way the retina is organized, and then the OCTs that we can look at each day that can show the same amount of resolution on a clinical basis. So we thought that we would put those together because really ophthalmology, neuro-ophthalmology is about structure-functional correlations. And you can see that's what we do every day. In the top left, there's a patient with macular degeneration giving you a central scotoma. In the top middle, retinitis pigmentosa, peripheral visual field constriction, and so forth. You can see the bitemporal hemianopia due to the pituitary tumor, optic head nerve head drusen, ischemic optic neuropathy, and in the bottom right, you can see a compressive optic neuropathy from an optic nerve sheath meningioma, giving you a central field defect. That's the daily practice. So the applications of this technology, how do we bring the two together? We know that retinal and optic nerve pathology correlates with structural and functional alterations in the brain and vice versa. And we know that there may be resulting central visual pathway changes when you have problems at the level of the retina. So what if you've had long-standing retinal disease, optic nerve disease? Will the central visual pathways change structurally and or functionally? And that's important if you kind of think down the road about sight restoration therapies, whether that's cortical implants, retinal implants, regeneration of retinal neurons, and so forth. So now we have the tools to be able to assess that, at least to some degree. And this was part of a grant that I worked on as a multi-PI kind of a consortium when I was at USC. And it was termed the Low Vision Blindness and Sight Restoration. And it was a combination of looking at from neuroimaging centers at USC as well as University of Pennsylvania and ourselves from neuro-ophthalmology. And it actually led to some interesting discoveries. I'll give you a retina, I'll start with the retina, as I probably should, for some representative examples, okay? This is one example of an outer retinal dysfunction. So there was RPE damage as well as outer retinal damage, okay? So we'll talk about a 30-year-old woman, motor vehicle accident about 9 years ago, and a decreased central vision in the right eye, okay? And if you see that line that I've drawn in the middle, that's the fovea, okay? And essentially, or basically the location of the fovea. And nasal to it, the two yellow arrows are showing atrophy of the RPE and the outer retina. And then temporal to it, you can see that that outer retina is intact with those two yellow arrows, okay? And indeed, when you look at the autofluorescence image, you can see that there's atrophy in that region where you saw the lack of normal organization on the OCT. So this is essentially the nasal papillomacular bundle, okay? So the fovea is essentially the area where your visual field separates left and right. So you can see in that area where we saw the OCT was abnormal, you see the autofluorescence changes. And autofluorescence is going to be abnormal when there's an abnormality with lipofuscin, which is mostly stored in the RPE and the outer retina. So this is an outer retinal disease. And if we look just to the right of that, when we stimulate that area of atrophy, there's hardly any response through microperimetry. And when you stimulate the other parts in the green of the retina, in those areas, there's a normal response. So as you'd think, in that area where there is that traumatic maculopathy, loss of RPE, you see very little response from direct stimulation of that retinal area, okay? And that's how the tools we're going to use. Now remember, just show you very quickly, that is the right eye, that's abnormal, and the nasal aspect of the right eye, correct? And we know that nasal fibers cross, okay? So let's go to that right eye, and you can see that right eye drawn there, those nasal fibers are cross, and they go to where that red circle is, which is in the left occipital lobe, okay? And when we do the fMRI, we see that correspondingly, as you'd expect, the area of blue, that's essentially showing where there's decreased activation in the fMRI, corresponding with that region of atrophy that you saw in the nasal papillomacular bundle of the right eye. So you see that nice structural functional correlation all the way back to the cortex, okay, with it using the fMRI. And in the opposite hemisphere, on that right hemisphere, you can see that it's perfectly fine, and there's a normal response because that's coming from the other eye. Okay, and then if we put this in fundus coordinates, take that microperimetry again, that signal, and you see that abnormality there on the right, and that correlates exactly, if we put the cortex in fundus coordinates, with the area of suppression or lack of activity in the brain itself. Okay, so when you have an area abnormal in the retina that would correspond to a certain part of the visual cortex, we can show these abnormalities with functional MRI. And at that point, at least, when we're looking at these patients, the visual cortex remained intact when you look at the actual volume of the cortex. So there weren't structural changes, but there clearly were functional changes. Okay? And then we kind of took that further. There were 12 people in the retinitis pigmentosa group. Obviously, there was long-standing vision loss, anywhere from 7 to 30 years in these patients. And we have seen, and this is probably not something I have time for in this talk, but the long-duration patients actually have some cortical thinning, and there are some changes downstream, which may be recoverable to some degree with stimulation in the future, which may be something we can talk about in the future. But analysis demonstrates interesting cross-modal plasticity. So let me just kind of walk you through this. Okay, this is a patient, 55 years old, that had some residual central vision. That microperimetry that you can see, I don't know if there's a laser on this, but okay. This looks like something you'd, all right. So they see that here, there's a lot of peripheral vision loss, but you can see centrally there is a little bit of vision left. Okay, so when you look here, this is the left eye and that's the right eye, 20/60 and 20/50 vision. So when you put a flashlight to this person, you just have them in an fMRI, just look at flashes of light, as you'd expect, there is some response in the visual cortex because there is some central vision. There's response there. And the same thing here in the left eye, there's some response. And then when you have them do like a tactile stimulation, invoking your somatosensory system, admittedly, because there's normal vision, or there's at least some vision to stimulate that cortex, you don't get much in terms of a shape recognition or sandpaper. It's not eliciting any response in the occipital lobe because there is enough vision to stimulate that area, okay? Now, if we look at another patient, this patient has much poorer central vision, okay? Bare LP with the left eye and hand motions with the right eye. And as you'd expect, when you shine that light, there's very little response. A hand motions patient, a little bit better response. But what happens is when you have them do the sandpaper, the shape task, there's much more avid activation of that occipital cortex, which shows that there is some colonization of that unused visual cortex by something else. And that's kind of like the principles you may kind of look behind to see for Braille and so forth. But here, we're not getting much stimulation from the visual cortex. It's pretty prime real estate, so somebody moves in, and that somatosensory system is able to activate activation within the primary visual cortex, which is phenomenal. The question is, when you bring the vision back, if you can, will it go back to activation with vision? And that's likely the case. So let me go switch gears a little bit to the optic nerve. And as I mentioned, this is an inner retinal problem, and it's a 68-year-old man that had an acute onset of right visual field loss 6 weeks prior, so kind of a more acute presentation. And here you can see the red arrows show the outer retina is fine, as you'd expect in a patient with an ischemic optic neuropathy. It's a ganglion cell problem and a retinal nerve fiber layer problem. And sure enough, you can see the superior retina in this area is thinner. That RNFL is gone, and the ganglion cell is also thinner, which would correspond with the inferior visual field loss. Okay? And sure enough, on on autofluorescence, because it's an inner retinal problem and the lipofuscin within the RPE and the outer segments of the photoreceptors are normal, autofluorescence is normal, okay? But sure enough, with the microperimetry, you see when you stimulate that superior retina, which is now atrophic, you hardly get a response. But when you stimulate the inferior retina, you get a response, as you'd expect. Okay, so the microperimetry shows that nice altitudinal flavor that we would expect. Okay? And remember, just remind yourselves of the way the visual cortex is organized. The superior retina travels through the parietal lobe and extends into the superior lingual gyrus. And in blue, the inferior retina travels through the temporal lobe and extends into the inferior lingual gyrus. So that organization is there for us to kind of look at. And then when we do the fMRI studies, as you see, again, just like the other case, you can see that superior occipital cortex, superior lingual gyrus, has a lack of activation relative to the inferior because those superior retinal projections are actually not activating that part of the cortex because it's atrophic. So again, even with optic nerve disease, we can see this nice structure-function correlation back to the cortex using fMRI. And at least at the time that we had to look at these patients, the structural anatomy was normal. There was no downstream change. Remember, this is only a 6-week patient, but we have scanned people with many years of ischemic optic neuropathy, and it did not appear that they had loss of true volume of the tissue. So potentially, that's something to look forward to. So as I mentioned, structural changes, is it just a matter of duration, detectability, or both? Not sure, but at that point, no real structural changes were seen. But we do know every day in the clinic, and Sam and others will attest to this, we see evidence of retrograde degeneration every day. Let me give you a quick example of a patient that had come in with some decreased vision. We don't know when. He says, I haven't seen well for years. 20/20 vision in both eyes. We did a visual field, and you can see a right homonymous hemianopia. So the right half of the visual field is lost in this patient. And then as part of our evaluation, we did an OCT. This is the ganglion cell analysis, and as you know, the OCT is measuring that primary visual neuron, the part that's the pregeniculate system, right? And when we see this pattern, when you can see that homonymous loss that is here, that corresponds with the visual field loss, generally speaking, you think that's anterior to the LGN, right? Because that should be an optic tract lesion, because you're imaging the primary visual neuron at that point, okay? So then the clinical assumption at this point was, well, he probably had a contralateral optic tract lesion. Okay, but then when we do the MRI, you can see that it's not anything wrong with the tract. It's actually an old occipital infarct on the corresponding side. This is obviously the left side, and therefore right homonymous hemianopia. But this is a great example of transsynaptic retrograde degeneration. So of course you'd expect the area encircled, which is the area of the encephalomalacia, to be abnormal with the tractography, but the whole bundle atrophies as you move anteriorly. You see, so essentially what happens, it depends what you consider anterior and posterior, but as you move toward the eye, in an eye-centric world, you call this anterior, but nonetheless, the abnormality is here, but the whole bundle shrivels up as you go the other direction. So retrograde degeneration is seen every day. And if you look in the old textbooks, we never thought that you would see atrophy at the level of the optic nerve from an occipital lesion, but we now have these tools to be able to see it. And the OCT is really key in that, and we see this pretty much every day. So retrograde degeneration does occur. So essentially, to summarize, detailed structure-functional correlations, they're resolvable and they're possible with modern techniques. This isn't something totally far off. These techniques exist. Diffuse and tensor imaging is used every day in clinical practice, whether it's for demyelination, tumor planning, and so forth. There's, of course, implications for neurodegenerative disease. Many people are interested in this. Obviously, because you can see connections. You can see connections from the primary visual cortex to the limbic system, to the frontal cortex, parietal cortex. And essentially, there's so many diseases that are not localizable to one area. Having the ability to integrate this functionally and structurally is obviously important. And there's applications to amblyopia, strabismus, and so forth. One area that we're interested in, and Amr Hassan, who's one of the retina fellows who understands the retina is just a part of the brain, I guess, is interested in looking at this as well with me. And what we're looking for is patients that have strabismus and maybe non-amblyopic strabismus to begin with. And then when you correct them, how does it change their cortical activation? And those are so many low-hanging fruit that you can analyze with this particular techniques. Okay, so thanks so much for your time. This is a thing that I find interesting, but I don't think I have any time, do I? No, it's fine. Thank you. But nice to be here and appreciate your time. Questions? Or do we take questions now? Or? Okay. Thank you.
But there's one piece in the middle because we're jumping from the eye to the brain. All the brain is great, especially if it's a the one piece that's missing here is the
Yeah.
because in this
At the level of the LGN? Yeah, I think so. And I think this is where we have. Oh, sure, yeah. So Alex is mentioning that we're kind of jumping from the eye all the way to the cortex, and the LGN, lateral geniculate nucleus, is obviously an important place where processing is occurring, there's parsing of information and so forth, and asking whether this type of imaging can help resolve changes at the level of the LGN. And I believe it can. I think essentially, because we're actually seeing changes at even smaller structural resolution than what you'd require at the LGN level, but we haven't got to the point yet where we're able to make those, clinical assertions. Now, when I was at SC, I think many of you may know, and I'm sure Sarah does, Arthur Toga, and they had created this really amazing center there, and they had dropped this 7 Tesla unit through the to the roof into their thing. And one of the goals was to study hippocampus, right? And essentially that's obviously important in neurodegenerative disease and so forth. And the LGN and the hippocampus have a very similar shape and size. And the thinking was that many of those protocols that they've developed for hippocampal evaluation could be used for the LGN as well. Because subcortical, same size, so forth, layers. And I think so, we just have to utilize the resolution that's there. Anything else? Cool. Please.
Very nice presentation, Dr. Patel, as always.
Thank you.
And I loved very much the way you correlated the morphology changes in the brain with those in the retina. And you started with this recent, let's say recent in the related to the onset of the condition. So recent changes in morphology in the inner retina in the retrograde fashion starting from the brain. My question is, do you have any evidence of changes that are later? Because the retina is a neuronal circuit, so retrograde changes starting from the inner retina towards the outer retina with the photoreceptors are exactly in the same fashion as from the brain to the retina. Do you have any late, very late, like autofluorescence images, OCT images to see if later in the dynamic of the condition, the photoreceptors are affected as well?
That's a good question. Well, that first case, the traumatic maculopathy, that was a 9-year duration. And so the studies were done 9 years after that person had the trauma. So I think that's a good point, that the autofluorescence changes and the changes that are there in the outer retina were longstanding in that situation. And those downstream effects were seen at the cortical level. And I picked those 2 cases because one was relatively recent, which was the optic neuropathy at 6 weeks, and this was 9 years for the maculopathy, but we got very similar findings at the level of the cortex.
Very nice, thank you.
Welcome. Thank you all.
All right, next I would like to invite up our next speaker, Dr. Kimberly Jamieson. She is a cognitive scientist conducting her research at the Institute for Mathematics, Behavioral Sciences here at UC Irvine. She's an affiliate faculty of our Center for Translational Vision here at GHEI and is also the director of UCI's Color Cognitive Laboratory. I would like to invite her up to give her talk on human color vision and perceptual psychophysics.
Good morning. Thank you for inviting me to participate in this meeting. Can everyone hear me okay? Okay, good. Okay, I'm a cognitive scientist, and my main interest is in understanding, since about the 1990s, how retinal genetics affect color perception in normal human observers and in observers who have genetic mutations in the photopigment opsin genes. The particular genes I'm interested in looking at are X chromosome linked. The assay that we do for the assessment of the opsin genes involves assessing, of course, the rod sequences and the S-cone sequences, which are on different locations, different genes. And X-linked chromosomes are recessively inherited. So we typically are looking at 3 populations of photoreceptors, and that's the standard model for normal color vision observers. Obviously, there are normal color vision variations that are associated with the X-linked photoreceptor variants, and these reflect variations that occur as severe as dichromacy, which is protanomaly and deuteranomaly when you have, or when you have protanope and deuteranope, when you have missing M and L cones, or anomalies where there are shifts. Like, for example, in this slide, there's 2 forms, excuse me, got to find the pointer. There's 2 variants of L. The color reflects, of the different types of cells, reflects where their peaks are in the electromagnetic spectrum. The normal model, though, is considered to be this 3-cone model. So we know that these give rise to anomalies and deficiencies, as simulated here in these pictures by Vinoy from 1995. And we also know that there is naturally occurring mutations that are happening on the X-linked photopigments that give rise to different phenotypes. And one particular phenotype that we've been studying since the 1990s has been a phenotype where you have a SNP occurring in exon 3 at codon 180 of the L cone. And this has the property that it gives rise to a different phenotype compared to the standard normal phenotype. Standard normal phenotype is here on the left, and the different phenotype that arises as an obligate expression of X inactivation is shown on the right. The difference is there's an extra photoreceptor. The question is whether this affects perception. So when we do our photopigment ops and genotyping, this allows us to look at the front-end processing constraints that are present in the retina and to see whether or not those actually affect a number of things, including photoperception, including color perception variation. And I came to UCI in 2001 with a technique that I had been using at UCSD in medical genetics with Dr. Linda Wasserman. And I was very lucky to find Chris Kenny and Sherry Adelano, who helped implement the method here and improve it greatly. The method basically uses the idea that we look for SNPs that occur at different loci on the X-linked L and M opsin genes. And those are responsible, respectively, for sensitivity to reddish lights and greenish lights. And we look to see whether these SNPs, when present in the genetic array, give rise to phenotypes that affect color perception. We already know in the case of dichromacy they do. The question is, can they explain normal variation in color vision? This is possible because the L and M opsin genes are extremely homologous in their sequences. They're about 98% homologous. And there's really only 18 sites where they differ, and 7 of those sites are known to control or impact spectral sensitivity of the cone classes in the retina. And this has been known since 2004. So one of the things that we can do as a psychophysicist is I can ask whether or not front-end processing variation that arises from these genetic SNPs give rise to different color experience. And this is something that I talked about at the Vision Mixer back in February, and I also talked about this type of result at the 2019 Bench to Bedside. But basically what we can do is we can ask whether or not the sunset image, given 2 normal observers, but one who has a genetic SNP at codon 180 and exon 3 in the L cone pigment, whether or not it gives rise to different color experience. And we have found through hyperspectral image analyses that we can empirically identify with, for example, cone contrast thresholds, psychophysical measurements, the sensitivity of each of these classes in these different subjects, a standard normal trichromat and an obligate expressor of these 4 pigment retinas. And we can ask whether or not if we do a subtraction of the pixel by pixel image strength for these standard normal observers compared to one expressing an extra photopigment, whether or not that gives rise to a different image, a different perceptual experience. And in this particular instance, this heat map shows that we can identify 66% of the image pixels that look different for this person who has normal color vision with the extra pigment class compared to the standard trichromat model. The way we do this is look at a number of techniques: heterochromatic flicker photometry, minimum motion and isoluminance, which I talked about back in February at the Vision Mixer. We also use cone contrast threshold assessment, which is a more established method that's used in the clinic, actually, and it's used by the United States Air Force to screen their pilot candidates. And what this is basically finding out for these cone isolating stimuli, 3 separate types of experiments, where the subject can identify the gap in the Landolt C. So you see a gap on the left for the L cones, a gap at top for the M cones, and a gap at right for the S cones. And the question is, if you vary the cone contrast relative to the achromatic background contrast, that how far can subjects go to identify variation? So here you see, it may be very difficult for you to see the cone contrast down here at this level, but up here it's quite obvious, okay? So we run a bunch of psychophysical trials, lots of observations, and we find for the different subjects what the values are for each cone-isolated cone contrast, and we are able to diagnose them essentially and their color vision capability. Those are quantitative numbers that we can use in models to do things like create that approximated image that I showed you earlier. Now, one finding that we note is that this group that we studied, the L-cone 180 heterozygous females, in these types of experiments, like cone contrast threshold, are about 25% more sensitive than than the cone contrast thresholds typical for age-matched controlled normal trichromats. And this isn't for all exon 3 codon 180 SNP participants. It's for a subset. It's not a universal finding, but it's something that we are studying to try to find out what differentiates some of the participants who carry this genetic potential from the others. But we do find some that are having more sensitivity, if you will, to the spectra in reflectance spectra that is dialed into their extra photopigment class, which is an interesting finding. And this has been demonstrated psychophysically a number of ways now by several different labs. And we can also generalize these cone contrast threshold ideas to the clinic, and that has been done with Chris Kenny and led by Dr. Andrew Brown and other colleagues here at Gavin Herbert. And we have looked at it at different clinical populations and found that it's a useful measure psychophysically to look at. And in particular, it helps us appreciate, for example, AMD patients who are at the stage where they have intermediate or worse disease processes, and they have actually reduced color processing cone contrast thresholds, as shown by these plots here, where you have intermediate advanced AMD and neovascular AMD, and these shapes for the L cone and the M cone and the S cone are obviously quite different from the healthy control profiles. So this is an index or a way of psychophysically assessing these patients, to learning what their visual impacts are from these disease processes. And these are the types of color metrics that haven't really been used a whole lot in the clinical literature, but they may be useful for us to appreciate different clinical populations and the properties of their perceptual experience as their disease progresses. Now, I want to take a minute now and just say how I came about to be here in the Gavin Herbert Eye Institute. In 2013, when I saw the intercampus call for the opening of the Gavin Herbert Eye Institute, I was looking at that time for a colleague to work with me in medical genetics to do this opsin sequencing. As I said previously, I already had it, was working on it at UCSD. When I moved here back to UCI, I needed a colleague, so I ran over to the Gavin Herbert at the opening, and I introduced myself to Dr. Steinert, and told him what I was interested in, and he said, "You need to talk to Chris Kenny." So he walked me over to Chris Kenny, and I started to explain to her what I was working on. And the first question she asked me was, would you suspect a relationship between AMD and photopigment opsin genes? This is the area that Chris was working on for a very long time, looking at the mitochondrial DNA relationship to AMD risk and severity. Why might we suspect a relationship between AMD, photopigment opsin genes, and color perception? Well, there's lots of reasons, actually, but the ones that first came to mind when Chris asked me the question is that we do know that the population that's most likely to possess these opsin gene SNPs for the X-linked photopigments are the ones who are also at greater risk for AMD. Those people of European ancestry and Caucasian ancestry and of a particular mitochondrial DNA haplogroup. We also know that the focal region of AMD deterioration is the region of the retina where the X-linked photoreceptors are more dense, and the prevalence of AMD is greater for females compared to males, and females are the ones who have two X chromosomes who are, when they have X inactivation expressing the phenotype, they're obligately expressing alternatively these different alleles, which means that they have more opportunity to express the extra pigment classes that are mutated. And so we know that the fovea is in the center of the macula, and we know that the highest proportion of L and M cones, those X-linked photopigments that have the mutations, are also in the fovea. Okay, so we look at this statistically to ask the question of whether or not we can find a relationship between AMD and opsin gene mutations that are different from the reference sequence that we see in the wild type. And so we for each participant that we look at specific opsin gene loci that are of interest. They're of interest for a number of reasons, not the least of which is they affect the spectral sensitivity of the photoreceptor types when they're expressed in the retina. And we compare those data to AMD features like type of AMD, wet or dry, properties, severe, binocular, monocular, things like that. And what we've done to date is we collected recently additional subjects. We originally had around 135 subjects, and we got sort of hamstrung by some various events in life. And by last year, we had 221 subjects, 124 AMD, and 97 controls. And what we do is we look at this statistically, but sometimes because of the complexity of the categories in these matrices, you saw I have lots of types of AMD, lots of types of SNPs, and on the opsin gene, we run into a computational problem with the statistics where we don't have the power to find the associations that we think should be there. So one technique that we can use, and we've explored in the past using, for example, gradient boost models and other kinds of models, is that we can look at machine learning to ask whether or not the kinds of decision trees that you would use to predict whether or not AMD and opsin gene SNPs are associated, whether or not those trees can be looked at iteratively using machine learning to find out if there is an association there through the machine learning indices. And we can talk a little bit more about the machine learning if you want in the question and answer section. But basically, using random forest models analyses, what we're looking for is classification accuracy. What that means is we're looking to see whether or not we can classify accurately from this large data set whether a patient is AMD or whether a patient is a normal control. And it turns out when we look at variables like all the SNPs that are on the L and the M opsin, and age, and sex, and mitochondrial DNA haplotype, we find that we're already classifying AMD from normal at about 71% accuracy, which is pretty good because 80% is sort of what you want to achieve in a data set like this with a machine learning model. We find if we remove mitochondrial DNA haplogroup data, that the classification of AMD from normal drops to 64% accuracy, which is simply a trend suggesting that having the mitochondrial haplotype data in the data set is one further boost that allows us to quantitatively classify AMD from normals. So mtDNA haplogroup information is important for differentiating opsin gene, the role of opsin gene SNPs and normals from AMD, and it improves the accuracy of differentiating AMD from normal using option gene sequence data. Which mitochondrial DNA haplogroups are indicated in the feature importances of the random forest model analysis? Well, the ones you would expect from the literature. We find that H and HV surpass our threshold of importance, indicating that they are protective and help us discriminate between AMD and normals for that reason, and J and K are not protective, and they help us discriminate between AMD for those haplogroups. And we also can apply random forest modeling analysis to tell us about specific SNPs on the 2 genes that we are considering. And we look at all the SNPs that are possible as candidates, including age and sex are on this grid as well. And what we find is that these SNP features, some of them exceed the mean decreased impurity threshold that mean they are important for differentiating AMD from normals. If we collapse down the one that is appearing at codon 180 right here, just to look at the global influence of that particular SNP, we find 2 SNPs pass the chi-square test analysis to indicate that these 2 SNPs, codon 153 on the LW sensitive gene and exon 3 codon 180 on the L sensitive gene, pass the threshold for being important for predicting and differentiating AMD from normal patients. And you should note that these particular genes have also construct validity from the point of view of perception, because this is the one we were studying that showed the perceptual difference, and this is one that gives rise to an optical density heterozygosity in the retina, which we also believe to be important because of potential variations, as Dr. Farrington would say, in these particular 2 loci being implicated in affecting the metabolic ecosystem that might exist across the lifespan for the phagocytotic processes that are involved in the daily cleanup of the turnover of these cells. So the model that we have working now is that if we can do these biochemical analyses of these different kinds of alleles and see how they change the amino acids that are present, that we can actually look at the byproducts of what is happening as they are processed through age. And this is something that we might be looking at in plasma soon with another colleague. So summary, our investigations to date suggest an association between AMD and X-linked photopigment SNPs that are known to impact retina phenotype and perception. There's good theoretical basis as well as a practical rationale for why X-linked photopigment SNPs might play a role in age-related drusen formation. And moving forward, we hope to continue Chris Kinney's vision on this issue to increase sample sizes with more AMD patients and more normal controls, and work with these kinds of analyses to separate wet and dry AMD groups and severity levels. Thank you very much. I would just like to acknowledge my project collaborators. Chris Kinney, I've worked with for more than 10 years. Sherry Adelano as well. The heavy lifting for the genetic assay stuff is Sherry Adelano's creative work. And more recently, I've begun working with Mustafa doing the machine learning portions. Thank you. Sorry.
Thanks. I'm Christine Curcio. When the Knights were looking at variation in opsin back in the 90s, I wondered why. But now it's a lot clearer to me, and that's because we know about subretinal drusenoid deposit, which is an extracellular deposit between the photoreceptors and the RPE. And for a bunch of reasons that I can talk to you about at the break, we think that they're made up of lipids that probably originated from the outer segments. And so now I could believe totally that you get some sequence variant in an opsin, you're going to get different propensity for that kind of deposit. So I'm wondering if you have phenotype information on your AMD patients that might shed some light on that question.
Well, so the question is, can we predict lipid interaction in the outer segment from A&D demographics or clinical diagnosis information for the SNPs, the SNP question? We don't have that because that's not necessarily what the patients were engaged in the study for. They were mostly engaged to give their DNA to isolate their mitochondrial haplogroup. And, well, I have to look into that. And I hope Andrew Brown's here because I do want to ask him if we can get at the plasma or some other DNA that we need to incorporate some more subjects. So that's a really excellent suggestion, and I think we should look at that. Thank you. Thank you very much.
All right, we're going to keep on moving. I'd like to invite up Dr. Cheryl Ng next to speak. She's an assistant professor here at UC Irvine within the Department of Cognitive Sciences. She earned her PhD in X-ray crystallography from the National University of Singapore. Here at UCI, her work focuses on visual sensory integration, where she combines computational modeling and physiologic optics to further our understanding of visual perception and how the brain interprets visual information. I'd like to invite her up to speak.
Thank you everyone for having me. So let me try to get myself organized here. How do okay, great. Okay, so this is really a collaboration that is a cross-country effort. So what I'm showing you here is a picture of the Niagara Falls in upstate New York, where this collaboration, cross-country collaboration, very first started. But the reason why I'm showing you this picture is to get some definitions in place so that we are all on the same page. So when I talk about orientation, I'm really talking about how these streaks are angled. So for example, if you see here, these ones are almost vertical but slightly slanted towards the right side. When I talk about motion, I'm really talking, when I talk about direction of motion, I'm really talking about the trajectory of an object translating across space. So now let's come to what I'm going to talk about today. Oriented blur biases the perceived direction of motion. That's really quite a mouthful. So let's just summarize this. And how do I move forward? Sorry. Oops. How do I move forward? Oh, here. Okay. Let's just summarize this and say that my talk is about how astigmatism causes people to see weird movements. So what is the relationship between orientation or astigmatism and motion? Well, here, as we know, our perception of orientation really depends on motion. Here, I'm just going to move this tiny little dot across the screen, and you see nothing much happening except the dot moving. But as soon as I begin to speed it up, you see just by virtue of making a move, we see a smear or what we call a motion streak or motion blur. So, and you could have already experienced this yourself when you're in a car and zooming by, you see everything in the world looks horizontally stretched. So perceived orientation depends on motion. Likewise, I can also say our perception of motion depends on orientation. Here, I'm just going to do a little demo and ask you in what direction is this line moving. All of you would say down and to the right. But really, if I open up the aperture, you would see that the line is actually moving horizontally instead. Another one. This line, of course, appears moving downwards. But as soon as I open up the aperture, you begin to see that this line is translating horizontally across the page. So what I'm trying to say here is just as motion can bias the way our brain interprets orientation, orientation in turn also biases the way our brain computes motion. And there has been many studies showing that motion and orientation are highly interrelated, which takes us to the purpose of this talk, astigmatism. One of the most common cause, if not the most common cause, of oriented blur in our eyes, in human vision. So to a person with the rule of astigmatism, okay, here we go. Sorry about that. To a person with the rule of astigmatism, you can see how the world looks horizontally stretched. If you have diagonal astigmatism, you would either perceive things to appear stretched out like this. Now you have a bias for orientation in the diagonal here like this or like this. But whatever the case, the point I'm trying to make here is that notice the orientation of your blur goes along the power meridian. So because we know that orientation and motion affect each other, now the question really becomes, does astigmatism bias the way we perceive motion? Because now our orientations are all messed up. And I think this is an important question to ask because motion is all around us. Many of us have astigmatism, and motion becomes especially hazardous when people with astigmatism start driving at night at high speeds. And it's already been shown that astigmats have problems at night with driving, presumably when the pupil size increases and the effect of astigmatism becomes bigger. So it's an important question to ask, even though astigmatism is such a common issue, even though blur is only restricted to one orientation. Okay, so now how do we proceed with this? First thing we did was to recruit our participants, and we had 5 people to participate in our experiment. We covered up the left eye and tested motion perception through the right eye. You see that our participants came in all sorts of flavors, mainly low myopes. Some of them had astigmatic correction. Most of them did not. We had 1 hyperope, 1 ametrope. But in all these cases, you see that there is some residual refractive error left over. It may be because that they were being undercorrected by the eyewear. It may be because of more complex refractive errors that the eyewear cannot address. But whatever the case, these leftover refractive errors give rise to blur, natural blur that all of them have. And you can see, this is a point spread function, how a single dot of light spreads out going through the anterior segment of their eyes. And you see here that many people come in all sorts of different shapes. All the blur are in different shapes, some more astigmatic looking, some more symmetrical. So before we could run our experiment, we first had to deal with all these different natural shapes of people's eyes. And the way we did that is to use adaptive optics. Many of you would know adaptive optics as a technology that's in use in imaging, but I use it for a totally different purpose. I use it to measure perception. But even with this application, the principle is still the same. We have a wavefront sensor sensing all the way that light distorts going through the eyes, the anterior segment. This wavefront sensor talks to a deformable mirror, where that is able to put on a complementary shape in real time to negate all the distortions, so that now you get a very nice, sharp focus of images on the retina. So here, I'm going to show you how this residual refractive error decreases to a near zero as soon as I switch on the deformable mirror. There we go. So now we have a pair of magic eyeglasses, and it allows us to give people perfect vision. So where do we go from here? All while that is still happening, we induce people, our participants, with 1 diopter still power either in this axis or in this axis. Now we have a very nicely tight control experimental condition where these round, nice-looking dots become stretched out either in this orientation or in this orientation. So now we are able to measure how do people perceive motion with these types of oriented blur. And the way we did that was really just to present a bunch of dots that is moving along the screen. We tested 3 factors, the first one being coherence. So coherence is just the number of dots moving together. 80% coherence, 80% of the dots moving together in one direction. Half the dots moving here together in one direction. And in this case, all the dots are moving randomly. We also tested the effect of direction. So there can be motion going upwards, there can be motion going diagonally or horizontally, really in all directions in increments of 45 degrees around the circle. We also tested the effect of speed. So there can be slow movement, medium, and fast movement. And you would already figure that slow movement creates very little motion streaks. Medium movement, a little bit of streaking. Fast movement, a lot of streaking. So all that conditions, that's a very big, wide array of things to test. Let's just break them down. And first question we ask, how did people with astigmatism perceive slow and highly coherent motion? Now here I am showing you a movie of 80% of the dots moving in this upward direction. And at the same time, I'm presenting still power in a 135 degrees axis. Remember that blur goes in the power meridian, so the blur is along this orientation. After looking at the movie, the person's task, our observer's task, is to turn the dial towards and point towards the direction that they think the movement is. And that's what I'm plotting out over here. This is the result from one observer. And the color of the data points represent the direction of the true motion, the actual direction the dots are moving in. The position on this clock face indicates where the person turned the dial to. So in most cases, this person got this trial correct. They were seeing upward motion as going up, but occasionally they got it wrong where they saw this upward motion as going 45 degrees or 315 degrees instead. This was how this person did with a diagonal movement, left to right movement, and all other movement. What I'm going to do now is to superimpose all the results of all different subjects, and each subject is color-coded. Now you begin to see two observations. First observation is that people were accurate in making movement judgments along the orientation of the blur. So when we presented orange and blue type direction of movement, you see that all of them got it correct. Next thing you begin to see is that people were getting things wrong in the orthogonal to the blur. So over here, you see where the dots were, where the data points are supposed to be populated by only pink and green, you see that there is a smattering of other colors. What this means is that people were misperceiving, for example, downward motion as going up and to the left instead, and people were misperceiving, for example, the red ones, upward motion as going down and to the right. So accurate perception along the blurry orientation, inaccurate motion perception in the orthogonal. And this is not an occasional artifact. This is not just mistakes that people were doing. I could switch the blur over to the other orientation. So now I'm presenting 45 degrees still power, and now blur is going in the opposite instead. You see here now that, again, people were accurate in this along the blur, where they once were inaccurate with the other astigmatic, the other axis of astigmatism. And here again, now you see how people are being inaccurate in their perception, orthogonal to the blur, even though they were accurate before in the other condition. Remember that these two participants are not two different groups of people. They are the same group of people. So it must have been that astigmatism is just messing up their motion perception. There we go. And I can even quantify this effect. I calculated or measured calculated accuracy along the blur and in the orthogonal to the blur. Accuracy is basically just defined as the number of times people saw motion for what it truly was, like in this case. So obviously you see here that greater accuracy is found in the blur, along the blur orientation, and people were not so good in orientations that were in the orthogonal. Okay, let's now just switch gears and talk about how people perceive fast and incoherent motion. So this is a case where the dots are all moving randomly, and you think that if the dots are all moving randomly, people's responses will be all over the clock face because they would be perceiving random motion. Now here I am presenting them with 135 cell axis again. And here you see, instead of perceiving it random motion, people were all biasing the motion in the orientation of the blur instead, which is really quite intriguing to us. You could swap the axis of the astigmatism. Now you see blur going in the other orientation, and now a huge cluster of data points fall in the other orientation. And this is not an occasional effect. Each subject is symbol coded. Multiple instances of the same symbol indicates multiple instances that we presented this random motion to them. You see that for all participants with every single presentation, they were perceiving motion along the blur orientation. So again, we computed the number of times when people saw motion along the blur and in the orthogonal to the blur. And again, we get a huge effect over here. So conclusion, we have biased percepts of motion that is biased to along the blur orientation. So I have just talked to you about two extreme conditions, highly coherent slow motion that's seemingly easy to do, and highly incoherent and fast motion that's seemingly difficult to do. So what happens in between? First thing you notice is that people are much more accurate at the mid-speeds. So you could decrease the coherence level, the number of dots moving together, and you see that the colors are still clustered and very nicely segregated. People were bad at the slow motion and the fast motion, but you would see that their mistakes were going opposite to each other here. With slow motion, you would see that they were making more misperceptions in the orthogonal to the blur. With fast motion, you see that they were biasing their perceptions, the direction of motion perceptions, along the blur instead. So what's the conclusion we can take home from this? Astigmatism gives rise to biases in orientation that causes your motion to be messed up, your perception of motion to be messed up. If you're talking about slow and coherent motion, you are accurately perceiving direction along the blur, and you're inaccurately perceiving along the orthogonal. With fast and incoherent motion, you are just biasing your percept to be along the blur axis. So how do we explain this? One possibility is that we are mistaking the astigmatic blur for a motion streak. So in a fast condition, motion streaks are a cue, and so people think that this blur indicates motion along these directions. However, it's not a cue at slow speeds. There is no motion streak at slow speeds. So people are not going to use that blur as a cue for motion. Instead, our percepts are being repelled in the opposite instead. So we have other cool experiments that we did with our adaptive optic system, but I want to stop here. I'd like to acknowledge my team, my former team at the U of H, people who are involved in patient care, people involved in subject recruitment and paperwork, my former advisor, Gun Young, and people involved in the technicalities of AO. So thank you for your attention.
Good talk, Cher. Thank you.
Thank you.
I have a question. Do you know if illusory motion has the same types of effects? No.
No, I don't know. This is just one of the preliminary studies that we did just before I left Houston. So there's a lot to be done over here.
So it may be that minimum motion at isoluminance, for example, may be different in astigmatic patients.
Maybe, we don't know.
Yeah, okay.
That's a good point. And besides that, we also have something called chromatic aberration.
Right.
So yeah,
Yeah.
that's something
Cool.
to be tested.
Okay, thank you.
Hey, Cher, beautiful work and wonderful presentation. So obviously all of this works with one eye occluded and only the other eye. So if you have binocular vision, presumably astigmatism will not be identical. So what is your comment on how this will work with when both eyes are
Exactly.
engaged?
We don't know. Well, the thing is, if you have with the rule astigmatism, then probably you would be the same in both eyes. But sometimes people have diagonal astigmatism. We know it's different in both eyes. That's one of the things in the pipeline. We have a binocular adaptive optic system that we can. I'm actually trained in binocular vision, so that's one of the things that we want to do. Yeah, thanks. Exactly.
Yeah, to be big. So I gotta imagine what spot size is played in that would be protocol.
Exactly. So the question really is, why do we use even this kind of global motion? So there's two reasons. One reason is that it's very easy. It's very easy to create streaks. Second reason is because if you increase the spot size, then we're talking, so there are two stages of perception, right? One stage when you're extracting info, when your V1 in your retina is extracting information from your retinal image. And then after that, you build it up again in the extrastriate cortex into your perception of the world. With this type of stimuli, we can test for what we call global motion, which is what is happening in your building up stage, your extrastriate cortex. If you increase the spot size, then you're talking about local motion during the extraction stage, which we do not want to test out here. But you have a good point that objects in the world are also big. So obviously, we have to do something about this. Yes, so that is something else that we have in a pipeline to test.
Very nice presentation.
Thank you.
When you calculated the percentage of coherence, how did you translate? So the patient answered in which direction was the movement. So how did you process this answer to get the final coherence in 80%, not 50%?
So how big is the effect, you're asking?
Yeah, you expressed coherence in percentage.
Yes.
And I saw a scale from 0 to 100. And at one point you said this group or patient has 50%, this group has 80%. So how did you process the answer of the patient? This is the direction of movement.
It's all
in
measured.
Percentage.
It's all measured. So 80% coherence is what's shown on the screen to the patient. Everyone had the same 80%, 50%, 0%. It's whatever that's presented to the patient. All the patient needs to do is to turn the dial, and that dial can be in increments of 1 degree. So it's very graded. You can change your responses.
So if you have non-parametric, the comparison cannot be done with t-test or whatever.
No, I didn't do any statistics on this.
Okay.
Yeah.
Great, thank you.
Okay, share one more question. And this is about, I'm here.
Oh, hi.
Hi.
Sorry.
Wonderful, intriguing talk. So I was just thinking if the experiment would be possible in mice with some optokinetic measurements of the eye movement. And if you could put your glasses with astigmatism on the mouse and see what is happening with the learning. If after, let's say, month or couple months, if the mouse, the blur and the motion is changing.
Exactly, exactly. So that is one thing that we want to test. Well, I didn't think about in mice, optokinetic drum, but I thought about in patients. Like a lot of people have astigmatism. I don't think many of us are so bad in the world because otherwise we'll be all bumping into things, right? So there is some neuroplasticity going on, some adult plasticity going on. So that's one of the projects that I want to test too. Like how do you take people with natural astigmatism, do they still generate this effect? If you remove their astigmatism artificially by either giving them eyeglasses and correcting them immediately, do they show an after effect of the overcompensation? So that's one of the things that I want to test. Thank you. Okay.
All right, we're going to shift from blurry images to hopefully very clear pictures of the retina. Our final talk this morning is going to be given by Dr. Yehua Zhang, who received his doctorate in precision instrument engineering and optical engineering, and is currently a professor of ophthalmology at the UCLA Doheny Eye Institute, where his work focuses on the development and use of advanced ophthalmic imaging to further our understanding of the retina.
Thank you. Hello, everyone. It's a great pleasure to present you my past year's research and also we have a pending patent on the development of adaptive optics, macroscopy, and ophthalmoscopy. First of all, I will give you a tour about adaptive optics and show you how this technique helps high-resolution retinal imaging, and we utilize this technique to study choroid retinal disease and also study the retinal hemodynamics. Adaptive optics originally developed for astronomy imaging. Oops. Here is showing here when the star from deep space comes through the atmosphere, get to the telescope, the image is blurred. The reason is that the wind or the change is in the atmosphere distort the wavefront. The trick here is that we apply a device called a wavefront sensor and the wavefront compensator to look at this here. And after AO, that means the wavefront is flattened, and so the image is cleared. Similarly, in the imaging human eye, we shining a light into the eye. The light coming out from the retina or from the eye is distorted by the human eyes, the optical defect, and causing the image blur. Similarly, we put a device, a wavefront sensor and a deformer mirror, and we take off the blur, and we get a clear image showing individual cells. So the two components, just for IO, is the wavefront sensor and the wavefront correct. I won't go to technical detail, but just showing the triple benefit of adaptive optics. The first thing, if you look at the image taken without IO, it's a blur and showing dark. So that means, first of all, they increase the brightness and also improve the contrast, enhance the resolution, showing the smallest retinal cells in the human living eye. Here's the slide showing the foveal center cone. This is a very foveal center, and then this is about 2 degrees, about 600 microns within inside the eye. So the smallest one here, pretty much 2 microns, and away from the foveal center, the cone become bigger. And further away from here is pretty much 6 degrees away from the foveal center. We see the cone surrounded by the rod. And we applied adaptive optics. Oops, sorry. Oh, sorry. Yeah. Can we play the movie here? Oh, it's fine, because basically there are 2 major instruments, imaging modality. First one is the scanning laser ophthalmoscope. It's scanning the light, use a laser, and collect the light from the eye, just through a pinhole, forming the eye-fast image. Another one is optical coherence tomography, the OCT, utilizes the interferometry of the eye, and they get the cross-section of the image. Here we developed the adaptive optics scanning laser ophthalmoscope and the optical coherence tomography, and we imaged the eye-fast image that's the foveal center, and also the inferior 1 degree, 16 degree away from foveal center. With the smaller dots, they are rod, and they are cone, and this is showing the cross-section of the retina. Compared to the commercial OCT, the difference of the AO OCT is that it resolves laterally the individual ellipsoid zone of the cone photoreceptor. Also, with back to 1990, Dr. Kershaw published a seminal work about the human photoreceptor topography, really established our understanding of the fundamental histological structure and the cone rod and the distribution in the human eye. So we are very pleased to replicate, and her guidance, we have replicated her work in the living human eye. So actually the shading area is from the histology study, and here we measured the cone density in the human eye, that's superior to inferior, and the temporal to nasal direction. Also, now with this technique, we can study, I will show you how to study this, the age-related macular degeneration. The AMD is a leading cause of vision loss among the older adults. It affects 10 million of old Americans, each third largest cause of the vision loss. It impacts daily activity, particularly in the macula, because that's really rich in cone. That's working for our fine vision and color vision. It's a complex disease. It affects photoreceptor supporting system, but eventually affects the photoreceptor. So this is why adaptive optics can play an active role in studying this disease. So, because for this, for the best way to prevent the late AMD is treating retarding or preventing early AMD. But for such a complex disease, where we start, actually, I started to study the drusen and the pseudo-drusen back to 10 years ago. The drusen is a hallmark of AMD. From the OCT, we can see it's and prosthetic RPE, and also pseudo-drusen is a newly appreciated independent AMD risk factor. Dr. Kershaw and Dr. Spade, they work together and combined with the OCT and histological study, and they come out a conclusion, suggest that the pseudo-drusen SDD. The SDD is the histological correlate in the living eye. So working with the wonderful mentors, one of them actually are sitting here today is Dr. Kershaw. I'm so grateful to their help, and we hired the patient, and we performed we studied the SDD's impact on photoreceptor in AMD. Basically, we had the patient with a different degree, stage of AMD, and we examined their best correct visual acuity, and we performed multimodal imaging, including color-founded photo, SD-OCT, and reflectance, red-free autofluorescence, and we performed AOSL plus OCT. So this is the color-founded photo near IR blue reflectance, because we see this intertrace, that's the pseudo SDD, and also this is the near IR image. And take a OCT, get the two, look at these two entities. That's the two bigger SDD. And we scan this area with a high resolution AOSLO. We review the cells and the very distinctive hypoannulus surround a hyper core, showing some cell-like structure. And with the adaptive optics, we characterize the different stage AMD impact on photoreceptor. Here we can see the stage 1, a healthy eye that's evenly distributed cone and rod. And for the stage 1, that's a little bit of development between the EZ and the RPE. Photoreceptor reduce the reflectivity, and with the increase of the subretinal druthenoid deposit, the reflectivity further reduced. Interestingly, we go to the stage 3, when lesion broke the EZ band, we see this distinctive feature showing this kind of weird structure. Initially, we thought that these cells, but once we performed AOCT across this lesion, we found that actually these cells, they are not cells. Actually, they are lesion materials here, because we can clearly show. Now we demonstrate how adaptive optics SLO, we acquire image in eye fast and the cross section. We actually get the 3D high resolution image. And also we follow the image, same patient over 30.5 years. We can see the SDD are dynamic and have a life cycle. That means they can expand, shrink, and also reduced, and this one from stage 2 becomes stage 3, and this the tiny one become even bigger. Also, AMD imaged the RPE degeneration associated with the SCDD. We found it very interesting, the hypertransmission band beneath summer SCDD. But this hypertransmission actually doesn't correlate with the size of the SCDD. But with the high-resolution retinal adaptive optics, we found actually they are associated with the regressing SCDD. That means with high-resolution image, we clearly imaged the change of also the regressing of the stage 3 SCDD. We show the hypertransmission. We know that one of the important role of the RPE is just to block the light and absorb the scattering. But now the light penetrates the corridor. That means it's definitely something wrong here. And we also studied this phenomenon with AMD's other risk factors, for example, photoreceptor length and corridor thickness. These hypertransmission are very well correlated with those factors. So this we are thinking this is really an indication of RPE degeneration. We also adaptive optics image the intraretinal hyperreflective foci inside the eye. The hyperreflective, the IHF actually was thought to the strongest predictor for the development of AMD. The high-resolution retinal imaging we disclosed here is that it shows the photoreceptor mosaic actually was distorted or damaged. So also we show that some cell-like structure corresponding to the RPE spacing. We are guessing there might be the migrating RPE cells towards the inner retina. Histological study actually has suggested that the major part of the hyperreflective foci actually constituted by the RPE cell, migrating RPE cells toward the inner retina. Also, we imaged the cone photoreceptor loss in the clinically non-remarkable area. Actually, we know that the lesion damaged cone photoreceptor, but what about the cells in the unaffected or clinically non-remarkable area? Adaptive optics found that AMD eye with SCDD exhibited accelerated the cone decrease compared with those eyes with AMD, but without SCDD. So just to summarize, the AO imaging disclosed the ultrastructure of AMD in the living patient. The pseudodrusen or SCDD are in the subretinal space, directly impacting on the surrounding photoreceptor, the optical property. SCDD are dynamic lesions. The RPE posterior to the RPE degenerate with SCDD progression. The hyperreflective foci has associated with a significant loss of visual function and photoreceptor degeneration in eyes with intermediate AMD. The cone loss in clinically non-remarkable area. So recently we developed the adaptive optics enhance the fluorescence lifetime imaging of ophthalmoscopy. Fluorescence lifetime is a measure of the light, the molecule excited at the excited state. So it's a measurement, it's a unique feature to the molecule and its environment. So a FLIO is an emerging imaging modality for assessing the metabolic function, because it can utilize the abundant fluorophores in the RPE and the retinal structure. But the current problem, the current FLIO problem is that it excites the whole depth of the retina. That means we really don't know where the photon comes from. We can see the fluorophores from the different retinal layers, but because the light excites the whole retina. So with adaptive optics, we can compress the light just focusing on a single layer of the retina. Also, that's really achieving a serial level imaging. That means we're showing both structure and function. So I don't go much this one. Here's showing the excitation at 473. It has high fluorescence exciting efficiency, and except the emission detecting is from 500 to 700. This configuration is exactly the same as the commercial fluid. We want to, through this study, to understand what's the clinical fluid. Here is showing the image of the fluorescence lifetime. Look at the scale bar here. That's 100 microns. So this is the photon image. If we look at the photon image, that's very interesting. We see the honeycomb structures. Actually, they are the RPE cells. So now we really demonstrate that we really can compress the light within the RPE cells. So compared to the commercial fluid, the significant difference to our instrument, our development is that we really know where those photons are from. So this is a significant finding. We are very excited by this ability. The reason is that RPE plays a pivotal role in many diseases. So also, the high resolution image we're showing the inhomogeneous metabolic activity as indicated by the autofluorescence lifetime. This is very interesting if we will compare the histological study showing the distribution of the lipofuscin and also the melanosome lipofuscin melanosome distributed inside the eye. So this is really, we are approaching the level to perform something like a EM to understand not only the structure, but the function at the same time. So here, we found that the RPE the fluorescence increased with the eccentricity here. Compare the commercial, again, compare the commercial. This is apparently not something new, but the thing is that it's new. We know it's RPE. That's the difference from the commercial instrument. Also, we show the similar, that means the autofluorescence life increased with the eccentricity and increased with the age. And so to summarize here, that AO fluid, we demonstrated a robust confocal imaging that revealed the RPE structure. So that's really the precise measurement of the autofluorescence lifetime in the RPE cells. That's very hopeful to characterize the metabolic function and the underlying autofluorescence light source. So this is could be a significant step to understand the deeper underlying AMD's pathophysiology. Finally, I give you a quick tour about how adaptive optics really help to understand the basic, not only the basic science, because we look at the blood flow here, that's the very foveal center, there's the foveal avascular zone. Here, that's showing all the capillaries. The size pretty much 4 to 6 in diameter, and the red blood cell, the white blood cell flowing inside here. So the erythrocyte, that means the red blood cells flow, reflect the function of the retinal microcirculation. The reason is that the biological response to the hemodynamic force actually played a critical role in the vessel modulation. And also the capillary endothelium cells detect the change of the flow. That detect the function, modulate the vascular change. So that means functional change really precede the structural change. We know recently OCT is a great, OCT angiography, great device to detect the things that for the vascular, the capillary drop. But what we will ask, what happened between the vascular breakdown. So the reason is that we found a gap in our knowledge that if we, from the Navier-Stokes equation, we found that what we missed currently is the change of the velocity. That means currently that's really missed structure. This instrument, I give you a quick tour. This is high speed, 400 frames per second. We really can catch. I'm not sure how can I show you here. Excuse me, can I show you really that's showing catch the blood flow, showing individual red blood cells flowing inside the retina. And then we trace each individual red blood cells. We found, okay, wow, that's great. That's really post-tau to cardiology. Okay. So basically the high order dynamics characteristic is really sensitive than the traditional one. As I'm showing here, disclose the age and the hypertension related change. So probably I can, stop here. Basically, that's what it is. Adaptive optics is a great technique, and really it's a tool, it's an enabler to help the high resolution image to understand what's going on in the retina. Thank you.
Thank you, everyone. Let's continue the program, and it's an unusual part of the program, and it's with sorrow and mourning, but also a celebration. We want to acknowledge the passing of Chris Kenny, a cherished colleague, mentor, friend. I want to acknowledge many of the people here that are dear to her. Many of us, of course, consider ourselves great friends and colleagues, but there's some family members here, and of course, her husband, Tony Nesburn, who's on our faculty and was the vice chair of research for 20 years here, as well as Jack Schollerman, the chair of the Discovery Eye Foundation. That is, Tony and Chris and Jack were the founders of this meeting some 10 years ago. The initial idea came from Jack Schollerman of the Discovery Eye Foundation, and glad to have you here, Jack, as well. So on December 21st, 2023, the vision research community lost Chris Kenny, MD, PhD, a world-renowned scientist and clinician, and one of the key faculty members of the Gavin Herbert Eye Institute and an active member of the Center for Translational Vision Research. Chris is from New Mexico, proudly so, the land of enchantment, Santa Fe, New Mexico. There's her family home, her elementary school. Her junior high school didn't make it. It's got repurposed to another school, so it's not photographed here as things happen. But there's her high school, where she was an outstanding student there, of course. She was a competitive swimmer, straight-A student, class president, and honor society president. She began her higher education at the University of New Mexico, and somewhat bizarrely, I didn't think it happened in the 70s. I thought that's more of an older vestige. She wanted to be a pre-med student, but she was initially denied because she was a woman. But she persevered and did get her degree in biology and chemistry, then went on to earn a PhD in anatomy and biochemistry at Arizona State University in Tucson, and then moved to the University of Southern California, where she was a postdoctoral fellow in collagen and biochemistry. And then she went on to UCLA in med school. They didn't seem to have a problem admitting women. And again, strange that you would have thought this was a prior era. Still don't know what to make about New Mexico then. But again, after graduating from getting her MD at UCLA, she went on to residency at White Memorial, which is a Loma Linda affiliated hospital in LA. She was very dedicated to that work. She served as an outstanding ophthalmologist and researcher for a 35-year career, and she very much loved her work. She began her practice at Cedars-Sinai in Los Angeles and stayed there for 15 years. In 2002, the Gavin Herbert Eye Institute, at that time, Peter McDonald was chair. He then left to become the chair at Wilmer Eye Institute, where he still reigns, and he recruited Tony and Chris and their many of their group to come join us in 2002. And again, we had the good fortune and good sense, Peter did, to recruit her to UC Irvine to lead the mitochondrial research laboratory. I want to acknowledge that for all those years, I attached myself to that lab, and that became was a happy collaborator with her, and we shared that lab. I brought in the fellows, most of them international fellows, to work with her. We shared ideas, and she was my closest colleague and collaborator from a research standpoint. And in fact, as I've shared with Tony, the reason I stayed here, I was at one point thinking of leaving. David Boyer was busy trying to recruit me to go to LA, and Chris arrived, and I fell in love with the research that we could do together, and I've happily stayed ever since. So in many ways, I owe my career here at UCI to Chris, so I always think of her and miss her so much. She had great research interests. The Mitochondrial Research Lab focuses on the effects of drugs on mitochondrial genetics of patients with retinal diseases, such as AMD and diabetic macular edema. We recruited Chris to support her research interests to delve deeply into identifying the molecular and genetics changes in mitochondria associated with human disease, including AMD, diabetes, lupus, Alzheimer's, Parkinson's, and various cancers. Her work explored which drugs might target and protect the damaged mitochondria so that cellular homeostasis and longevity could be prolonged. Most recently, Chris and her lab group performed morphological, biochemical, molecular, and genetic studies demonstrating that mitochondria from patients with age-related macular degeneration are damaged and dysfunctional. Using the transmitochondrial AMD hybrids, as they're called, these cytoplasmic hybrid cells with identical nuclei from an immortalized cell line, but mitochondria removed from patients with different AMD phenotypes, she showed that were developed by Chris and her group, show that cells possessing mitochondria from patients with AMD have a significant loss of viability, impaired mitochondrial functions, upregulation of pro-apoptotic genes, and enhanced vulnerability to oxidative stress. But that treatment with humanin G, something that she's been working on, and I'll get to that in a moment, and small humanin-like peptides protect and rescues the cells. So again, Chris took a unique approach to her work using these hybrids that have revolutionized the field of mitochondrial research, showing that mitochondria have wide-ranging biological effects never before imagined, and opening up the field of mitochondrial therapy to careful investigation for AMD. Her group, in collaboration with Dr. Howard Fedoroff, pictured here, who is a former dean of the School of Medicine here at UCI, investigated various drugs and substances that keep the mitochondria healthy and ultimately improve the health of the retinal cells in AMD. In these AMD hybrid cell lines, they show that phytofibrate restores mitochondrial health and increases cell longevity. They identified a novel mechanism of action for phytofibrate that can upregulate a PGC1 alpha, the mitochondrial master gene, and significantly improve the molecular and metabolic status of the mitochondria in AMD hybrids. In addition, she and Dr. Fedoroff discovered that combining phytofibrate with E12, which is known as camphorol, prevents its metabolism in the liver and it can increase the amounts available to cross the blood-brain barrier. The discovery could have significant impact on therapeutic potential for AMD, especially as it is an oral medication. And in addition, this same approach to develop mitochondrial targeting drugs could be pursued for therapies to treat other diseases, such as Parkinson's and Alzheimer's and various other significant conditions. Chris was a highly accomplished clinician scientist with an impressive track record of imaginative and pioneering research. She advanced scientific knowledge, as mentioned, of mitochondria tremendously, earning a reputation as one of the world's foremost experts on the subject. She published more than 185 peer-reviewed manuscripts. She was the recipient of numerous research grants and awards from the NIH and private foundations for her work in macular degeneration, keratoconus, and the study of other eye diseases. She was the recipient of the University of Arizona Foundation Award for Meritorious Performance and other awards. As you can see here, she also kindly served in the NIH Visual Science A study section for many years and was its chairperson from 1998 to 2000. Chris accomplished groundbreaking work as a research director of the Discovery Eye Foundation that I alluded to earlier, founded in 1970 by Rhea and Morris Pinus. The Discovery Eye Foundation relocated its labs to Orange County in 2002 with the recruitment of Chris and Tony at that time. And Chris served as the research director and board secretary of the Discovery Eye Foundation. She played a key role in running the research nonprofit, which has contributed, amazingly, again, this is a grateful patient organization that Tony and Chris founded, but they've contributed over $15 million to the Gavin Herbert Eye Institute. I think actually over $16 million at this point. During that time, she advanced scientific knowledge of mitochondria tremendously, earning a reputation of one of the world's foremost experts in the subject. She also discovered entrepreneurship here. The research Chris conducted through Discovery Eye Institute, the Discovery Center for Eye Research, brought out her entrepreneurial streak. She co-founded Almond Therapeutics with the goal of developing mitochondria-targeting drugs for various age-related conditions, including AMD, Parkinson's, and Alzheimer's. In 2015, the Gavin Herbert Eye Institute And the Discovery Eye Foundation, again, as I've acknowledged, Jack Scholerman's effort in this, as well as Tony and Chris together, developed the first Bench to Bedside symposium. Chris took on the role of organizing symposiums, so all the symposiums to date have been organized and led by her. I think, Vladimir, you're involved with last year as well. And now with her sad passing, Vladimir has taken on the mantle, and thank you very much, Vladimir, for your help with this and for all that have done for this. But Chris was the co-founder of this meeting and the director of this meeting for all the years up to now. She was a dedicated mentor. Every student, every resident, every fellow with whom Chris came into contact was amazed by the time and individual attention she gave to them. They described her with words such as kind, warm, engaged, and life-changing. From asking probing questions to uncovering creative ways to ensure they had the resources to conduct their own research, and to mentoring countless proteges in her own lab, Chris always had time to devote to nurturing the next generation of researchers. These are some photos of her and Tony off on their many adventures, which they enjoyed to do. Chris had a fulfilling life with Tony and had adventurous spirit outside of work. Every year, Chris and Tony explored things far and near, and there they are in Africa in the upper left, Antarctica there on top, and of course we all recognize the Taj Mahal, Agra, upper right, and these are Indians, elephants in India, and then an adventure in the Amazon. And more features of Chris. She loved her dogs and her family. This is the Kenny family camping in 2023 in New Mexico. For every year for 50 years, they always camped somewhere in her beloved home state of New Mexico. And then at the bottom, you can see there several times a year she would visit the Kenny Santa Fe homestead. You can see the adobe structure there, near where she was born and grew up. Chris with her Santa Fe family in 2023 there. Again, Chris was always actively engaged in so many things. This is her every year, of course, all of us, the pilgrimage to Arvo in the upper left. And this was celebrating her birthday with Marilyn and Sherry and others from her lab. And there's Tony as well, of course. The tributes to Chris poured in with her passing. Many more than we can list here, but Dr. Jameson, who gave a lecture earlier today, in everything she did, Chris Kenny was always dedicated to the well-being of others. I think that's a profound and true statement. I think many of us would put Chris in that category. There's always more than one, but it's always like the nicest person you ever met. There can't be more than one of those, but she was in that group of people that you would say that about. She was an amazing, kind soul. Jing Yang, one of our former faculty and a co-founder of our research program together with her husband, Henry Klassen. Chris was a personal role model for me, my mentor and dear friend. Our brainstorming sessions were truly inspirational, and no one can replace her. You can see here Henry's comments as well. More tributes come in. Here from Nian Lin, Zha, Winston. Dr. Kenny was a truly selfless person. Her dedication to her work, her colleagues, and her students was nothing short of admirable. Seeing the closed door of her office every day is a sad reminder that she is no longer with us. And again, a comment from Dr. Kefaloff there as well. There's more comments here from Dr. Brown. Chris was unconditionally kind. That's a recurrent theme we see. From Sherry Adelano and Marilyn Chua, who both were the devoted technicians that worked in her lab, and they're here. I saw them earlier. Again, Chris's true legacy lies with the physicians and scientists she mentored and inspired, and how she encouraged us all to be the best version of ourselves. Chris was a remarkable scholar who forged new territory in AMD research to lead toward a potential treatment and took mitochondrial science in daring new directions. Her research integrated experts from disparate fields, including stem cell biology, neurology, and ophthalmology, with the ultimate goal of finding a cure for age-related macular degeneration. We mourn the irreplaceable loss of a cherished friend, an exceptionally collaborative researcher, and a mentor to countless individuals, including the many international research fellows at UCI, several of whom are here today, over the past 20 years that we shared. Chris was beloved by all. She had a warm and engaging personality and and was one of the kindest souls we ever knew. We hope to find many ways to honor her in our department for years to come. I'm privileged to call her a friend, a colleague, and an intimate colleague and soulmate in many ways, all the things that we shared. I miss her and mourn her, as we all do. Thank you for all you've done for us, Chris. You're up, Steph.
Thank you, Dr. Cooperman, for such a wonderful tribute. My name is Stephanie Liu. I'm on the faculty here in the Retina Division. I would like to take the opportunity to pay my own tribute to Dr. Kenny. Dr. Kenny is such a special friend. I started my residency back in 2005, and she was my research mentor. I had many research mentors in my life, in college, medical school, but she is the best of all. And I remember she just started her mitochondria research at that time, and she knows how challenging it can be to do research during residency. So she actually carved out a small section of the big research project just for me, and then she guided me through from the experiment design, actually work in the lab, work on the PCR. I still remember how dedicated she was. When I was in the research lab after hours and the weekends, Chris was always there. She's always working. And then she was always happy to see me there. And then because of her guidance, I actually won the research award for that year. Thank you, Chris. Later on, a few years later, I joined the faculty, and then she became a colleague. And then at that time, she was still a comprehensive ophthalmologist at the time, so we share a lot of patients together. She is a wonderful clinician. Not only she's a top-notch research scientist, but she's also a wonderful clinician. Patients love her. And then later on, of course, she dedicated all her time to research, so she stopped practicing. But I still see her at the departmental meetings and then conferences. Every time she sees me, she always gives me a big hug, huge smile on her face. She celebrates every single little win and every single little advancement I had in my career. She is such a wonderful friend, colleague, mentor. There are no words I can use to describe how wonderful she is. She's just such an awesome, awesome human being. And then she's always gonna have a special place in my heart. And then I miss her so much. Thank you. Okay, now we're gonna return to our scientific session of this morning. I would like to introduce the keynote speaker, Dr. Deborah Farrington. Dr. Farrington is the Chief Scientific Officer and the Director of the Doheny Eye Institute at UCLA. She obtained her biochemistry PhD from University of Kansas. After that, she finished her postdoctoral training at the American Heart Association, also at the University of Kansas. And her research team has been investigating the critical questions driving the field of age-related macular degeneration. What are the cellular changes that occur during aging? What factors tip the balance of pathology? And how does the cell respond to disease? Her research labs uses tissue cultures to investigate the underlying pathophysiology of age-related macular degeneration, and then to identify drugs that could be used for AMD. Let's welcome Dr. Farrington.
Thank you for the wonderful tributes that we just heard about our colleague and friend, Chris Kenny. And this is such an honor to give this first lecture in her honor. I named this lecture after the title of our co-authored chapter, Mitochondria, the retina's Achilles heel in AMD. And Chris and I had a lot of discussions about the name of this chapter, and we really liked the definition of an Achilles heel because we really feel like it captured the essence of what we both thought about the mitochondria with age-related macular degeneration, and that is an Achilles heel is a deadly weakness in spite of overall strength, that can actually or potentially lead to downfall. And I hope by the end of my talk today, you will also agree that the mitochondria is the retina's Achilles heel. So my relationship with Chris started here at the Beckman Center, and it occurred during the Beckman Initiative for Macular Research, which was later renamed the Ryan Initiative for Macular Research in honor of Steve Ryan. This is a meeting, a scientific meeting that is sponsored by Doheny Eye Institute, and it occurs annually. There's about 150 people that attend, and it's 2.5 days of basically like a think tank where we have different groups of individuals that are assigned specific topics that are related to the burning questions in AMD research, and then each of the groups comes up with kind of the key points about that topic, and then we share those ideas. But this is a picture from 2019, and the group, of course, was discussing mitochondria. And you can see the delighted expression on Chris's face. She was among all of the people that love mitochondria. We refer to ourselves as mitochondriacs. So I thought this was a really appropriate place to start her tribute. So Southern California, that's the place where it all started. For me, it was back in 2009. The first Beckman Initiative for Macular Research is where I first got to know Chris. And I dug back through my emails, still left over from the University of Minnesota, and I found in 2010 an email to her talking about our collaborative project, which we did get funded through the Beckman Initiative for Macular Research. And then she and I co-produced this tutorial for the people that attend the Beckman meeting, and you can even still listen to this tutorial. It's very long, I want to warn you. It actually went into 2 parts because we had so much to say about mitochondria. And then we have a couple of really, I think, important papers together, one of them being a chapter that's part of the 3-volume set of Ryan's Retina, which if you're a retina specialist, this is going to be on your bookshelf. But we co-wrote this chapter on mitochondrial genetics of retinal disease, and it'll still remain in the coming years. And then the paper at the bottom that I'm showing you here is probably one of the most important papers that my laboratory published with her, and it really redirected our research, and that's going to be the focus of most of my talk today. I want to talk just a little bit longer about the transmitochondrial cybrid work that Chris pioneered. Barry introduced this topic, and also our moderator talked about this too. But basically, this work really revolutionized how we think about mitochondria, especially in the retina. So what Chris did was take ARPE19 cells, which is an immortalized retinal pigment epithelial cell line, and got rid of all the mitochondria, fused those cell lines with platelets from patients, and created these cybrids, which have the same nuclei but different mitochondria. And she published, probably I missed a few papers, I'm thinking. She published at least 31 papers on these cybrids using different human donors of the mitochondria, and she showed clearly that gene expression was changed, cell growth rates, stress response, response to drugs, and there's a lot of other things that were looked at. This was paradigm shifting, her ideas that emerged from this research. And if sitting on NIH grant review panels, that's one of the things that we're instructed to do. If this research is going to be paradigm shifting, that raises it to the top. And so I really think that the research that Chris published on the cybrids raises it to that level. So before all of the publications, the current paradigm was that the nucleus regulated everything and that the mitochondria was simply supplying energy. And what Chris's work in the cybrid showed was that the mitochondria and the nucleus are cross-talking. And that makes sense, right? The mitochondria is already sensing when there's more energy required or when calcium overload happens or a number of other things, and has to communicate that back to the nucleus so that gene expression can be altered to match those changing needs. And so that's what her work really solidified, is the importance of the mitochondria and how much it talks to the nucleus, and there's great implications for our work in AMD, knowing that information. So I'm eternally grateful for Chris expanding our knowledge base about what mitochondria does in the cell. And so now let me start with my research talk. So I'm going to talk about mitochondria in the context of age-related macular degeneration. This is the #1 cause of vision loss among individuals over 60 years old. And the graph that I'm showing you here is actually a compilation of research that was done in the US, Australia, and Europe. And what you can see here is from age 60 on, there's a sharp inflection in the number of the prevalence of AMD. I also want to just highlight the macula for those of you that are not in retina research. This cartoon here represents an eye. Oops. Represents an eye, and you can see this little divot here. That's the macula region, and the macula is where the light is focused as it comes through the cornea and the lens. And this image here is an en face image of what your ophthalmologist would be seeing, and you can see that the macula is temporal to the optic nerve, but in dead center of the eye. And it's amazing that this important part of the retina is only about as big around as the tip of your little finger, but it's also involved in your high acuity and color vision. And I refer to it as the high-end real estate in the retina. I think it's important also to recognize, those of us that work on human diseases, what the impact of that disease is on individuals, individual patients. And so I'm going to use this Amsler grid as a way to start that discussion. An Amsler grid is basically exactly what it looks like. Your ophthalmologist will give it to you on a piece of paper or on a piece of cardboard and say, just look at this every day, and if you see changes in the shapes of the lines, let me know. So it's a low-tech way of monitoring your vision every day. And hopefully you're seeing horizontal and vertical lines here. This is no disease. But then you start to see this distortion of horizontal and vertical lines. That's one of the first symptoms of AMD, and then eventually that becomes more prominent, and there's a scotoma or black spot in the central vision that occurs. These changes make it difficult to read. As you can see, central vision is obscured and it's blurred. It makes it very difficult to drive, and hopefully this person is not driving anymore because you can see that the main area of focus is occluded. And then finally, this is a series of pictures about how difficult it becomes to recognize faces. So as you can see, this disease has a significant negative impact on the individual's activities of daily living and quality of life. So we know a lot about what happens to the cells during the disease. We're still trying to figure out what's causing those changes. So we'll start here with a healthy retina. The outer retina is the area that's most affected by the disease. And with AMD, one of the cardinal features that occurs that your ophthalmologist will see is the formation of drusen between the RPE and Bruch's membrane. Eventually, there's photoreceptor loss and loss of the RPE. So my studies have mostly focused on the retinal pigment epithelium, but there's a lot of challenges in studying AMD disease mechanism. One of them is what I talked about earlier, the macula. It's quite unique. It's found only in primates. This is a disease of aging. It takes greater than 60 years to start to manifest till it's evident. And there's also an environmental and a genetic component to it. So these two main reasons make it very difficult to find an animal model that truly replicates all the cardinal features of AMD. Although I want to just say for those of you that use animal models, there's great utility in it in that it's meaningful in telling us about what pathways or specific proteins are important for retinal homeostasis. But what my lab has done is use human donor tissue that I've obtained from the Lions Gift of Sight in St. Paul, Minnesota. And we use the Minnesota grading system to evaluate the disease severity of the donor when they died. And I'll talk about that in the next slide. But we've taken this donor tissue and analyzed protein expression, mitochondrial DNA damage, and in cultured cells, primary cultures, as well as induced pluripotent stem cells that we have differentiated into RPE. So those are the different uses of the tissue. So I want to talk a little bit about the Minnesota grading system, because this is really one of the most important components of my research program, and that is using well-characterized donor tissue, characterized for this stage of the disease. And what I'm showing you here on the screen are high-resolution photomicrographs of the bare RPE. So the eye bank actually will get the donor eyes, dissect off the neural retina, and then take these pictures for us. And you'll notice that in each of the different slides, there is a ruby sphere placed over the optic nerve, and that's so that this bullseye can be correctly positioned and sized over top of the macula. And the Minnesota grading system was developed by my colleague Tim Olson, and he based it on the ARADS classification for patients. And we use drusen size and location and RPE pigmentary changes to evaluate the disease severity in the patient, in the donor eye, excuse me. So there's 4 stages, and the first stage is no disease. And then there's early, intermediate, and advanced disease. And advanced disease is where vision loss occurs. And you can see there's a few obvious drusen here by this intermediate stage. So I'm a biochemist by trade, and so we started off in my brand new laboratory at the University of Minnesota looking at protein expression, and the technique that I used, which was kind of the advanced technique for the day, with 2D gel electrophoresis. This is an example. These are large format gels. This is back when I used to actually put gloves on and get in the lab. You can see how large the format is. We could resolve about 400 to 500 proteins at one time, or I should say protein spots at one time. Now, the proteomic technology that I use is label-free proteomics. We can easily quantify and identify 7,000 proteins. So there's been a big advance in 20-some years since this picture was taken. But we published a number of papers on the proteomics of both the neural retina as well as the retinal pigment epithelium. And I think our most important discovery was that the mitochondria in the RPE are damaged with AMD. So this was a pretty bold hypothesis for this young assistant professor making in the field of AMD research, and especially since I did not train with really well-known people in ophthalmology. So we knew we needed more evidence for this hypothesis, and so we turned to a different technique called using long extension PCR to amplify mitochondrial DNA. For those of you that are not mitochondriacs like Chris and I were, just a little primer on mitochondrial DNA. It is found in the inner space of the mitochondria in close proximity to the electron transport chain. So it's in an area where there's a lot of reactive oxygen species produced, which can damage mitochondrial DNA. And it's also a circular genome, shown here, of about 16 kb, so it's pretty small and easy to amplify. So using this technology, we went ahead and looked at 118 different donors, and you can see at the bottom there the number for each of the stages of the disease. And just to remind you, stage 1 is control, stage 4 is where vision loss occurs. And this is actually from the macula region. We did a 5mm punch of the RPE and then analyzed mitochondrial DNA damage. And what you can see here is that there is a steady and progressive increase that correlates with disease severity. And the other important thing to mention is that mitochondrial damage is occurring before vision loss occurs, and that offers an opportunity for intervention. So this is the result that got us really excited about thinking about mitochondrial interventions and treatments. So you can't measure mitochondrial function directly in postmortem tissue because of all of the things that occur after death. But what we did instead was to culture the retinal pigment epithelium from our donor tissue. And there's a picture there on the screen for you. And then we measured mitochondrial function using a seahorse assay. And I know Chris did a lot of seahorse assays in her lab. But just to explain this to you, this is a piece of equipment that you can put a plate of cells that you've cultured into the equipment, and then it measures oxygen consumption rate of those cells, and that's a direct reflection of mitochondrial function. And then when you apply the different mitochondrial stressors that I'm showing you here, then you get this trace that looks like this, and you can calculate basal respiration, maximal respiration, and ATP produced by your cells. So it's very informative in telling you about the function of the mitochondria in your cells. And so we did this in a robust number of cells from donors without AMD and those with AMD. And just to point out, this is the trace from donors with no AMD and AMD, so I'm already telling you the answer. We saw that there was a significant decrease in both basal respiration, maximal respiration, and ATP produced by the mitochondria in cells that came from donors with AMD. So far I've given you 3 data points that support the hypothesis that mitochondrial dysfunction in the retinal pigment epithelium plays a role in AMD pathology. So one of the bits of information that came from those meetings that I've told you about, the Beckman meetings, Ryan Initiative for Macular Research, is that we talked a lot about the GWAS studies. And if you remember back in 2006, there was the publication of 3 papers simultaneously in high-impact journals that cited CFH, SNPs, as being associated with AMD, and that was the start of an explosion of genetic investigations in AMD. And one of the things we talked a lot about at the Beckman meeting was, what does all of this mean, and does it tell us anything about disease? And so this is a review paper that was written by one of the participants at the meeting, Emily Chu, and basically they identified at least 5 different pathways that seem to be correlated or associated with AMD. And what this suggested was there are multiple pathways that can lead to the same clinical phenotype, which is the diagnosis of advanced AMD. And that suggested to us that there's going to be no single therapy that really will be universally effective. I think if there's any pharmaceutical companies in the room, just cover your ears when I said that, okay? Because I know you don't want to hear it. So Chris and I talked a lot about, we're really hot on, the mitochondria. Is there a patient population that should be targeted? So we went back to the samples, these 118 samples that I've told you about, that we've published mitochondrial DNA damage, and Chris's lab did the lion's share of this work where they genotyped all of these samples for 10 common risk alleles that are found here at the bottom. These have all been associated with AMD. And I think Sherry is in the audience right now. We were talking about me sending her samples while she was pregnant and saying, please analyze these samples. So anyway, and what we found, this is just some of the data. The important finding here was that what emerged from this analysis is that the SNP in complement factor H, the Y402H SNP, actually correlated with more mitochondrial DNA damage for homozygous individuals with homozygous risk for this particular SNP. So this got us really excited, because if you consider what I'm showing you here are 19 common AMD risk variants, and they're arranged in order by according to their odds ratio, and an odds ratio just tells you if you have this particular SNP, you have an X% greater likelihood that you're going to get AMD, and you can see that complement factor H is number 2 on the list. The other important feature here is that almost 60% of people with European heritage carry this risk SNP, and also more than 50% of AMD patients also carry this risk SNP. So we thought, boy, bingo, we've got a really great population to really focus our attention on. Now let me just tell you a little bit about this risk SNP. It's due to a substitution, a single nucleotide substitution that results in a single amino acid change in the complement factor H protein. It's a tyrosine replacing histidine. And The complement system, this is part of the alternative pathway, which is involved in killing invading pathogens, maintaining tissue homeostasis. So it's very important to stay active, but when it's overactive, it can cause inflammation and cell damage and death. And CFH is the negative regulator of this pathway. In other words, it's the brake in the system. So once this CFH SNP was identified, there were several important biochemical analyses of the actual mutant protein that were produced and published, and it was shown that the Y402H protein had reduced function and could result in overactivation of that pathway, meaning there could be more inflammation. And everyone knows that the pharmaceutical companies have really targeted the complement pathway with some of the newest developments with AMD drugs. And I also want to point out that this pathway also has a number of other risk SNPs that have been identified. So now we've refined our hypothesis. Now our hypothesis for the lab is AMD genetic variants drive disease through different mechanisms. And our focus has been on the CFH high risk in RPE. And so what we found so far is, again, going back to the data that I showed you from the paper that Chris and I published, there's more mitochondrial DNA damage with AMD, but when we separated those AMD samples out into low risk versus high risk, you can see that the high risk is significantly higher or more mitochondrial DNA damage. In work that I did in, or published in IPSC RPE from low and high risk donors with and without the disease, what you can see here, at least for mitochondrial function, looking at ATP and maximal respiration, in donors with or without AMD, the high risk allele caused mitochondrial function to actually be lower for both ATP and maximal respiration. So even though we had lower mitochondrial function with AMD, if you had the high risk, you even had lowest mitochondrial function. So again, something's wrong with the mitochondria when you have this high risk allele. And then finally, this is some of our unpublished work with Jinha Du. He did a metabolomic profile, and this is a PCA analysis of that profile that he was able to obtain with our cells. And you can see that there's clear separation of the high and low risk samples, meaning that metabolism is different in those two different cells. And this is in cultured cells. It's not even straight from the individual. All the conditions are the same. So I'll kind of finish up this whole talk and just tell you a little bit about some of our newest unpublished work. And this is a proteomic analysis of samples that now we've divided up by the stage of the disease, MGS 1 through 4, and also by CFH risk. And here it is, low risk, what I call mixed risk, which is heterozygous for the high-risk gene, and then homozygous risk. And I just also want to point out that there is an allelic dose that creates a situation where you have increased risk the more alleles you have of the SNP, the mutant protein. So for individuals that are heterozygous, they have about a threefold increased risk for developing AMD, but that increases to sixfold if you have both alleles that are high risk. So there is interesting correlation there. So we identified about 7,000 proteins in each of our samples and asked two different questions. In the absence of AMD, so just comparing MGS1, in the absence of AMD, are there genotype-specific differences in the RPE proteome? And then what proteins are altered with AMD progression and is based on genotype? So those are the two questions we asked. In the interest of time, I'm not going to talk about the mixed risk. I'm going to restrain it to just the low versus high risk. So our first question, are there differences without disease? And the answer is yes. There were 162 proteins that were increased in the high-risk cells. And if you read this pathway analysis that I've put up here, this is an enrichment analysis, almost everything up here is about mitochondria. And for those of you that aren't used to looking at these pathway analysis, the bigger the bubbles, the more proteins are involved in each of those nodes that they're symbolizing here. So the biggest effect is in the TCA cycle and respiratory chain. So they're increased in our high-risk samples without disease. We also saw some changes in lipids and nucleotide metabolism in those samples. Now, what about decreased proteins? There were fewer decreased proteins, but there were 64, and the pathways that were decreased in our high-risk samples included the stress response, which I think would be a negative thing for you to have fewer stress response proteins, and cell structure. Those proteins were decreased, as well as a few of the metabolic pathways. So I'll come back and summarize all this at the end. But let's move on to the next question. So now, what proteins are altered with AMD progression? And in this analysis, I'm showing you Venn diagrams of the comparison between each of the different stages of the disease to the control sample with no AMD. So if you're not familiar with Venn diagrams, let me just walk you through this for a minute. The numbers on the outside are the numbers of proteins that were changed from the control. So for example, in stage 2, in the low risk, there were 246 proteins that were altered with that early stage of the disease. There were 165 unique proteins looking at stage 3. There was some overlap, but not very many. So this tells us that as we move from one stage of the disease to the next, there's a different set of mechanisms. There's different pathways that are altered. So that's been suggested before, but I don't think anybody's been able to really show it biochemically. And so also I want to just point out that the low-risk samples, the greatest number of proteins that were changing was in early disease, whereas with the high-risk samples, it was in late disease. And these are the numbers of total altered proteins in each of the analyses. So if we look just at mitochondrial proteins, and now this is the comparison back to how did they change through the progression of the disease compared with where they started, which is no AMD. You can see that with the low-risk samples, they elevated their mitochondrial proteins immediately. And that was just the opposite of what we saw with the high-risk samples. We saw that compared with the no AMD, they actually go down. And I'll get back to that in a minute. So another way to look at the progression of the disease and what pathways are altered is to present the data like this, which is a heat map. The darker the color on the heat map, the more the p-value, the higher the p-value. And again, let me just explain. These upper ones here are upregulated here, and then the dark green colors down here are proteins and pathways that are downregulated, and that's with early, intermediate, and late disease compared with the control. Just to point out some of the opposite effects that we observed. There's the mitochondrial proteins that are upregulated with low risk and downregulated with high risk. There's lipid pathways upregulated with low risk, downregulated with high risk. VEGF signaling, which is going to cause your angiogenesis, downregulated with low risk, upregulated, especially at late time, high risk. Response to cytokines, which is an indication of inflammation, opposite effects, downregulated and upregulated. So we get some fundamentally different pathways going in opposite directions, depending on the genotype of the individual that supplied the cells to us. Going back to the mitochondria, since that's my focus here, this is just repeating what I just showed you. For the low-risk samples, we have an increase in mitochondria throughout the disease progression and a decrease in mitochondria based on the high-risk cells. So this just tells us relative to the starting point, the comparison here is with no disease, right? But we wanted to know, what's the relative total amount of mitochondria in all of our different samples? So to get to that answer, we went back to the intensity analysis from the mass spec data, and this is the plot from that. And it agrees with everything that I've told you about as far as pathway analysis. For the high-risk donors without disease, they have a lot of mitochondrial proteins. Low risk is here without disease. And then for going back to the high risk, those mitochondrial proteins drop as disease progression goes on through 2, 3, and 4. Just the opposite happens here with the low risk. They actually increase and then drop off slightly. So let me pull this all together for you. I know it's a lot of information, but it's very exciting. So to summarize, first of all, in the absence of AMD, we saw significant differences in our donor samples that had the high risk, and that is we saw lower mitochondrial function in our IPS RPE cells, and we saw increased mitochondria based on this proteomic data that I just showed you. And this is just a repeat of the pathway analysis here. And this is just to remind you that the electron transport chain and the TCA cycle are both present in the mitochondria. So our interpretation of this elevation in mitochondria without disease is that it's some type of compensatory response. Whatever the RPE is feeling, it tries to compensate so it can stay alive. And so we think that this might be its effort to do that. But based on this data here, mitochondrial function is still lower, so it hasn't compensated enough. So with AMD, in our high-risk cells, we saw lower mitochondrial function in the IPS RPE. We saw more mitochondrial DNA damage, and that is in human donor tissue. And we saw this decrease in the mitochondrial proteins as AMD progresses. With the low-risk cells, we see a different response. We see that they go from this level of mitochondria and increase immediately and then stay almost at that level, maybe drop off a little bit. And again, is that a compensatory response to the disease in these donors with low risk? Potentially it is. But nonetheless, we're seeing significant genotype-dependent responses to AMD, and even without AMD and in the high risk, we think that it might promote rapid disease progression. Remember, I showed you also that there were changes in stress response proteins and so forth. So to summarize, I hope that I've convinced you that we should think of the mitochondria in the retinal pigment epithelium as the Achilles heel, at least when it comes to AMD. And I showed you data from proteomic analysis and then also mitochondrial DNA damage and also some mitochondrial functional analysis that all support the idea that if you have the CFH high-risk allele, there are significant changes in your RPE that are going to make it more difficult to overcome the disease. So the conclusion here, and this is something that Chris was working on, is that the mitochondria are really a good target for early intervention, and perhaps patients with the high-risk CFH allele will be the best candidates for therapy. But I want to point out that genotype alone, it does not initiate AMD. And I'll go back to some of the data that was in the paper that Chris and I published that I hadn't talked about earlier. This is where we looked at mitochondrial DNA damage in controls without disease, and then these are individuals with disease based on the low risk, heterozygous mixed risk, and homozygous high risk. And what you can see here is that even in just having the low risk, you can still get the disease, obviously. So genotype alone is not going to protect you or initiate the disease. And we even have some samples that had the high risk for CFH, and those individuals did not have disease. But I want to tell you that we struggled to get a sufficient N of individuals over 60 that were homozygous for CFH and did not have AMD. So that's really telling in itself. But we have to consider that environment has a role in this, and this comes from the multiple hit threshold hypothesis. I think, Christine, you were part of this paper that suggested this, where age-related changes plus environmental insults lowers the threshold for disease, and that the genetic defect then can help initiate the disease. So you need more than just your genes, your genetic background, but environment and age certainly play a role too. So I want to close here and thank my team of new researchers at Doheny Eye Institute. They're all new to my lab, but they're a great group. Also my collaborators on my NIH grants, my funding, and of course, I want to honor Chris and all of the contributions she and her lab have made. You can see that the trajectory of my lab's research really depended upon our relationship that started back in 2009. So thank you very much.
We open for questions first, and then we'll have a special award. Okay, I'm going to open for a few questions first, and then we'll have a special award for Dr. Farrington.
So I apologize for so much data, but you know, it's tough to cover 20-some years in 30 minutes, so. Yes.
Yeah, good talk. Yeah, a lot of information. So I have two questions. So first question is that, what do you think about the mitochondrial damage is the cause or the result? So it's because the mitochondrial damage, so you will have the AMD, or do you think once the patient have the AMD, they may have the damage? And then the second is that you talk about so many genes have the mutation, and so you think there's no one treatment for that. But what can we get from your information? Do you think there's any way? Because if you have 100 gene mutation, you cannot use the gene therapy to cure it. And then any suggestion for that?
Right, thank you. So the first question was, do I think that mitochondrial damage is the cause or the effect? That's an important question, and it's something that comes up a lot. And I think that by dissecting the pathways that are different between the two genotypes without disease and then following the progression of the disease, we'll get to a better answer for that. For right now, we're still digging through the data, and I'll have we're looking more upstream now to see what might be causing the damage. And I think if we go upstream and see what those pathways are, we'll have a better idea. So I think mitochondrial damage is a symptom of something that's happening further upstream from that. The second question, again, it was about, treatments, okay. So if you notice, there's a lot of the complement factor, or I should say the alternative pathway genes that are involved, that have been identified and associated with AMD. So that gives you one idea that cluster would be perhaps treated with these complement inhibitors or whatever. That was the idea from the pharmaceutical companies. But there were other pathways too that might present a different focus for treatment. So it won't be 100 different treatments, but it might be a cluster of, say, 5 different treatments that's either based on the genetics or based on the clinical phenotypes that's emerging. We heard wonderful talks today by people that are moving the field of imaging forward. So there may be characteristics of those in those image analysis that are going to tell us that this patient might be amenable to a specific treatment. So I think we have to combine all of the biochemical data and the imaging data and the genetics and try to figure this out. It's going to be complicated for sure.
Hi there. Beautiful talk, and thank you for the wonderful tribute to Chris Kenny as well. I was really struck by the difference you showed in the mitochondrial protein between the low and high risk alleles. So where does the wild type or the non-AMD samples fall into that initial baseline?
So the MGS1 is no AMD. So it was high for the high risk and lower for the low risk. Does that answer your question? And then it changes over the disease progression.
Hi, I have a question about just the connection between the mitochondrial dysfunction and how you get to AMD. So it's really striking to see all these genes that are involved in mitochondrial function and metabolism. How do we get from there to drusen?
That was a talk I gave yesterday.
Sorry, I missed it.
Yeah, no, it's okay. It has to do with it has to do with bioenergetics. And this is some work that Jim Hurley's lab from University of Washington published a couple of years ago. What he showed was that glucose, which is one of the energy substrates, it goes from the choroid and passes through the RPE and actually goes up into the photoreceptors. And the photoreceptors uses the glucose almost exclusively for energy production. They don't use a lot of their mitochondria. The RPE, on the other hand, use their mitochondria exclusively under healthy conditions. And so they're taking lipids from the phagocytized outer segments, they're taking ketone bodies, they're getting lactate that's secreted by the photoreceptors as a byproduct of glycolysis. So they're happy with using their mitochondria. And so when we see mitochondrial dysfunction that happens, then we know that the cell, in order to save itself, has to start using glucose for glycolysis. So with the onset of AMD, glycolysis is upregulated in RPE. That means less glucose is going up to the photoreceptors, and eventually there's a bioenergetic crisis, and the photoreceptors can lose function and perhaps die. So that's the idea, is that there's coupling between both the photoreceptors and the RPE as far as the substrates that are shared between them. And when you alter that typical or healthy condition, that's when you have this havoc start to happen and create this crisis in the retina.
Thank you.
Now I would like to invite Dr. Cooperman, Dr. Kavanaugh, Dr. Pachowski, and Dr. Nasburn to the podium.
Let's move. Should we go over to there? Are you going to bring that over? Or?
Slow these days, my dear.
Don't worry, just take your time. You gotta work. Chris has some family members here. Can we get, there's a cousin and a niece, and Jack, can you come join us too, Jack Schulman? And Stephanie, come on up as well. Let's take a larger photograph, larger group photograph. Then we'll move on with the photograph. And gather. Good enough. Come on in, everybody. Thank you. Maybe, why don't you go back more to the middle? Tony, on there. Stephanie, why don't you come over here more? There's a family over there. There we go. How are you? Come on in. Come on in. All good? We need some more on this side. Oh, we need more? Okay. A lot of them come on over. Okay. There we go. Yeah, there we go. There we go. Got the picture of Chris there too. Wait a minute, you're behind the. Okay, perfect. Does that work? Or this way?
You know, you should be shooting us from down. this out of here so I can get on with the show here.
Want us to ship it to you? I forgot to take this off.
Okay, our next speaker is Dr. Dorota Skowalski and Kravchuk. So Dorota is she is the assistant professor here at University of Irvine and in the Department of Ophthalmology, Biophysics, and Physiology. Her research interest includes the mechanism of aging, age-related macular degeneration, molecular mechanism of glaucoma, and vision science. Let's welcome Dorota.
It's a really great honor to be here, and especially in this session. And of course, I have big shoes to follow, or well, big talk to follow. But I will try to bring your attention to events that are probably happening a little bit earlier than the disease development, which is probably quite well corresponding to what we just heard. So my laboratory is interested in aging as a mechanism, as something that happens in every organ and in every cell through the life, and how these mechanisms, different mechanisms most probably, contribute to development of age-related eye diseases. So, well, maybe before I need to just say that part of the work is overlapping with my consultation interest with the company that actually licensed one of our patents. So one of the things that I would like to first lay down is how we think about aging. And we really, as researchers, trying to bring this definition of aging over and over. And one of the major things is that even if the chronological age is progressing, the tissue is still working, and it's working pretty well. So what we came up recently at the Reimer meeting that Dr. Farrington just mentioned, we recently came into the conclusion that there are many compensatory mechanisms that are actually counteracting changes that happen through life. So we can call those changes damage, but eventually it's repaired. But maybe with some expedition of the energy, those repaired status is a little bit different energetic status or different homeostasis level, but still the system is still working. So this is really important because those compensatory mechanisms are doing all work through the life. And eventually, they are counteracting all the different small stresses that happen to our eye or other tissues, which would be like a small injury, small inflammations, light exposure, and some types of damage. So those compensatory mechanisms are making everything work. So what happens when actually the compensatory mechanisms cannot do the work? And this is when the risk factors come. And this is where how we think also about how AMD develops. We have some additional risk factors, which are not minor and not just going on through life, but even if they are through life, they are particularly, straining the system, so actually there is no more chance to come back to the normal. And those for AMD, for example, is obviously genetic risk, smoking, diet, or the lifestyle. And we have disease initiation and signs of early AMD. Why I'm going to it? Because actually this helps us to divide a little bit the disease on the early stages, even before grading, as we have heard, of the already visible signs of AMD. So the idea is to really bring back to normal, and how we can do that. Our laboratory is trying to understand mechanisms of these compensatory mechanisms and how everything can be brought together to keep the keep the balance between the damage and homeostasis. And 2 points of interest in my laboratory is epigenetic changes and membrane alterations. And basically, epigenetic changes can happen, is happening all the time. It has been very well shown by many different laboratories. And our laboratory is focusing especially on, in the context of aging, we are interested in DNA methylation changes. For those that are not that familiar, DNA methylation is the small modification of the one of the nucleotides that is changing property, biophysical property of this nucleotide, and as a result, the DNA is usually less accessible or basically has changed ability to respond to signal. So it has been shown already over 10 years ago that there is some group of Cs, because those are cytosines, the nucleotide that is changed, the group of Cs in the genome that so consistently are changed in aging that we can actually have something called a clock, epigenetic clock with, for example, 353 markers proposed by Horvath in 2013. And in the same year, actually, another clock was proposed with 71 markers, which it means the 70 or 300 points in the genome. When you calculate their methylation level, you actually can estimate the age of a person that gave you the specimen. To this extent that those clocks are actually used in forensics right now and very much explored what is the minimal number of those marks that we can actually use. Well, what really came into everybody's attention, also ours, is that there is one gene called ELOVL2, we'll come more to it, that gets increasingly methylated with aging. And this interaction is the super high correlation to this extent that if we take 4 different CPGs in the regulatory region of this gene, we can actually estimate the age of the person with ±5 years. Please see what I'm saying. 1 point in the genome, 4 nucleotides, ±5 years. So this is really remarkable. But why we are attracted to this gene? Because we are. Well, this gene is actually in the very known pathway of elongation of polyunsaturated fatty acids, PUFAs. So this gene is having a special role in elongating of carbon 22 on the omega-3 and omega-6 side to carbon 24 length PUFA. Well, importantly, this 24 PUFA is a substrate to become eventually DHA, which is most common and most abundant DHA in the membranes, and also to very long-chain polyunsaturated fatty acids, which are quite specific for retina and have a very specific role. So what do we know about those factors is from the studies that was published by Center, collaborative work on the Center, is when we looked at the distribution of fatty acids, PUFAs especially in the discs in the rim versus the center of the disc, we realized that VLC PUFAs and PUFAs in general, including DHA, are extremely enriched on the level of the center of the disc, while on the edges we have more saturated and short fatty acids. So basically this brings an idea that those VLC PUFAs, since there's such a strong compartmentalization, have a really important role in vision. It goes together with the findings that the lower levels of VLC PUFAs are in aged donor eyes, and even lower levels are found in the AMD donor eyes. So we and others are very interested in understanding metabolism of lipids, which of course we also heard recently how affected it is on the proteomic level in RPE. So we have also another thing that happens in aging is actually a ratio of omega-3, omega-6 fatty acids. This is very related to the fact of different substrates in the diet. So ELOVL2, right? So we are very interested in this gene, and first thing that we looked at is DNA methylation of its regulatory region in retina in aging. So we have found, and this is ELOVL2, and those lines are depicting different CPGs, so those different Cs that can be methylated, and here you have 9 of them. And we looked one by one, and indeed the 5 CPGs, which are a little bit distal from the very close to the transcription start site, are really higher, show increased methylation in aged retinas. This is also seen as a lower expression of the gene. This is 5 months old versus 26 months old retinas, and we are showing here the RNA scope, which is showing really RNA expression. And we have a lower expression of ELOVL2, and but not other genes in the pathway, which is quite important to say. They do not seem to be regulated by aging. And what is really interesting, we hypothesized the major expression of ELOVL2 is in cones, and our single-cell RNA-seq data of the 3-month-old and 19-month-old retina have indeed shown that the majority of ELOVL2 is expressed in cones, and there is significant loss of expression of ELOVL2 in aging eye. And next, we looked at the lipid composition. That's quite a logical point. And we realized that indeed what we observe is that fatty acids, this is the total fatty acids analysis from the whole retina. So we basically look at everything from all different cell types, of course, including cones. We see the total fatty acids level change in aging, but in particular significantly for the PUFAs, even more for LC PUFAs, so long chain and very long chain polyunsaturated fatty acids. And if we take all lipidomic, so this is now we take lipids, not only fatty acids, but all lipidomic data from the retina, we see that there is a deregulation of plasma membrane, well, actually membranes properties, because of course there is a lot of membranes which are not plasma membrane in the retina, as you know. And a lot of changes in the biophysical properties, which tells us, yes, there is a definitely problem on the level of composition of the lipids that can contribute to the problems in vision. Well, having said that, obviously we have changes in the aged retina when the ELOVL2 levels are lower. We have changed on the level of vision. This is the OMR in the dark conditions. This is a scotopic ERG, A wave, but we of course have many more data. But one of the important things was to ask whether the visual cycle is affected. And we did one of the assays that we did is a retinoid analysis, and we realized that actually the visual cycle, at least on retinoids level and also on protein level content, is not really changed. So the changes elsewhere, it's not really on the level of most probably rhodopsin. Well, another things that we observed, which are adding to the phenotype, is that RPE of the aged animals, which is not only us, it's already observed by others, is also showing a lower level of phagocytosis, which, as we know, is very important to removal of debris every day that are produced through the vision function. So, okay, but is it a ELOVL2 role? Is it really dependent on ELOVL2 gene? So for that, we actually generated an animal that that ELOVL2 enzyme cannot make this important reaction. And we already published part of this story before, showing how there is some accelerated vision problems. But here we have one of the examples of a very detailed analysis when we looked at 12-month-old wild-type and mutant animal and 18-month-old wild-type and mutant animals. And as you can see, there is a huge difference. But what caught our eyes is that actually ELOVL2 animals' levels of the response are very close, of mutant level response, are very close to the wild-type 18-month-old response, which is depicted here, which tells us there is a problem with, there is accelerated vision loss when it comes to the vision function. And we also observed important changes and significant changes in the fatty acid composition that was very similar to aging retina. We also observed a change in the lipid species. And finally, we again didn't see any change in the retinoids level, suggesting that this is not on the level of the visual cycle. And last but not least, we have found that the phagocytosis is also also lowered, efficiency of phagocytosis is lowered in the mutant animals. So the question here is, can we use this knowledge to design therapy, to think about potential therapy that will actually address the problem with the lack of lipids, lack of polyunsaturated fatty acids? So we decided to go for the proof of concept approach with intravitreal lipid supplementation. What we decided to do actually is to bypass the problem of lack of E-level-2 in aged animals and supplement them with intravitreal injection of its product, so E-level-2 omega-3 product. Then, as a controls, we of course wanted to look whether this is specific, and we looked at the upstream. This is actually EPA. We of course looked at the DHA supplementation, and then very long-chain polyunsaturated fatty acids, which also has been suggested to have some role in improvement of vision. So this is a very busy slide, but I don't ask you to read every little chart, but I want to bring your attention that only 24/5 supplementation gave significant improvement in vision on every level, scotopic and photopic. And there is a little bit of the improvement in the 30-6, which we are not surprised and actually pretty glad, knowing that it has been shown previously in some oral supplementation studies. And then, but neither DHA nor EPA are able to do that. So it just tells you that this is the two fatty acids that are most famous for being in supplements are actually not doing the role or doing the things that 24/5 fatty acid can do in the retina. We, of course, made many more assays, and here is in collaboration with Kefalov Lab. We have done a dark adaptation recovery study, and we observed that this is basically one eye versus another eye. So it's the same animal, one eye is injected with vehicle. So the eye that is injected with the fatty acid is actually recovering much better and faster. Then we also looked at the visual evoked potential, so the information is also brought further to through the pathway, and we see here also increase in the response of neurons. So last but not least, we look, okay, so what's the mechanism? So first of all, we looked whether we can observe any enrichment of the products of E-level-2, which is VLC-PUFAS or LC-PUFAS, in different lipid groups that could accommodate VLC-PUFAS in them. This will be PE, LPC, this is plasmalogens, PS, and PC. Those are different phospholipids that can accommodate those long fatty acids. And as you can see, all of them got some portion of the increase of the VLC PUFAS in there. We also looked at the transcriptional response, of course, and we took the retina from the one eye versus another. Again, the same animals, and this is a triplicate study. And we have observed that there is most of the genes, which is around 250, are downregulated with supplementation of this fatty acid. And if we look for the pathways that are kind of a clusters of pathways that we can detect, there is one which is really coming very much, is the immune response. There is a bit about phagocytosis, and from the further studies, we know actually this phagocytosis is related to microglia, and then cell migration, which we think it may be also microglia component. So last but not least, we looked at some genes more precisely, and here, for example, we have the aging brain pathway that has been found, that is found in GSEA, the gene set enrichment pathway analysis. And for example, we have seen that there is a really beautiful downregulation of those genes, so now we are not inflammation specific. And one of the factors, for example, is here, ApoE. So to corroborate the studies with the transcriptional and all other studies with immunostaining or protein detection, we looked at the several of factors, including C3, so complement part, and C5b9, which is the MAC component. We looked at their levels. This is in the aged vehicle-injected retina, and this is in the supplemented retina. I hope you see, as we see, that there is a really change in the composition. And one of the really also quite admirable changes also in the how the structure is preserved. So, well, I wanted just to tell you that when studying aging, we can actually learn quite a lot, and maybe we are able to find some new therapy ideas. So we know that we see these age-related changes in lipid composition. Now we think is before even the AMD phenotypes happen, which gives us, again, the window of opportunity for potential treatment and maybe slowing down some progression of some phenotypes. Well, we can also have, using approach as we just did here, using a DNA methylation, so epigenetic changes, we can also find new therapies. And last but not least, of course, this supplementation showed really beautiful results. And one of the things which is very new, and I will not tell you all the details, even if you ask me, we also found the variants in E-level2 that are correlating with AMD progression. And through that, I would like to thank all the collaborators and people that worked on this study. I mentioned Vladimir Kefalov. I need to also talk about our Polish collaborators, Andrzej Fojk, and sources of funding. And I just want to say a few words about women and vision research. Well, when I came here, I put together a committee for vision research in Center for Translational Vision. And guess who was the first person that joined the committee? That was Dr. Kenny. Thank you. I'm ready for questions.
Thank you, Dorota. I have a question for you. So when you do the experiment, do you see a lot of inflammation in the vitreous when you inject the medication? And how long does the lipid supplement last in the vitreous cavity?
Oh, that's a really good question. The thing is that our studies, we have everything is within 5 days. So it's a proof of principle study. We actually used a lot of animals, so next studies will have to go longer. Also, next studies will have to do with, we want to work with labeled lipids, so we actually can really see where the lipid is directed.
Okay.
Thank you very much.
Thank you, everybody. This concludes our morning session of scientific session.
Thank you. Thank you. Well, I think this was just a wonderful tribute to Chris Kenny's life and career. So thanks to all the speakers for their wonderful presentations. And it's time for lunch. And please don't forget to check out the posters. And we will reconvene here for the first afternoon session at 1:25. Thank you.
Good afternoon, everyone. We'll go ahead and get started. Hopefully you enjoyed that delicious lunch, and we'll go ahead and start our afternoon sessions. This is the most exciting session of all, the retinal vasculature in disease, and we're starting with our first speaker, Dr. Kalpana Gupta. She's a professor of medicine here at UC Irvine, specifically in the Division of Hematology and Oncology. She got her PhD from the prestigious All India Institute of Medical Sciences, and she's really led pioneering work in the foundation of understanding pain in sickle cell disease, and it's a topic I'm particularly interested in. So I'm excited to introduce her talks entitled Opioid Signaling in Retinal Vasculature Leading to the Promotion of Retinopathy. Please join me in welcoming Dr. Kalpana Gupta.
I'm really honored to be here and have to fit big shoes from the preceding talks. And I thank Dr. Mishra for this very kind and very warm introduction. And as he said, the focus of our lab is in one direction on sickle cell pain, but overall on pain. And it started with understanding how opioids signal in non-central nervous system tissues. And that led to this work that I'm presenting today on opioid signaling in retinal vasculature, leading to promotion of retinopathy. Okay. These are my disclosures. The work that I'm presenting today has nothing to do with all my other activities that you just saw. So why did I think in the first place of opioids and angiogenesis? And the reason is the parallel histories. How do I use the pointer on this? It's okay. So if you look in the pink and the yellow boxes, opioids opium was refined around the same time in late 1600s when angiogenesis was discovered by the physicians, and they said that the heart pumps blood. And it was after Leonardo da Vinci had depicted that it looks like the branches of a plant. And then Linnaeus classified the opium poppy in 1757, and a few years after that, John Hunter coined the term angiogenesis. So they were running pretty parallel. And then in research on pain and nervous system was the area for opioids, and for angiogenesis, research led to the understanding of endothelial cell activation, inflammation, and others. And it was not until 1971 when Judah Folkman felt that by cutting the blood supply to the tumors, we can really regress the tumor growth.
It's very dim. That's the
Oh,
issue.
I see. Okay, that's all right. Thank you. And in 1989, VEGF was discovered by Napoleon Ferrara, and it was also called the vascular permeability factor. So Judah Folkman always insisted it should be VPF/VEGF. And the delta opioid receptor was discovered in epididymis, not in the central nervous system, rather in the reproductive organs in 1992. So they were at very parallel histories, although there was some diversification that opioids act via the G7 transmembrane domain G protein coupled receptors by inhibiting cyclic AMP traditionally. And there are 4 different opioid receptors which have been characterized by now. The mu opioid receptor is the one which mediates analgesia and is very central to the central nervous system. Whereas delta, kappa, and nociceptin opioid receptors have other functions, and they're still being discovered and what is their role in pain. Whereas VEGF acts via the receptor tyrosine kinases, who have a mitogenic function. And as they say, mitogenic means that they would induce cell proliferation and promote cell survival. Whereas the inhibitory role of the opioid receptors in reducing cyclic AMP traditionally would mean that they would inhibit the cell survival and proliferation. However, upon continuous activation of these GPCRs by mu, it can also lead to superactivation of cyclic AMP, thereby increasing cyclic AMP and reversing its functionality from an inhibitory role to a stimulatory role. And we took advantage of this fundamental understanding of GPCR superactivation, and the reason was that in chronic pain conditions, opioids are used for very long periods of time, such as in cancer pain or in sickle cell disease pain. In sickle cell disease pain, they can be used all their life because pain can start in infancy. So we wanted to understand what is the role of opioids in angiogenesis. And the reason was that when any molecule enters the systemic or enters the body, how is it transported? It is transported through the blood vessels. They are our transport system. And this transport system is manned by the endothelial lining. So the endothelial cells come in contact with anything entering. And the other reason was that the mu-opioid receptor was just cloned on the endothelial cells as I was entering my postdoctoral research at the University of Minnesota. And so what I did was I wanted to understand how these opioids, particularly morphine, which is a good representative of mu opioid receptor and agonists or for painkillers, I started examining the role of morphine in the endothelium. And what I found was, since the receptor had just been cloned in 1990s or characterized, not much was known about it except that it binds to inhibitory GPCRs. So when I looked at it, I found that it activated the PI3 kinase, and via the PI3 kinase, it activated AKT signaling, PKB-AKT, which is a survival-promoting signaling molecule, where it plays a very central role, and it inhibits apoptosis. And this pathway could straight away lead to MAPK-ERK phosphorylation. But in case of opioid receptor signaling, what I found was that it acted via nitric oxide. And if we inhibited nitric oxide, morphine would not activate MAPK-ERK. Now, this was a very characteristic specific feature of VEGF receptor signaling, but not of FGF, not of PDGFR-beta, or any other mitogenic growth factor. So this made us wonder if opioids or GPCRs co-activate VEGF receptor, or how do they work in concert. But anyways, we first wanted to look at how opioids really acted all the way downstream into a functional output of cell proliferation and angiogenesis. And we found in tumor models, in other ex vivo models, multiple models, that opioids promoted angiogenesis. They did so by promoting a dual system, that is the mitogenic pathway as well as the survival promoting pathway, and by increasing cell cycle progression in the G1S phase. So this led us to think and confirm in many different ways that opioids have the potential to promote angiogenesis. And in the last about 20 years, many other investigators and my lab have contributed majority of this work, and we have found that it promotes cancer and endothelial cell proliferation, migration, invasion, epithelial to mesenchymal transformation, angiogenesis, vascular permeability, immunosuppression, and also inflammation, depending upon the situation, cancer progression, and metastasis. And these are supported by several retrospective studies in different cancers. There was a small body of literature where they used very high dose opioids, like 70 milligram per gram of mouse, and they found that there was an inhibitory effect on tumor growth, etc., opposite of what has been seen by majority of the investigators. All this is reviewed in pain by us and others. So then we wanted to see, we had been working more on the cancer and angiogenesis and taking it all the way to clinical translation. And we have just completed a national trial on these findings to see if it has an impact on lung cancer progression and if it could be inhibited by a peripherally only acting mu-opioid receptor antagonist, naloxegol. So the results are still being analyzed. But at the same time, we were interested in seeing how the retinal vasculature responds to it. And the reason was, since I was working on sickle cell disease also, that they get retinopathy. And the retina is a very sort of a puristic system which has a high amount of vasculature and therefore a lot of endothelium within the retina. And retinopathy is a cause which occurs in many other conditions other than sickle cell disease. So what we found was similar to our findings in the human dermal microvascular endothelial cells and other endothelial cells. We looked at the retinal endothelial cells that we isolated from a wild type C57BL6 mouse. And what we found, we did these studies in parallel with VEGF because we were very curious to know how these two interact, or are there separate pathways and how are they acting in the same manner. So we found that morphine and VEGF both stimulated phospho-MAPK-ERK phosphorylation which is the mitogenic signaling pathway, in the retinal endothelial cells of mice. And also, they promoted the phospho-PKB ACT pathway, which means they also promoted the survival of the retinal endothelial cells. And functionally, we saw that both morphine and VEGF at a dose response, as you can see, with increasing dose, they increased cell proliferation. And what we also looked at, whether there was synergism between the two or there was an additive effect. And when we incubated these retinal endothelial cells with morphine plus VEGF at a lower concentration and a higher concentration, we see that they have an additive effect as compared to one or the other alone, which means that there are multiple other pathways by which each of them might be acting other than the nitric oxide MAPK pathway. And both of them also inhibited apoptosis. This is an assay of apoptosis, where we see that morphine and VEGF inhibited almost 90% apoptosis induced by serum starvation in these retinal endothelial cells. At the same time, we also found that morphine as well as VEGF, they stimulated a pro-inflammatory signaling, which is the STAT3 pathway. And this was also time-dependent similarly in morphine and VEGF. And there was very nice nuclear translocation because this is required for the transcriptional regulation of cytokines by JAK-STAT3 signaling. So this led us to think that morphine and VEGF might be acting in a similar manner, and maybe morphine is co-activating the FLK1 receptor, and that is why they act in a similar way, and vice versa. So here I show you that we did the IP with mu-opioid receptor by immunoprecipitating more using a specific antibody, and we could pull down the phosphostat-3, which means that it was going via the mu-opioid receptor. And then we did the same thing by trying to pull down the FLK1 KDR, and we see that FLK1 was also associated with mu-opioid receptor because IP more pulled down phospho-FLK1 KDR. So this confirmed that there is a co-activation and the two receptors may act together. However, if you look at this, we were able to publish these findings after about the largest number of rejections I've ever had on my paper. So I thought I must be crazy. This was my first R01, and I thought I'm done. And I think I'm thinking in the wrong direction. So we published it in a journal of -17 at that time. But we published it in 2006, and our paper had hardly come out that I saw this on the cover of Blood, which now has an impact factor of about 25. And so this showed in zebrafish, Vladimir, it should make you happy, that the gamma-2 subunit of the GPCR was required for the activity of KDR-FLK1 to stimulate PKB ACT, or the survival promoting signaling. So, and also some other pathways. So this made us very happy at that time because we thought that we are not crazy and we have a life. But otherwise, I would have thought I'm done. But now, in the present situation, when this happens, we say we are scooped. So all of you are here. I'm sure you have gone through these pathways, like mine. So the question then was, everybody said, so if anybody takes morphine, they will get cancer. So we were wondering, why is it that we see this upregulation of mu-opioid receptor in all these, in cancer cells? So what we did was we tried to understand, because it was also known that FLK1 KDR was not expressed on the quiescent endothelium, and many investigators in the beginning who were working on it had difficulty looking at it until it was knocked down by Napoleon Ferrara and it was lethal. So what we did, and then it became known that the fertile environment, which is replete with cytokines and growth factors, et cetera, leads to upregulation of FLK1 KDR. So we tried to mimic those conditions using a complete medium and a serum-free and growth factor-free medium. And what we found was that the mu-opioid and delta opioid receptor expressions were upregulated. And again, then after that, we then divided this and we used separate growth factors and VEGF stimulation. And we found with the growth factor cocktail, as well as VEGF alone, we could induce the expression of mu-opioid receptor seen by RT-PCR as well as Western blotting. So this confirmed that it is likely that opioids being used under normal conditions may not do anything. But when there is a fertile environment, like in cancer and other conditions, it may induce growth. So to further confirm and take it a step ahead towards translation, we actually took several prostate cancer cell lines. These were non-metastatic, and the DU and LAP are highly metastatic and very aggressive prostate cancer, human prostate cancer cell lines. And BPH1 is the benign prostate hyperplasia cells. So we incubated these cells and then collected the medium and then incubated our endothelial cells, which were human dermal microvascular endothelial cells, isolated by our lab from neonatal human foreskins. And we incubated these cells with all these conditioned medium. And what we can see here in these, that the BPH1 did not stimulate the expression of mu-opioid receptor or FLK1 significantly, but the two highly metastatic cancer cell lines, DU and LAP, really upregulated more expression very, very significantly, as well as that of FLK1. But the other, which were not highly metastatic, those prostate cancer cell lines also stimulated expression of mu-opioid receptor. So this confirmed that mu-opioid receptor is important, and silencing the mu-opioid receptor actually inhibited the phospho-MAPK phosphorylation. Vladimir is showing me the watch, so we'll go to retinopathy. And let's see what happens in retinopathy. So in retinopathy of sickle cell disease, and why we studied it is, I think many of you may know what is sickle cell disease. And in sickle cell disease, actually the hemoglobin molecule becomes sickle shape under low oxygen because of a point mutation in the beta globin chain of hemoglobin. So retinopathy is a disabling manifestation of sickle cell disease, and promotion of angiogenesis by opioids suggested that opioids may promote retinopathy in sickle cell disease. And an analysis of 1,000 patients in England showed that at least 92 of them had retinopathy in adulthood. So this led us to use a sickle mouse, which is called New York mouse, which expresses 25% of human sickle hemoglobin along with mouse globins. And when we treated these mice for 10 months and 15 months, we see that there is an increase in neovascularization. And in different parameters of neovascularization, we can see here that they increased the ends, which means the number of vessels, the vessel length, and the nodes which give rise to vessels. And these were actually inhibited by naloxone. Naloxone inhibited almost all three of these parameters of angiogenesis in these. And and so I would now like to end this by showing you that. And there was a decreased number of mu-opioid receptors. And so in brief, we can sum it up that there's pain in sickle cell disease. Morphine is used for pain, which would increase pro-inflammatory cytokines and growth factors and beta endorphin, which could lead to increased opioid receptor expression. And all of this can lead to retinal neovascularization, and this pain will be continued, and the cycle thus continues. And this is my team, and I'm most indebted to Jerry Luddy from Johns Hopkins, who taught us how to do retinal neovascularization. And thank you very much. And my my entire research was summed up by my son then in high school in this poem, who wanted to be a poet, but instead he had a transformative effect because of my work partially, professional hazard, became a neurosurgeon.
Thank you, Dr. Gupta. We have time for just one question.
Yeah.
We just have to keep things moving. So if, and if Dr. Gupta will, of course, be here. We have
This talk, I was kind of wondering about the physiology of the receptor on the endothelium. I was wondering about the physiology of these receptors. So would these be playing a role in neovascularization? In other words, would it be helpful to antagonize them with naltrexone or something? Is that something that you've thought about looking at?
Yeah, so there was a picture and the graph where we use naloxone. The only problem is if you use any of these antagonists, since morphine is used for pain, then they would also.
About in the physiologic situation where
We did. Yeah, we did all those, and they do inhibit. And we use different specific inhibitors. And as I said, right now, there are at least 3 mu-opioid receptor antagonists which act only peripherally. So they are being used in patients for opioid-induced constipation. So they do not reach the brain and do not cross the central nervous blood-brain barrier. So we have also used that in the clinical trial. Thank you.
Thank you. Thank you so much, Dr. Gupta. So we'll go on to our next talk. I'm excited to introduce Dr. Gostab Ghosh. He is an associate professor of ophthalmology at the UCLA Doheny Eye Institute. He received a PhD from Stony Brook University in biomedical engineering, as well as a postdoc in vascular biology at Boston Children's. He's done some amazing work, and it's getting a lot of press with multiple publications recently in the high-impact journal Diabetes, and I'm very excited to introduce this talk. It's entitled Stiffening of Retinal Vessels Threatens Vision in Diabetes. Join me in introducing or welcoming Dr. Ghosh. Thank you.
All right, good afternoon, everyone. I would like to begin my talk by first thanking Dr. Kefalov for kindly inviting me to come here today and share some of the work we have been doing over the past few years that is beginning to implicate retinal vascular stiffening in diabetic retinopathy, which, as you may know, is the major retinal microvascular complication of diabetes. Okay. Let's see. All right, so in the advanced stages, diabetic retinopathy is characterized by vessel multiplication and leakiness, as you can see in this fundus photograph, that eventually leads to a retinal fibrosis and tractional detachment, leading to this loss of vision. It affects almost about 40% of all individuals with diabetes, is the leading cause of vision loss in the working age population. Among the current pharmacological treatments, anti-VEGF is the most popular, and yet it is not very effective in a large population of patients. So that has led to a growing recognition that perhaps this condition can be better addressed by tackling it in the early stages that is marked by significant degeneration of the retinal vessels. As you can see here in this photomicrograph, these arrowheads indicate acellular vessels, meaning these vessels are devoid of endothelial cells. They're all dead. And as a consequence, they collapse over time, leading to retinal hypoxia, which leads to a compensatory increase in VEGF, and that's responsible for the multiplication and leakiness that eventually leads to vision loss. Work in our lab has been focused on understanding exactly how diabetes causes this loss of retinal vessels. And the idea is that if we can understand that, probably we can block the progression of diabetic retinopathy at the early stage. Now, seminal work from Tim Kern's lab and others have implicated vascular inflammation as a major contributing factor in the degeneration of retinal vessels in early diabetes. And this vascular inflammation is characterized by the adhesion of circulating leukocytes to these activated retinal endothelial cells. And by activation, I mean that they undergo this NF-kappa B dependent ICAM1 upregulation. Once these leukocytes adhere, they become activated, produce a lot of cytotoxic and pro-inflammatory factors that eventually kill the endothelial cells, leading to the degeneration of vessels. Now, numerous studies have now shown that endothelial activation is a rate-limiting step, because if you simply block ICAM1, you can block the adhesion of leukocytes and the corresponding death of the endothelial cells. And so with this sort of background, and since I'm a vascular biologist by training, when I entered this field of diabetic retinopathy, the question that I was interested in asking is, how do retinal endothelial cells become activated in the first place? Now, there are a lot of very important studies that have been performed that identify a very important role of soluble cues in the form of hyperglycemia, oxidative stress, advanced glycation end products, and a lot of pro-inflammatory factors secreted by the neuroretina. Again, seminal work coming from Tim Kern's lab. But in our lab, we are looking at the same problem, or we are asking the same question of how endothelial cells become activated from a rather engineering or physical context. And by that, I mean we are interested in understanding the role of mechanical cues, specifically the role of vascular stiffening. By vascular stiffening, I mean that retinal vessels that normally feel like a plastic straw become significantly stiffer in diabetes and start feeling like a plumbing pipe. Now, why do we care about vascular stiffening? That's because a lot of past studies have already shown that changes in vascular stiffness contribute to many vascular complications that range from the development of abnormal tumor vasculature, as shown here in these images. This is my past work from a postdoc. And others have shown that changes in vascular stiffness also contribute to increased leakiness in larger vessels, such as aorta. That is one of the major initiating events in the formation of atherosclerotic plaques, and also edema in the lung vessels in response to sepsis. Changes in vascular stiffness can do all of this because it has the ability to alter endothelial mechanotransduction. And this endothelial mechanotransduction then alters endothelial function, leading to all these pathological changes that we just discussed. So what is endothelial mechanotransduction? Well, the term mechanotransduction classically means the conversion of mechanical forces that can manifest in different forms into intracellular signaling pathways that eventually alter cell biology. And now a lot of seminal studies have already identified that such mechanotransduction pathways control a whole range of cell behaviors, ranging from changes in enzyme kinetics, controlling cell cycle progression, regulating gene transcription in the nucleus, and remodeling the cell-secreted extracellular matrix. Now, these concepts are not just specific to endothelial cells. Just because this talk is about vascular biology doesn't mean that this is restricted to endothelial cells. You find a very similar concept across the entire human body. And this sort of schematic or this illustration gives a very good idea. We just looked at the risk of vascular stiffness in the development of atherosclerosis in the vasculature. We also know that tumors are significantly stiffer than the normal stroma, and that's how they're often diagnosed, through palpation. Astronauts who spend an awful amount of time in space lose bone mass because of the loss of gravitational forces. And finally, the very simple connection of touch is a form of mechanical force. And it was the discovery of touch-sensitive, mechanosensitive ion channel piezo by Arden Patapushan at Scripps that was awarded the 2021 Nobel Prize in Physiology and Medicine. So with this background, the basic question that we came down to was, does diabetes increase the stiffness of retinal blood vessels? And if so, whether that would increase vascular inflammation and associated death of the retinal endothelial cells. The work I'm going to present today is the work of my postdoc Satish and my graduate student Irene Santiago. So the first thing that we did was we isolated the retinal vessels from mice, and we subject them to a technique called atomic force microscopy. This is a very common technique used in physics and materials science. And what it does is basically brings down a cantilever that literally pokes on the vessels. And depending on the degree to which it bends, you can tell how stiff or soft your substrate is, in this case, the retinal vessels. So based on these atomic force microscopy measurements, we showed that retinal vessels in diabetic mice are 2 to 3-fold stiffer. And this correlates with significant increase in the endothelial activation in these vessels, as judged by ICAM1 mRNA upregulation. And as we know that ICAM1 is the endothelial receptor that leukocytes use to bind and transmigrate, we sort of expectedly found that there's also an increase in leukocyte infiltration in the retinas of these diabetic mice. Now, when you look at a vasculature, you see an endothelial cell that is lining the luminal surface that is lined outside, right outside by basement membrane. It's a very thin, specialized extracellular matrix. So once we found that the entire retinal vessel becomes stiffer, we asked whether this is a combined effect of both cell and the subendothelial matrix stiffness. It's very hard to do these studies in vivo or in intact retinal vessels, so we developed a simpler retinal endothelial culture model. And when we grow these cells under high glucose conditions for 10 days, then decellularize, leaving behind only the matrix, we can again bring down our atomic force microscope cantilever and measure the stiffness. And as you can see here in this bar graph, the high glucose treated cultures produce a significantly stiffer matrix that corresponds with a significant increase in the cell stiffness as well. Now, I want to point out that cell stiffness and matrix stiffness are not uncoupled. They're not two separate things. They're actually connected because cells form physical adhesions with their underlying matrix. And this sort of mechanical interaction is defined by this principle of mechanical reciprocity that says that changes in matrix stiffness produces a corresponding change in the stiffness of the overlying endothelial cell. We next asked, well, how exactly is the retinal vessel becoming stiffer, or in other words, the matrix becoming stiffer? We zeroed in on this enzyme called lysyl oxidase because it's a copper-dependent amine oxidase that promotes collagen cross-linking and matrix stiffening, exactly as you see in this schematic right here. And it was previously shown in some seminal studies that it contributes to vascular inflammation associated with atherosclerosis and sepsis. So it turned out to be a very good and a first pass target for us. Not surprisingly, we found that levels of this matrix cross-linking enzyme lysyl oxidase is significantly upregulated in the retinas of diabetic mice. These are whole retinal measurements. We looked at the retinal cross-sections and we found that indeed, even in the vessels, which are labeled here by collagen type 4, you indeed see a significant increase in collagen expression at the protein level. We found a similar trend in human retinal endothelial cultures that we subject to high glucose treatment for 10 days. Importantly, when we give these diabetic mice an oral inhibitor for lysyl oxidase, this diabetes-induced increase in stiffness no longer happens. And importantly, by preventing this stiffening, you prevent endothelial activation, leukocyte adhesion, and the capillary degeneration associated with diabetes. Interestingly, when we did a functional study, we found that this diabetes-induced loss of contrast sensitivity, that is one of the hallmarks of early diabetes, can be significantly rescued by treating these diabetic mice with a LOX inhibitor. Now, at this stage, we don't have any evidence to indicate whether this is a direct effect of lysyl oxidase inhibitor on the neuroglial cells, or is it an indirect effect through the vasculature by regulating inflammation. And those are some studies that we are yet to perform. So the next question that we asked is, well, how exactly is LOX upregulated in diabetes? I'm not going to take you through the data in the interest of time. This is already published, but this is sort of the net sum. And we've identified age-rate signaling as a major determinant of LOX upregulation in the retinal vasculature, where this binding of RAGE by age leads to NF-kappa B activation that then activates or upregulates RAGE, ICAM1, and lysyl oxidase simultaneously. RAGE sort of feeds back to amplify this signaling pathway. ICAM1 goes to the cell surface, binds leukocytes. LOX goes to the matrix, cross-links and stiffens that matrix. And through this stiffening, it feeds back to activate the mechanosensitive NF-kappa B. Right, so once this NF-kappa B is activated, you can imagine that it's, this LOX-dependent matrix stiffening has kickstarted this feed-forward mechanism. So you have two feed-forward mechanisms, one at the matrix stiffening level and one at the soluble level. And this, these leukocytes that are adhering eventually then contribute to retinal vascular degeneration, as we already discussed before. Now, preliminary studies from our lab are showing that similar to age-RAGE, I should mention that by blocking this matrix, LOX-dependent matrix cross-linking, we are able to block NF-kappa B activation and simultaneously RAGE activation. So, and this is an important point because this inhibitory findings show that LOX can be treated as an alternative downstream target to block age-RAGE activation in diabetes, which is otherwise very difficult to target directly. Now, preliminary studies from our lab are also showing that similar to age-RAGE, TNF and VEGF also upregulate LOX, where the LOX upregulation response to these factors cause matrix stiffening, and the stiffer matrix then feeds back to upregulate their corresponding receptors, thereby setting in motion another feed-forward loop. Now, if you remember, in an earlier slide, I basically presented this concept where we said that endothelial activation, in other words, ICAM1 upregulation, is a rate-limiting step. So in our lab, we are very interested in understanding exactly how does vascular stiffening increase ICAM1 expression. One of the early targets that we started looking at is eNOS, or endothelial nitric oxide synthase, because it produces quite constitutive baseline level of nitric oxide that exerts anti-inflammatory or anti-ICAM1 effects. Not surprisingly, their levels were significantly inhibited in diabetes. And then we looked at the mechanism of how this could be happening and how this could be linked to increased stiffness of the retinal vessels. This eNOS can be activated in a calcium-dependent or calcium-independent manner. In a calcium-dependent manner, it basically binds to the influx of calcium binds to calmodulin, and this calcium-calmodulin complex can then activate eNOS, producing more nitric oxide. Now, this calcium influx is in turn sort of regulated by, among many channels, TRPV4. It's a mechanosensitive calcium-permeable channel that is very sensitive to cyclic stretch and shear stress. And so we just connected the dots and we asked, well, if it's already sensitive to mechanical forces in the form of stretch and shear stress, could it perhaps also be sensitive to changes in stiffness? And so that led us to this hypothesis that we show here. When we looked at the TRPV4 mRNA levels in the retinal vessels, we found that diabetes significantly reduces it at the mRNA level and the protein level. And in human retinal endothelial cell cultures, we see a significant drop in the calcium influx, which is a major sort of assay that is used to determine TRPV4 activation. Importantly, when we block the LOX and as a consequence prevent the retinal vascular stiffening, this downregulation of TRPV4 is significantly rescued, both in vivo and in vitro. And the most important slide, so at least based on our current data, shows that by directly activating TRPV4, by administering this TRPV4 agonist in diabetic mice, we can rescue this drop in vascular eNOS, and with it, we can prevent this diabetes-induced increase in ICAM1. And as you remember, this ICAM1 expression is a rate-limiting step in diabetic retinopathy. And so with this, I would just like to conclude by saying that our studies so far show that diabetes or high glucose activate a lot of different pro-inflammatory factors in the retina that increase retinal lysyl oxidase, that contribute to the stiffening of the retinal vessels, both at the endothelial and the subendothelial matrix level, that together downregulate TRPV4, that in turn downregulates eNOS, leading to ICAM1 upregulation, leukocyte adhesion, and subsequent degeneration of the retinal vessels. Now, this is a very technical summary, of course, but there's also a conceptual message I would like you to consider when you summarize all of our work so far. And that is that if you take endothelial cells in particular, or any cells in general, that all have the same genes, exposed to the same soluble risk factor, in this case, let's consider high glucose, but subjected to differences in cell or subendothelial matrix stiffness, you can produce a completely different response. In other words, what I'm trying to say is simply changing the cell matrix stiffness alters the endothelial cell's sensitivity to the same soluble factor in much the same way that changing the tension of the guitar strings using tuning knobs produces a very different tune in response to the same finger strokes. And so just how a guitarist spends a substantial amount of time trying to fine-tune the stiffness of the guitar strings just so they can produce the most optimal biological tune, our findings are beginning to identify the need to optimize the stiffness of the retinal blood vessels so that these blood vessels can now produce the optimal biological tune. With that, I would like to end. This is obviously the most important slide of all. I have many people to thank, but most importantly, Satish, my postdoc, and Irene Santiago, who contributed extensively to this project, a lot of funding sources. And last but not the least, Tim Kern, one of you, who's been an amazing collaborator, a friend, and more importantly, a mentor as has transitioned from a non-diabetic retinopathy field into diabetic retinopathy. So thank you, everyone, for your attention. I'm happy to take any questions.
So I have a question regarding the protein composition of the extracellular matrix changes. So do you observe any in your exactly decellularized, for example, system upon glucose treatment? Do you observe special glycosylation, for example? And then whether the LOX, your enzyme of interest, would be less effective on healthy matrix if there are changes?
Oh, okay. Great question. So we have not looked at glycosylation levels, but we do see a ton of LOX immunolabeling in matrix secreted by HRX grown under high glucose conditions. We also know that under the high glucose culture conditions that we use, there's a lot of advanced glycation end products forming. And so if I just connect the dots, I would say that LOX is able to cross-link the matrix in the presence of age modifications. Beyond that, we don't have any evidence as of now.
Thank you.
Hi. So that was a terrific talk, Eric Borman. Yeah. I had a question about you talked about the piezo mechanoreceptors. Did you do? I was waiting to see if you were going to show something from there. Yeah. So have you done anything with those?
No, but that's the direction, exact direction we are heading because, see, I was not trained in the eye, so I'm still taking baby steps. And I'm very careful before I head out into a new direction, and I run every idea by Tim. And so far, we have an agreement that this is sort of a good direction to start with. But once you hit TRPV4, which is mechanosensitive ion channel, piezo is a very close cousin.
Right.
So it just basically gets wrapped up in the same story. But yeah, we are heading in that direction.
Great. Look forward to hearing about it.
Thanks.
For the sake of time, maybe your question is the last one, and then
thank you.
And then we'll reserve them for after. Yeah.
Okay. Thank you. Very interesting talk. I just wanted to ask you: have you looked into TRPV4 knockout or piezo? I mean, not piezo1, but TRPV4 knockout, and what is known in diabetic retinopathy? And also another point I wanted to note: you show that in TRPV4 protein active levels are gone down in diabetic retinopathy. Despite that, when you give an agonist, you're rescuing it, and how is that working?
That's very interesting. So I'll answer your second question first, if that's okay. So TRPV4 agonists are known to increase the expression of TRPV4 as well. We see that in our findings, and others have reported that too in the cardiovascular field. So I don't know if it is something to do with this particular agonist from GSK that we use, but this is sort of consistent with what's known in the literature. So that's why we are not very surprised. And your first question was TRPV4 knockouts. We haven't. But we understand that the TRPV4 agonist that we administered in the mouse was IP. That's not the ideal way to target TRPV4. It was more of a proof of concept. So if you've just received adeno-associated virus that can selectively upregulate TRPV4 in the retinal vasculature. So we are taking that route to address the importance or the role of vascular versus non-vascular TRPV4, because TRPV4 is ubiquitous in the retina. It's not specific to the endothelium by any means. But it was a good proof of concept that if you can increase vascular TRPV4, you at least can suppress the vascular activation markers. But yeah, there's a lot of work that needs to be done in that direction. Yeah.
Thank you so much.
Thank you.
All right, thank you so much. We're on to the last part of this session. Last but not least, Dr. Kyle Mara is a resident of ophthalmology at the UCSD Shiley Eye Institute. He completed his MD PhD at UC San Diego in bioengineering under Martin Friedlander's lab. Very excited for his talk today. It's entitled Bioactive Extracellular Vesicles from a Subset of Endothelial Progenitor Cells Rescue Retinal Ischemia and Neurodegeneration. Thanks.
Thanks for the introduction, and thank you for the invite to present my work. As you mentioned, I'm Kyle Mara, and I'm going to be presenting a selection of data from my dissertation research in Marty Friedlander's lab at Scripps Research. He mentioned the title of my talk, and to begin, I'll start with some background. The majority of retinal diseases are caused by abnormal angiogenesis. And current therapeutic agents include VEGF antagonists, which aim to prevent VEGF-induced endothelial cell proliferation and vascular leakage, or panretinal photocoagulation, which destroys peripheral retinal tissue to reduce metabolic demand to match blood supply in the remaining central retina. And while these therapeutic approaches have been effective in restoring vision in a subset of patients, these strategies target manifestations in later stage disease, and they don't really do anything to target and alleviate the ischemia that drives the abnormal pathologic angiogenic response. A promising alternative strategy is through the promotion of physiologic vascularization and vessel stability through the use of stem or progenitor cells. Years of research, including multiple publications by the Friedlander lab, have characterized endothelial colony-forming cells, or ECFCs, that function as true endothelial progenitor cell types, and they may serve as a promising cell source to achieve just this effect. The use of progenitor cells is often envisioned as transplanted cells acting as replacement parts for tissues that are lost or damaged or diseased. And this is usually envisioned as them integrating into, in this case, the endothelium. However, publications from the Friedlander lab and others have demonstrated two important findings. First, Friedlander lab demonstrated that ECFCs were not actually a homogeneous group of cells functionally. ECFCs with high expression of the hyaluronan receptor, CD44, were effective in rescuing animal models of ischemic retinopathy and retinal neurodegeneration. On the other hand, ECFCs with low expression of CD44 or CD44 low ECFCs do not. And secondly, rather than integrating into host vessels, endothelial progenitors were observed to remain in the vitreous of the eye, and they assumed perivascular positions in regions of retinal ischemia. This suggested that ECFCs, rather than forming mosaic vasculature with host vessels, deliver their effect via a paracrine mechanism. And that's when I entered the lab. My hypothesis was that this paracrine mechanism was via extracellular vesicles. I proposed that the improved neurovascular trophic effects of CD44 high ECFCs was mediated at least in part by extracellular vesicles released by CD44 high cells. So extracellular vesicles is actually a blanket term for three distinct types of bioactive cargo-filled vesicles that are shed from cells. Exosomes are one that we're growing to be more familiar with as the literature continues to grow. There's also microparticles and apoptotic bodies. Exosomes are the smallest in size. They range from 40 to 120 nanometers, and they are of endosomal origin, whereas microparticles are a little larger, 51 to 1,000 nanometers, and they're generated by outward blebbing of the plasma membrane. And then finally, apoptotic bodies are shed during programmed cell death. Why are these interesting? Well, they're loaded with a diverse array of proteins, small molecules, and nucleic acids, and EVs facilitate intracellular communication both near and far through the transfer horizontally of this bioactive cargo. And so as a result, EVs can target multiple cell types with multiple mechanisms all at the same time, and this multimodal approach is a promising novel strategy towards treating more complex retinal disorders. More practically speaking, extracellular vesicles are also a promising therapeutic option because of their resilience to long-term storage, a low immunogenic profile, and since they're devoid of replicative machinery, they circumvent the risks associated with tumorigenesis that is associated with intact stem and progenitor cell therapy. So here's the spoilers or take-homes from my data. My data show that EVs harvested from these CD44 high ECFCs, or I'll be talking about them as EVs high, rescue the murine oxygen-induced retinopathy model of ischemic retinopathy and the RD10 mouse model of retinal neurodegeneration, whereas EVs harvested from CD44 low ECFCs, referred to as EVs low, do not. And when I injected into the OIR model these EVs, the EVs high home to sites of ischemia and accumulated in pre-retinal neovascular tufts, as well as perivascular macrophages and microglia. And finally, knockdown experiments and small RNA sequencing demonstrated that the vasculotrophic and neurotrophic effects of EVs high is reliant on combinations of specific microRNAs. So EVs were sorted into CD44 high and CD44 low populations and then plated. Conditioned media was then subject to a EV isolation protocol where I, in brief, employed ultrafiltration to concentrate the sample, size exclusion chromatography to separate vesicles from soluble proteins, and then repeat ultrafiltration to make the sample more dense in its composition of EVs. And once these EVs were obtained, they were first evaluated in the OIR model. In this model, newborn mice are exposed to 75% oxygen beginning on day 7 of their life, and they remain in oxygen for 5 days, during which the retinas undergo a process of vasoobliteration, whereby the central retina becomes devoid of vasculature. When the mice are removed from oxygen, hyperoxic conditions on P12, they're immediately in a state of ischemia, and that drives a pathologic neovascular phase that lasts until P17. And this results in the formation of pre-retinal neovascular tufts that can be quantified. So in this model, mice are typically treated at day 12, immediately upon removal from the hyperoxygen into room air, and they're sacrificed on P17 to assess for 2 key outcome measurements: the percentage of retinal area covered by this neovascularization, which is shown here in red, and the percentage covered by vasoobliteration, shown here in yellow. And we quantified this now with a deep learning algorithm available at OIR-SEG that I assisted in the creation of during my PhD. So in OIR, EVs high rescued both the neovascularization and the vasoobliteration. EVs low failed to rescue both NV and VO. And EVs from HUVEX also had no effect, which suggests that this effect may be endothelial progenitor cell specific. Some additional controls were non-conditioned ECFC or XFM media or HUVEC control media, M200, that was subjected to the same EV isolation protocol so that I can ensure that no component of the media was responsible for the effects of the EVs that we were seeing in EVs high. And then finally, when EVs high the sample was depleted of vesicles via an overnight spin, the sample no longer rescued OIR as well. EVs high were then stained with the lipophilic dye CM-dye before injection into the OIR model, and immunohistological chemistry showed that EVs high within pre-retinal neovascular tufts are seen in figure A. And they're also seen within superficial macrophages and microglia in figure B. EVs high were also found within lectin-stained microglia throughout Z-stacks of the retina, which prompted me to create retinal cross-sections in C through E. And you can see in figure C, you have colocalization of the CM-dye stained EVs with microglia and macrophages in the retinal ganglion cell layer in C, in the internuclear layer in D, and in the outer nuclear layer in E. The neuroprotective effects of EVs high were then also tested, and we used RD10 mice with inherited retinal neurodegeneration. In the RD10 mouse model, mice begin to experience photoreceptor cell death around P21, and that begins concurrently with atrophy of the deep vascular plexus. By day 45, the outer nuclear layer is degenerated to about one cell layer thick through apoptosis, which begins around P25. And mice in this model are typically treated around P14, and key outcome measurements are, at least the ones I'll be showing, are apoptosis in degeneration in the outer nuclear layer, as well as retinal function measured by electroretinography. When mice were injected on P14 and sacrificed at P28, tunnel staining revealed significantly less apoptosis in the ONL of mice treated with EVs high. And EVs high also preserved the thickness of the ONL on P28 as well. And in order to investigate the effects of EVs high on preserving retinal neural function, RD10 mice were then again treated on P14, and ERG measurements were taken on P42. Not shown were ERG measurements also taken on P28, but both demonstrated that EVs high significantly improved both rod and cone-driven signals at P42 compared to EVs low or untreated controls. So these data and additional literature informed my second hypothesis that the improved neurovascular trophic effects of EVs from CD44 high ECFCs is mediated, at least in part, by neurotrophic or vascular trophic microRNAs that are upregulated within these EVs high. And so to investigate the reliance on CD44 high cells and EVs from CD44 high cells on intravesicular microRNA, ECFCs were first transfected with short hairpin RNA to knock down dicer1, a protein involved in the biogenesis of microRNAs. And eyes were then injected with the ECFCs first and demonstrated a failure to rescue the NV of the model. EVs were then isolated from these cells, and injection of EVs from ECFCs with knockdown expression of dicer even more drastically failed to rescue the model. Both NV and VO were failed to rescue in OIR mice. And together, these data suggest a critical role for intravesicular microRNA in the mechanism of EVs high. So I subjected samples of CD44 high ECFC-derived EVs and CD44 low ECFC-derived EVs to small RNA sequencing. And by comparing their intravesicular cargo on small RNA sequence, I found 9 upregulated microRNAs in EVs high. 7 of these 9 I was able to successfully validate using RT-qPCR, and I identified these as candidate, quote-unquote, candidate microRNA that may play a mechanistic role. But this was still kind of a long list, so to narrow this list of candidate microRNAs, I injected mimetics of each of these candidate microRNAs into the OIR model, and then evaluated which of those were neurovascular trophic. And 6 of these microRNAs, as listed here on the left, rescued neovascularization, while 3 of these microRNAs rescued both neovascularization and vasoobliteration, seen here on the right. And so the last few slides were a good deal of data, and this is a kind of simplistic way of looking at it. And altogether, these data made a short list of candidate microRNAs. And these were microRNAs that were upregulated on microRNA sequencing, validated on qPCR, and trophic in OIR mice. And they included microRNA 7, 5P, 23A, 3P, 216A, 3P, and 503, 5P. ECFCs with knockdown expression of individual and combinations of candidate microRNAs were then generated, and then their EVs high were injected into OIR. And EVs high from cells with knockdown microRNA 503 expression no longer rescued NV, while MER23A knockdown in ECFCs produced EVs high that no longer rescued VO. When all candidate MERs were knocked down in the cells and EVs high were collected and injected into OIR, then the EVs high no longer rescued both NV and VO. And so the role of microRNA 5035P as an angiogenic as an anti-angiogenic agent is pretty consistent in vitro and in vivo across the literature. For example, in cancer models like glioma, in colon cancer, or hepatocellular carcinoma, MER5035P has been shown to inhibit tuber angiogenesis by targeting either VEGF-A or VEGF-A regulators. It was also, MER503 was also found to be downregulated by hypoxia-induced expression of HIF-1 alpha in tumor cells, which leads to the increased expression of VEGF-A and other vascular growth factors. So in summary, literature suggests that 503 is a promising therapeutic target in regulating angiogenesis in response to hypoxia. In addition, MER23A was shown to be was shown to decrease oxidative stress, decrease neuron apoptosis, and neural inflammation in order to improve both vascular and neural outcomes. So in summary, these data have demonstrated that EVs shed from ECFCs with high expression of CD44 provided neurovascular trophic rescue to mouse models of ischemic retinopathy and retinal neurodegeneration. EVs high home to areas of ischemia and assumed perivascular, accumulated within preretinal neovascular tufts, as well as perivascular macrophages and microglia throughout the layers of the retina. And third, a collection of microRNAs found to be enriched in EVs high demonstrated neurovascular trophic effects in OIR, and combination of these MERs account for, at least in part, the multimodal mechanism that EVs high account for in vivo. I'd like to end here with an acknowledgement to my lab. Dr. Martin Freelander was my PI, and he's been an incredible mentor. And everyone here contributed in some way to the development of my research skills and project. I also want to acknowledge the funding from the Lowy Medical Research Institute, UCSD's T32, and I received a F30 to support my MD PhD as well. Thank you.
We have time for a few questions.
Thank you. Sarah Ham Alvarez from USC. That was very nice work. I have a couple of unrelated questions. The first has to do with the origin and packaging of the microRNAs in the CD44 positive cells. Do you know what's unique about those cells, or if you transfect CD44 into the low expressing cell line, can you convert the extracellular production? The second unrelated question is, can you speculate on the use of lipofectamine or another agent to deliver the 503-3P instead of endogenous vesicles, which may be difficult to purify and to isolate in sufficient quantities?
Sure. So in terms of knocking in or knocking out CD44, that's kind of been a age-old question in the Friedlander lab. They've known for many years that CD44 high cells, whether they're lineage negative or whether they're bone marrow progenitor cells, all have better effects than CD44 low cells. But the mechanism behind CD44 high is not really clear just yet. We have had some, I started some experiments where we injected CD44 high ECFCs that were GFP positive and CD44 low ECFCs that were GFP positive in retinal, in the mouse models, and then I was able to rescue those cells back by flow cytometry. And then I got those samples. I was going to do some small RNA sequencing or PCR, but unfortunately that's when COVID hit. So I don't actually have the clearest idea of what the role of CD44 is. I do know that when you subject the EVs to single particle flow cytometry, there is no difference in the level of expression of CD44 on the surface of EVs. So that leads me to believe that it's more likely a factor of what's in the vesicles in this scenario than what's on the vesicles. But I think a lot more research needs to be done to really make that a conclusion, per se. In terms of packaging with lipofectamine, I haven't myself done experiments of that nature to date. I think it is certainly worthwhile trying to extrapolate which microRNAs from this data are worth developing independently of EVs, which admittedly are containing multitude of other things besides just 503-23A. But we can kind of glean ideas of what might be useful from these types of experiments where we know within this natural packaging of vesicles, delivery of that cargo is effective. But specifically, no, I can't say I know exactly what would happen if you package them otherwise.
Hi, nice talk.
Do you have any idea what makes the EV's high home to areas of ischemia? Why not universally?
Yeah, I don't know exactly why, but I know that that is a quite useful trait of ECFCs as a whole. So in models of neural infarction elsewhere in the literature, if you inject ECFCs into the contralateral hemisphere, they will still home to the area of ischemia. So it's some type of maybe innate cellular function that they are able to understood where they're needed, but the mechanistic component of that is still not well understood.
One last question here, and then we'll head to break.
Oh, hi. I was wondering, when on your initial slide on extracellular vesicles, you mentioned that they have low immunogenicity, and I was wondering if you've done any like comparative like experiments to show like how the immunogenicity of extracellular vesicle compares to just things like lipid nanoparticles, which are like very kind of common things that people use to like deliver therapies and whatnot.
Yeah, so the immunogenicity of every extracellular vesicle is kind of dependent on cell source and also the conditions upon which that cell was placed. So I have looked at the surface profile of these vesicles, and I see HLA ABC isotopes, but in very, very small expression. And that has been corroborated in the literature, which is able to kind of co-culture them and demonstrate a low immunogenic production when they're cultured with like macrophages, microglia. They're not significantly upregulated in the expression of inflammatory markers. But that's always in comparison with other vesicles. And so particularly mesenchymal stem cells are known to have a very, very low, almost control type of immunogenicity in these experiments. I haven't seen anywhere where they've been compared to other non-vesicle types. So I can't really speak to whether they compare favorably or not to, let's say, nanoparticles.
Oh, thanks, appreciate it.
No problem.
Just wanted to compliment you. You're at this stage in your career, you're, I think, going to start your residency in a month.
Yeah.
And for the body of work you've done and the presentation, pretty impressive. So
Thank you.
Kudos. That was really nicely done.
Thank you. Appreciate it. Take care.
And please make sure to apply here for fellowship.
Thank you, Dr. Mara and Dr. Kupperman. A couple of quick announcements for me before the coffee break. Dana Collinson asked me to mention that we have the clinical colloquium meeting of the Eye Institute, September 6th and 7th, which is Friday and Saturday. So if you have interest in attending, reach out to her and she'll give you details. And she also wanted me to correct myself. There will be no dinner. It will be just a reception after the poster. So don't get your high hopes too high. But the reception will be pretty good. So again, stick around. We'll take about 10-minute break for coffee and maybe a couple of more posters. And we'll reconvene here at 2:55 for the last two sessions. We'll have those back-to-back, and then we'll have the poster ceremony at 5 o'clock. Thank you.
Okay, good afternoon. My name is Andrew Smith. I'm assistant professor here at the Eye Institute, and I'm going to be starting out the glaucoma session. And so it's with my deepest honor to be able to introduce our first speaker, Dr. Natik Piri. He earned a PhD in molecular biology from USSR Academy of Sciences in Moscow, in Moscow, and then has been at the Jules Stein Eye Institute since 1991. He's done a lot of research in about retinal ganglion cells, and I think we're going to hear a little bit about that now. And so I'll turn the time over to him.
Thank you, and thank you organizers for inviting me to speak at this symposium. So today I'm going to talk about some of our research towards designing, we hope, clinically relevant treatment options for glaucoma. Oops. So why do we need new or alternative treatment options for glaucoma. We know that glaucoma is treated by lowering and managing intraocular pressure, and it works for many people well. But in some patients, disease is still progressing, and that can lead to loss of vision. The problem is, or difficulties with designing alternative treatments, is that glaucoma is a multifactorial disease. So multiple factors involved in different types or different forms of glaucoma, and those factors may be also different in different patients within same type of glaucoma and may change with the progression of disease. So here's some of the factors that have been identified to be associated with glaucoma pathogenesis. If you think about designing treatments based on the genetics of glaucoma, we know that, according to some estimates, less than 10% of primary open-angle glaucoma are associated with mutations in one specific gene. So most cases are polygenic and involve multiple genes. If we look at GWAS studies, only several years ago, in 2019, 74 loci for glaucoma, primary open glaucoma, have been identified. In 2021, we have already 127 genomic loci associated with primary open glaucoma. So it's a complex disease in terms of pathophysiology and also genetics. So neuroprotective studies and other alternative studies have been proposed for treatment of glaucoma. And here's a list of common factors that have been targeted by neuroprotection. Regeneration, stem cell therapy, and cortical implants also have been proposed. And it seems like, at least in my opinion, cortical implants can be very promising to treat patients with complete or near complete loss of vision, since neurons in primary visual cortex and or V4 are targeted directly, bypassing the eye optic nerve and LGN. But let's talk a little bit about neuroprotection. Here's pretty much latest information about clinical trials that involved neuroprotective strategies, was taken from this paper published in May of 2024. And you can see different strategies have been proposed to be evaluated, including growth factors, compounds targeting metabolism, anti-inflammatory drugs, some supplements that can improve mitochondrial function and metabolism in general, stem cell therapies. If you pay attention, most of these strategies are designed to target one specific factors. And if you have in your group of patients with disease that are not, for instance, affected by mitochondrial dysfunction, or it's not the major factor that involved in glaucomatous neurodegeneration, so probably only those that have mitochondrial dysfunction as an important factor will benefit from this study or from this treatment. The others won't, or to very little extent. So we're thinking more about finding treatment that can be applied to as many people, as many patients as possible. Of course, there's no treatment that fits, one treatment that fits for all. But we think that if we target certain protein or pathways that are responsible or involved in stress response, those pathways could be beneficial in terms of treating as many patients as possible. So we know that aging is a major factor for glaucoma development. And aged cells perform their function normally until they are stressed out. So if you have spikes in metabolic stress, these cells are more vulnerable, they can be damaged, their function can be affected, and they can degenerate or die, right? So the important mechanisms that are involved in protection of cells from stress are stress response mechanisms, which include heat shock protein response, UPR, unfolded protein response, oxidative stress response, DNA damage response. We particularly interested in heat shock response because members of heat shock response, particularly heat shock protein 70, is known to be both anti-apoptotic and also can repair the damage in terms of refolding damaged protein, prevent protein aggregation, and by doing that, repair those cells that anatomically are normal. They have their synapses connected in both retinas and in LGN, but functionally, their function is affected. So in normal cells, in young cells, stress induces heat shock response, shown here. I'm not going to describe the entire pathway of activation of heat shock response. If you have questions, I can answer them later. But in aged cells, heat shock response is diminished, and that basically makes these cells more vulnerable to stress. We don't know exactly what's the mechanism behind diminishing stress response in aged cells, but in C. elegans, this deactivation of stress response has been associated with reproductive period. So basically, once they pass that reproductive period, C. elegans don't use their stress response is inactivated. So now I'm going to talk about some of our studies. Of course, without going into details, animal models that we use, there's 3 animal models. It's intraocular pressure elevation model shown here, and we use laser trabecular photocoagulation to increase intraocular pressure and maintain for at least 5 weeks. And that's the systems that we do our studies, including neuroprotective studies. And all studies are performed while we maintain the elevated intraocular pressure. In this model, we observe cell loss at 2 weeks about 10%, and at 5 weeks about 20 and 30%. And that is a design to mimic human glaucoma, which is relatively slow progressing disease. The other models we use is optic nerve crash, and sometimes we use also optic nerve transection. Those are very severe damage to optic nerve models, and they lead to rapid specific degeneration of ganglion cells. And by 7 days, you have 50% loss. By 2 weeks, almost 90% or even more retinal ganglion cells degenerate. So one interesting observation we made that links stress proteins to glaucomatous damage is that when we analyze retinal transcriptomes in these animals models for glaucoma, we see coordinated downregulation of crystalline genes at 2 weeks after IOP elevation. So it appears that IOP elevation decreases the expression of number of crystalline genes, and we show that by PCR and its data from retinal transcriptome, also at protein levels. The dynamic of these changes in crystalline expression is changing with more damage. Expression of crystallines goes up, but we're seeing that the reason why it goes up because the entire retina under the stress, and they induce expression of crystallins. We know about crystallins as a major lens proteins, but in fact, these proteins express in different tissues, and they have high homology with small heat shock proteins. They're considered molecular chaperones, and in most stress situations and due to trauma, there's an overexpression of crystalline, which is understandable because cells trying to deal with this stress and preserve cells from damage and degeneration. And that has been shown in many neurodegenerations in humans and animal models, as well as injuries to the retina. But in 2 other glaucoma models, they also observed downregulation of crystallins, so it appears to be consistent with our observation. So we think that downregulation of crystallins can compromise cell survival and therefore contribute to cell death in this animal model. And we also showed, I think for the first time, that expression of crystallins is very strong in retinal ganglion cells, and that makes our hypothesis about their involvement in glaucomatous degeneration even closer. So to prove whether or not crystallins have cell protective effect or retinal ganglion cell protective effect, we made expression constructs and transfected retinal ganglion cells by electroporation, which is minimally invasive. Of course, I'm not proposing to use as a clinical treatment, but it's fast, but at the same time not very efficient because only 40% of retinal ganglion cells were transfected. But we think that non-transfected cells can be used as internal control. So by transfecting this 40% of retinal ganglion cells, we observe about 95% increase in cell survivals in retinas transfected with alpha A, and about 75% increase in cell survival in retinas transfected with alpha B. And those are 2 main crystalline that are known to be stress response members of crystallins. So, and we know about those proteins more than about others. So, and those observations were made in animal models with direct injury to optic nerve. In this study, crystallins were associated with neuroprotections in animal model for glaucoma or intraocular hypertensive animals. These animals were fed with LBP, which is lithium barbarin polysaccharides, and it's an active ingredient in goji berry or lithium barbarin or wolfberry. And feeding of this LBP to rats pretty much prevents the cell loss in these animal models. And crystallins, both alpha and beta crystallins, were upregulated in these retinas 10 times, suggesting that crystallins are the proteins that provide this support for cells in this animal model. You can buy goji berries in health stores like Whole Foods, Sprouts, or what have you. And there are also supplements, goji LBP40, you can buy at Amazon or some other sources. So we think that this could be a clinically relevant treatment options for glaucoma, at least in some patients. Our main focus when we talk about stress proteins is heat shock protein 70. It's known to be the major protein of heat shock's protein stress response, And it has not only anti-apoptotic functions by binding various caspases or pro-apoptotic factors, but they also can prevent protein aggregation or misfolding, or if proteins are damaged beyond the repair, HSP70 can assist their degradation. So by doing that, we're not only hoping to prevent or increase the survival of ganglion cells in glaucoma, but also restores the function of those cells that are intact but lose their function. So as a proof of principle, we made AAV2 vector that codes for HSP70, and we transfected retinas with this vector. Most cells that were transfected, as expected, were retinal ganglion cells because AAV2 has high affinity to retinal ganglion cells. Transfection wasn't uniformal. Inferior retina was transfected much efficiently than superior retina. Overall, if we count cells in the entire retina, we see about more than 100% increase in cell survival rate. In inferior retina, the effect was more than 300%, confirming that HSP70 has strong cell protective effect. So again, we're not proposing AAV2 HSP as a treatment option for glaucoma patients. Therefore, we look for alternative ways to stimulate expression of heat shock protein 70. And there are a number of drugs or compounds that have been approved by FDA, and they can stimulate expression of HSP70. And they're doing that by binding to HSP90. Again, I'm not going to go over the entire pathway, how HSF1 stimulates expression of heat shock proteins. But we actually decided to use 17-AG and cilastrol because these two compounds were shown in multiple clinical studies that they can have cell protective effect in various animal models for neurodegenerations. Today, I'm not going to talk about 17-AG study. I'll cover cilastrol study. Cilastrol is a triterpenoid which is isolated from thunder of God vine. It's been used in traditional Chinese medicine for many hundreds of years. And like I said, in comprehensive drug screening, it was shown very strong cell protective effect in vitro and in vivo models for neurodegenerations. The cell protective effect of cilastrol has been associated with several pathways. Number one, it's activation of heat shock stress pathway. It can also induce expression of proteins that have antioxidative effect, and that will reduce oxidative stress and damage. And it also can diminish microglial activation. All these factors have been associated with glaucoma pathogenesis. Okay, we observe strong cell protective effect by intraocular injection of cilastrol and also by systemic injection or systemic delivery of cilastrol. One side effect we thought it's a side effect was that animals treated with cilastrol did not gain any weight, and later there was a publication in Cell that cilastrol can be used also as treatment for obesity. So patients, if they treated with cilastrol, have double benefits if needed. And those studies were performed in animal models with crash, optic nerve crash. In animals models with ocular hypertensive, again, we see a significant increase in cell survival And in fact, number of cells in celestral treated animals were only 5% less than those without intraocular pressure elevation. And in conclusion, in aged cells, the endogenous heat shock response and other stress responses are diminished. And that leads, and that leads to deterioration of stress adaptation. Crystallins and heat shock protein 70 show strong cell protective effect, and they can be induced very safely and effectively with goji berries or LBP and celestral. And using celestral and HSP-based strategy can add to treatment options, can be added to treatment options to protect retinal ganglion cells and improve their function. Thank you.
Thank you so much, Dr. Peer. I think maybe we have time for one question as Dr. Zoda is making his way up. I will ask one question if that's okay. Oh, do we have somebody? Oh, perfect.
I was intrigued by your idea of the goji berries. They're also the highest natural source of zeaxanthin, which is really good for your macula. So you could help protect against two major diseases by throwing a few berries in your cereal in the morning. Just thought I'd mention it.
Okay, thank you. We'll move on. Thank you so much.
All right, thank you.
Okay, next I'd like to introduce Dr. Gulab Zod. He comes from here. He is a professor of ophthalmology at UC Irvine. He did his PhD work at the University of North Texas in cellular biology, and he's going to be talking to us about the development of mRNA-based lipid nanoparticle delivery systems for genome editing targeting glaucoma. I'll turn the time over to you. Join me in welcoming Dr. Zod.
Thank you very much. I'd like to thank Vladimir and Dr. Vladimir and Ken for giving me this opportunity to present our work. I'm excited and honored to be here today. So today I'm going to show you data of what I have been doing at UCI since last year. So my lab is interested in understanding the pathophysiology of glaucoma and targeting this pathology by using small molecules or genome editing. So as you know, glaucoma is a group of optic neuropathy, and in the clinic, it is diagnosed as increased cup-to-dish ratio, and then this is associated with a peculiar optic nerve degeneration and lamina cribrosa excavation and cupping. And it is also associated with the loss of retinal ganglion cells. So this is the major pathology that leads to loss of vision. And this axonal damage extends all the way to the brain. Now, in the front of the eye, elevated intraocular pressure is the major and only modifiable risk factor. And we are interested in elevated intraocular pressure because we know that when the intraocular pressure goes up, it leads to retinal ganglion cell loss and similar vision loss. So to understand that, we need to understand what creates intraocular pressure in the eye. So if you can see here, intraocular pressure is created by aqueous humor dynamics. The aqueous humor is made by ciliary body. It flows through the posterior chamber into anterior chamber, providing nutrition to avascular system, and it filters through this specialized structure called trabecular meshwork. So this is nothing but a filter, which has lot of ECM, and you have a cells called trabecular meshwork cells that line that ECM, and it allow the passage. So if you look, the reason that this pathway is important because in primary open angle glaucoma, which is the major cause of glaucoma, about 70% people are of total glaucoma, there is increased resistance to aqueous humor path at this junction in the trabecular meshwork, leading to elevated intraocular pressure. So, and also I want to show you that why this pressure goes up is because if you look at this trabecular meshwork in the people, and these are the OCT images, this is an active process, and this is not an intrinsic, it is the contractility you're seeing it's a pulse given by the blood flow, and it is an active process that pumps this, allows this fluid flow. So in glaucoma, what happens in the trabecular meshwork, that this tissue becomes stiffer. We talked about stiffness, so this is another example of becoming stiffer. What we are interested in, the molecular pathways that causes this increased stiffness. So when you look at it, so all the things that happen in this tissue, and this is just the human eye from a normal donor, and this is from a glaucoma donor, and we see increased fibrosis. Here it is stained with one of the marker. We see reduced phagocytosis, which is required by these cells to take anything that comes in the pathway. And there are a lot of other things like increased active cytoskeleton changes and loss of the trabecular meshwork. So the current treatment, although the major pathology lies here in glaucoma, but the current treatment, at least the drug treatment, reduces intraocular pressure by reducing aqueous humor production, and some of them also increase the UV scleral outflow pathway like latanoprase. The surgical treatment allow the passage of fluid through trabecular meshwork, but they don't address the underlying pathology. So when I started my career, I was interested in understanding what happens to trabecular meshwork in glaucoma. So one of the way we approach this is using genetics. As you know, glaucoma is mostly about 90% of glaucoma, although it is complex, it is heritable. But we still don't know all the genes. But among the genes we know, myocilin is the single leading genetic known cause of glaucoma, primary open glaucoma. And this is showing in advanced stages, so about 30%, but in general, myocilin causes about 5%. Myocilin mutations are responsible for 5% POG cases. But it is associated with most cases of familial early onset glaucoma. So at the protein level, we do not know the function of the wild-type myocilin. It is a secreted protein. Most of the mutation, almost 100 mutations have been discovered. Most of them are in olfactomediene domain. One of the peculiar thing about this is in clinic, a lot of kids come with high pressure, and they end up with, if you look at it, most of them have myocilin mutation. So it is a big issue because they have to go through a lot of treatment and many surgeries. So and another thing about the myocilin is that the reason we are interested in this is because it's highly expressed in trabecular meshwork. So, any insult that you provide to this anterior segment, the TM cells make a lot more myocilin. So when this was initially discovered 20 years ago, a lot of groups actually tried to address what does it do. And all these studies, what they show is that the mutation lead to gain of function. So if you can overexpress myocilin, it doesn't have any phenotype in mice. Knockout of myocilin in human or in mice doesn't have any phenotype. You can also create a knock-in of a myocilin from the mouse. It doesn't have phenotype. So these studies actually indicated that there is something about the human mutant myocilin that is responsible for this disease. So when I was doing my postdoc, we created a mouse model where I got lucky and we saw that our mouse model overexpressed the mutant myocilin in the trabecular meshwork. And not only that, we saw elevation of IOP right from the age of 3 months, similar to POAG. And we showed that this mouse develops glaucoma, similar to what we see in the human patient. So, and at the molecular level, when we look at it, the wild type is secreted, but when you express the mutant one, which is tagged with the DS red, here you see that most of it stays in the ER, and it ends up causing ER stress, and which leads to pathology. So long time ago, we've shown that this pathway, that human mutant myocilin
leads to misfolding, and then instead of being secreted, it stays in the ER, causing the phenotype. So at the same time when we published this, the genome editing came out, and we were interested in targeting this gene by using CRISPR-Cas9. And that's where we met Feng Zhang, and we started this project. And the whole idea was that we can knock out this gene, because as I mentioned, people with the knockout of myelin or mice, they don't have any problems. So this makes easy target to just knock out the gene using the CRISPR-Cas9. And another thing is that this also provides one opportunity to take this genome editing to the clinic, and this myelin is one of the best example to target this. So here, I'm not going to go too much about CRISPR-Cas9, but we can engineer the Cas9, which is endonuclease, and we provide the guidance by using guide RNA. And if you have a nice delivery system, it can go to your cells, target the DNA wherever it needs to, and can create, when you're talking about Cas9, it can create a double-strand break. And depending on which mechanism you target, it can either cause the indel, so it can cause a knockout, or you can insert the right copy. So in this case, we targeted a knockout of myelin by targeting exon 1, and we published this before. Sorry, I think I missed the one slide here. Yeah. So one of the things when we talk about Cas9 is that how do you deliver to the cells? So here, initially when we started, these are the various methods you can deliver. From viral vectors to non-viral vectors. And so when we started out this with adenovirus, and we demonstrated in this paper that we can deliver Cas9 targeting myelin and rescue mouse model of the myelin glaucoma. So this is just an example where we have cells that overexpress mutant myelin, and when you do the Ad5 CRISPR-Cas9 treatment, we can rescue this phenotype. We can also rescue the mouse model, as you can see here. But one of the problems we had with Ad5 is that it causes inflammation. So we went to the next level, and we used the AAV vectors and a lentivirus. So I'm just showing the AAV here. Although AAV it's much safer, it's not specific. And most of the trophism is in the retina. And then next we use the lentivirus. So when we use the lentivirus, we see specific trophism to TM and corneal endothelium. So we further demonstrated that when you used in the cell culture model, the lentiviral mediated CRISPR-Cas9 reduces the protein while AAV doesn't. And we can also rescue the mouse model of glaucoma by giving the intracameral injections of this lentivirus expressing Cas9. So these are the control, and these are the one treated with the CRISPR-Cas9. So at this stage, one other important aspect is the off-target effect, because that's one serious side effect of the CRISPR-Cas9. And you see, this is whole genome sequencing, and you can see that although our we target the myelin gene perfectly fine, it also targets other genes, including this, and with a high efficiency. So this doesn't make good sense to go forward with this kind of technology. So two major problems we had. One is the off-target effect, and also delivery method. Lentiviral may not be the best one because it expresses the Cas9 for too long, and we don't want that with the genome editors. At the same time, I came to UCI and I started working with Dr. Palczewski, and I was introduced to base editing. So with the base editing, you can be precise. It doesn't create a double-strand break, and you can use this deaminase to just kind of swap the bases, and you can convert 8 to G. And in this case, what we use here is to create, change AUG start to valine so that the transcript is not made. So in this case, we also got interested in the lipid nanoparticles. As you can see here, we started working with Dr. Felgner group, and he's here today, so thank you. Here we make And the approach is that we want to deliver mRNA for Cas9 or base editor, and we encapsulate that with the lipid nanoparticle and guide RNA. So this is exciting because not all cells like mRNA or lipid nanoparticles, but our cells, these are trabecular meshwork cells, they're highly phagocytic. So in this case, it would make perfect sense. So I'm just showing you some data a lot of data that was generated by Dr. Shankar, who's my postdoc. And first, we just take a simple experiment. We injected lipid nanoparticles, LNP, expressing M-cherry mRNA. When we inject in the intracamerally, we see in the trabecular meshwork. And it's highly specific. It's nowhere else, it's only in the trabecular meshwork. We can also use the human donor eyes, and we can perfuse them. As I mentioned, in glaucoma, there is increased resistance to aqueous humor. And you can measure that in ex vivo model. We take a normalized, has a normal pressure. If you have a glaucoma eye, it has a high pressure. So we just perfuse the normal eyes with this M-cherry LNP, and we see it in the trabecular meshwork. So it shows the human significance. Then we wanted to see whether this actually shows any activity. So in this case, we made LNP Cree, and we used this reporter mice called MTMG. And the way it works is that this mice express red color everywhere. When you add Cree, tissue-specific manner, it converts red into green. So here you can see this. This is the trabecular meshwork whole mount, and this is control where you can see the TM region in this way, in this area. But when we add LNP Cree, we see red conversion into green throughout all the way in entire trabecular meshwork. So that shows that the Cree injected with LNPs is functional. So here you can see in the cross section, and here is the TM region. This is cornea, ciliary body, and then here you have the TM, only TM region where you can see the conversion of green, red to green, indicating Cree activity. We also made a mouse model where we can induce the mouse model by adding Cree. So I'm going to skip some of these, but I'm showing that you can induce our transgenic mouse that expresses mutant myelin. So we start with the normal mice, we induce with LNP Cree, then it expresses mutant myelin. So you elevate IOP, and it sustains IOP elevation. Then you can go and we made the LNP AB mRNA. And we just used the Pfizer formula, and we just took the AB mRNA and then mixed the guide RNA, and this is all the work done by Dr. Felgen in the lab, and he just gives us this formulation, and we inject intracamerally in the mice. So here we have LNP Cree injected mice. They elevate IOP, and after they elevate IOP significantly, at this stage we make them into two groups, and one group gets PBS, another one gets LNP-AB. And after LNP-AB injections, we see there is a reduction of IOP, while the other group keeps the IOP higher. And then we can also do the editing. Now here, we just collect the DNA from the tissue. We do the sequencing in this region, and you can see that we see from ATG to GTG, showing the specific change due to these base editors. Now you'll see that the editing efficiency is about 6%, which is not so great, but I just want to show you that when we take the tissue, we take the entire tissue. The editing is right here, so what we see here is like 5-fold less. So hopefully we can get a lot more editing when we isolate this tissue. Not only that, we can actually see in the cells that the Cas9 protein is made by these LNP Cree. So this is Western blot showing the Cas9 protein. As well as in the cell culture, you can see that the mutant protein is reduced after you add LNP-ABE. Again, I think the take-home message is that gene editing for targeting myelin could be the first glaucoma gene therapy, and we just need to have a lot of preclinical studies and then safety studies to make sure we can go to a human subject. And then I like to thank my team. Thank you.
Thank you, Dr. Zode. Are there any questions?
Thank you. That was fascinating. Just a quick question. Pediatric glaucoma is very difficult to treat, and because of the diverse mechanism, and just like you have shown on your graph, I think mycilin is probably one of the many causes. So, I could just go on the list of the various types of mechanisms of glaucoma in pediatric patients. So how do you implement the gene editing technique to treat glaucoma? How do you identify the patients with mycilin-induced glaucoma? How do you foresee that? How do you foresee this being applicable in a real world? How do you identify those patients? Did you understand the question?
Yeah, I understood the question. I'm not sure how you would go because that's part of the clinic. What I can say is that a lot of, like University of Iowa or other places, when they studied these mycilin mutation, they have the families and they know which person, like the kids come at the age of 3 months or like 5 years, they know it's because of mycilin mutations. And that's something you could target, but I understand that it's not easy because, and I think one of the things is that despite mycilin being the major genetic cause, it's not an entire, but because the treatment is the same, nobody focuses on identifying the mutations, right? Am I right? So because the treatment is the same at the end of the day. So if we make these editing treatment available, I think there is a hope that maybe there'll be more focus on looking at what genes are affected. And it's just the example of mycilin, but there are other genes from so many loci that have been identified. They have the similar mechanism. A lot of them are actually causing aggregation, extracellular or intracellular. They have the similar mechanism. So this provides opportunity. And you got to start somewhere, right? So this is one place. And the reason we target mycilin because we know so much about it. All of the mutation that we talked about other than mycilin, we don't know anything about it. So this is where we can start and hopefully we can make some significant. Yeah.
Dr. Zodi, Ken Lin here. Very nice talk. Thank you for the spectacular data that you presented. Quick question, I'm just wondering if you can comment on how much IOP reduction you observed after you edit the mycilin. Because I think one of the slides that you show, it was interesting. You can see little reductions of IOP after you genome edit the mycilin genes, but it doesn't go all the way back down to the complete wild type. And that might make sense, at least intuitively, for me to think about clinical correlations, because in people with primary open-angle glaucoma, their trabecular meshwork oftentimes, at least in the more moderate and advanced stages, their TM is so fused together that at that point, I wonder, even if you target mycilin, how much contractility or how much reductions in the stiffness can you achieve by modifying their genome? I'm just wondering if you can comment on that just based on the laboratory results you have.
Yeah, that's an excellent question. I don't think this treatment can be, because most of the patient would have such a bad pathology and trabecular meshwork. I think to start with, you would start with the young patients and where they just been identified, so we have some intact trabecular meshwork. But another thing you could do is that another aspect we study is a small molecule that also has more like a rejuvenating aspect, like HDAC inhibitors, and that we work in the lab. And there is a possibility that you combine the treatment that not only you target mycilin, but also give something that, like our small molecules, that increase contractility or make TM happy more again. So that's what I think could be possible, but to start with, you probably would need to start with the younger and some cases like that, yeah.
Thank you. Let's make TM great again.
Yeah.
What's the mechanisms why make the nanoparticle more selective for the TM cell versus other cell, while AAV does not?
I'm sorry, I didn't understand the exact question.
Yeah, so what's the mechanisms make the nanoparticle it's more cell-specific expression?
So this is the unique nature of trabecular meshwork cells. So anything comes in the aqueous humor, their primary job is to just engulf it. And the way the nanoparticles are made, if they get inside the cells, they try to escape the endosome. That's the one mechanism where they are degraded. So the Dr. Felgen's lab, when they give us nanoparticles, they are protected. And once you have mRNA release, it up to the cell to make it. So this is the unique nature of our cells. And if you look at all the lipid nanoparticle work, you'll see like only particular cells like RPE or TM, trabecular meshwork, will pick up this kind of things. Unfortunately, no RGCs, nothing can take it. So in this case, it's good because we don't want anywhere else. That will reduce the off-target effect and also make it much specific. So yeah, it's just, there's no receptor. I don't know any receptors that are expressed in the trabecular meshwork, but we know that these cells don't like all type of viruses. They don't like, they like only Ad5, but they don't do well. They doesn't transpect all of the cells. But our lipid nanoparticles actually cover almost 90% of the cells. So that's good advantage here. Okay, thank you.
Thank you so much. So now it's my great honor to introduce Dr. Alex Huang. He's Associate Professor at UCSD from the Eye Institute, also the Alfred Vogt Chair. He did his MD, PhD work at the Johns Hopkins University. He's going to talk to us about aqueous humor outflow imaging, which I'm really looking forward to.
Thank you. So actually, I'll make a comment about that last question. Because we're talking about nanoparticles, there's an explanation, depending on the size, of course, because once you're in the chamber, what we're going to talk about now is aqueous humor outflow. Everything's drawn to the trabecular meshwork. And if your size is right, you can actually get trapped there. So anyways, thank you everyone for coming. Today we're just going to be talking about aqueous humor outflow and glaucoma treatment. Thank you for having me. Here are my financial disclosures and my sources of research support, including from the NIH. So, as many of you know, particularly the clinicians, when we teach our students outflow, we use pictures like this on the left, where aqueous is made at the ciliary processes, and then outflow goes through outflow pathways. But this is a two-dimensional picture, and you have a three-dimensional eye. So you're basically implying to your students that they have to imagine that picture, rotate it 360 degrees around, and say that outflow is circumferential uniform. But there was always evidence that outflow could be segmental. There's areas going to be good, bad, here depicted by green and red arrows. And that makes patterns, where then the patterns, say, could be different between me or you, or between your left eye and your right eye. And that these patterns could then have disease relevance, where the high pressure part of glaucoma may be due to loss of these green and functional areas. So a number of years ago, we wanted to image outflow using clinically relevant techniques. So we took the spectralis, which is a tool that we can see patients with, brought it into the laboratory. It can perform fluorescein angiography. And so we took postmortem eyes, pig here on the left, human on the right, put fluorescein in the chamber, and just visualize where the fluid's flowing. So on the right, you've got a 75-year-old gentleman, died of leukemia. As the fluorescein goes in, you can see postlimbal signal or flow at 12 o'clock, which eventually moves to nasal as well as inferior nasal. Eventually, you get some signal inferior temporal as well. For whatever reason in this gentleman, not a lot of signal shows up superior temporal. And this is consistent, with the MIGS literature and what we thought, and a lot of things that, Don Minkler taught us about in terms of early development of MIGS. Now, the basic scientists have actually been down this road before, and they use fluorescent microbeads, kind of like nanoparticles, probably a little bit bigger. And what they would do is they would put these tracers in the anterior chamber, and they would be collected in the trabecular meshwork in, so to speak, high flow regions. And this was a way to segregate high flow, low flow TM as a way to understand the biology. So for example, Myrsican was an extracellular matrix protein noted to be elevated in low flow. Collagen 6, MMP3, noted to be elevated in high flow. So we wanted to see if we were similar. Oh, it's not moving forward. Oh, there it goes. So we performed the angiography to define high and low flow, and then did Western blot or immunofluorescence in these various regions, and had a similar result, meaning Myrsican's elevated in low flow, and MMP3 and collagen 6 are elevated in high flow, meaning that angiographic imaging externally is similar. In fact, we can just directly compare the two, right? What you have is angiographic imaging in the laboratory on the left, and in the same eye, we added the fluorescent microbeads. And you can see the high flow and low flow regions match. So what you walk away here is, first of all, the basic science approach of putting beads in the eye. This is a direct assessment of the trabecular meshwork. But the downside is this is not compatible for patients. As opposed to if you do angiographic imaging, this is compatible with patients, but it isn't technically a direct assessment of the trabecular meshwork. You're looking at it externally, but it is, however, at least reflective of it. Now, I'm going to skip through a bunch of basic work and get translational quick. Eventually, we decided to bring this to patients, and to do this, we collaborated with Heidelberg Engineering to take the clinical spectralis that you use to see patients with that's built on a table, and we install it on an arm. So now we can image people during surgery. So here you have a 65-year-old Chinese woman getting cataract surgery in the left eye. We're putting indocyanine green into the eye. You can see inferior nasal, there's pulsatile flow. This is what Gulab just showed with Murray's work with pulsatile outflow, believed to be related to heart rate. It's segmental, and it's also dynamic because you can actually see this region grow. Now, this is cataract surgery. She's going to look up, then she's going to look away, and so that outflow pattern can actually come and go. Now, what this woman's going to do now, she's going to reset her eye, and you're going to see another region here with pulsatile flow, and it's also segmental because it's only these two areas showing outflow. And then ultimately, again, it's dynamic because I want you to pay attention here. This was the region that originally didn't have flow, that developed it, that went away. It's actually coming back. Now, here's a larger number of patients. Imaged with angiography during cataract surgery, ICG on top, fluorescein on the bottom. These stains also kind of work as capsular stains too, helps with the rhexis. And so every row is a living normal person, and you can see that most of the postlimbal outflow is nasal, superior and inferior are variable. Temporal has the least, but some eyes, temporal can actually have the most. Now, these are normalized, so you might ask, what about disease? And so this is a collaboration that I've had with AIMS in New Delhi for some time. This is Dr. Shikha Gupta, who is an assistant professor at AIMS, and they started performing this imaging in India as well. What you're looking at here are adult Indians, normal, getting cataract surgery. You can see the angiographic patterns with the branching and the proper length and the proper arborization. Now, we didn't compare to glaucoma adults. Why? Because you can have glaucoma in adults without pressure issues, and this is an outflow measure, so this is definitely related to pressure. So we went to children, because in congenital glaucoma, pressure is very high, right? And so what you can see qualitatively is that we still see these branches. They're about the right length and so forth, but the arborization or the branching is actually depressed. So we went through some image analysis here, to try to quantify this. And what we can say is that the average branch length and the maximum branch length comparing normal adults to pediatric glaucoma is the same. But in pediatric disease, we have fewer branches, fewer junctions, and also fewer perfusion density as well. And so what this really reminds me of is, remember when we were in college and we all took physics, right? And we did circuit analyses. And what we learned was that if resistors are in parallel, total resistance drops. In this case, we're talking about loss of resistors in parallel, which is therefore increased resistance. So this is basically imaging that, at least in this cohort, is giving this idea of where the high pressure and where the resistance is coming from. Now, in adults, let's go back to adults, we can actually use both tracers, ICG first followed by fluorescein. And the point here is that the patterns are really similar. Why is this important? Because you can use one tracer, get a baseline, figure out what's going on, do an experiment, and then use a second tracer to see what the actual effect is. And this is where we get into concepts of targeted surgery, right? Should we be performing surgery where flow is good, like here in this nasal green arrow? In fact, this is how I was taught to do trabectome by Don Minkler. The idea here is that we're jumping onto these pathways that is already good. But if it's already good, what are you doing? Or should we do surgery where flow is bad to give yourself room for improvement? But if it's bad, maybe it's because the anatomy never supported it in the first place. So we had to test this, and this led to this collaboration that was funded by Glaukos and Heidelberg Engineering, where we went to Armenia. What you're looking at here are glaucoma patients. This region right here, once ICG goes in the eye, low flow. The two blue arrows are two iStent injects placed in the trabecular meshwork. We chased with fluorescein. What used to be low flow was rescued back in a patient. Look at this patient. This one's a little interesting. Similar initially, ICG goes in, the red arrow shows the low flow region, one iStent inject goes there, you run the fluorescein, and that comes back. But remember, to be on label, and we stayed on label for this experiment, there's always a second stent 2 to 3 clock hours apart. So the second stent now is near adjacent this baseline high flow area. So when the surgery is placed there and we run the fluorescein, we don't get a new pattern. You get that same pattern, but now it shows up fast, earlier, brighter. This is like a different type of improvement, 2 types in one eye. So what we walked away with was this idea that after trabecular bypass in a small number of patients, we had 14 patients, we see different types of improvement, including failure. But we weren't sure which one of these improvements was associated with better IOP reduction. So we needed to take another approach, and we went back to the laboratory. So this is Dr. Clemens Strohmeyer, who's an associate professor at Johannes Kepler in Linz. He joined my lab during COVID, and what he's doing right here is he's melding the angiographic system with the perfusion system that Dr. Zodi showed you, very similar, to be able to measure outflow facility. And the concept is, can we show that these different areas of segmental high and low flow have meaningfully different quantitative metrics? So on the one hand, we did an occlusion glue study. On the other hand, we did a surgical study. So here's the glue study. Step number 1, you get an eye, you figure out its outflow facility, you measure it. Number 2, you do the angiography. You define high, you define low. Number 3, we glue the trabecular meshwork shut everywhere except a baseline high or a baseline low. And then we measure facility again. Now listen, we're gluing the trabecular meshwork shut. So therefore, resistance always goes up, facility always goes down. But if we leave the residual spot that's open low flow, the drop is bigger than if we leave the residual spot high flow. The drop in facility is much more. And so what this tells you is that these different areas of the TM that we're targeting with surgery, they're not all the same. They do actually have measured different behaviors and parameters, so that targeting might make a difference. All right, well then let's get to targeting. Let's do the surgical experiment now. Step number 1, get an eye, measure the outflow facility. Number 2, perform the angiography, high flow, low flow. Number 3, perform a 5 millimeter goniotomy. You open the TM, high flow. Low flow, you measure the outflow facility again. So here you're looking at both pig and human eyes. When we do the goniotomy surgery in both species, the outflow facility goes up, resistance goes down. We're opening the TM. But if the surgery is done in a low flow region, it goes up bigger. If the surgery is done in a low flow region, IOP reduction is also larger. And so this was the first basic science or laboratory or physiologic study concept that targeting could make a difference using surgery, and that we should be targeting low flow. But wait a second, this arrows on the temporal side of the eye, was this a coincidence? Maybe we should just really be doing temporal surgery, because remember I told you, usually temporal has the least. And so this was studied in AIMS as well. This is Dr. Tanuj Datta, also part of the glaucoma group in New Delhi. And they're performing bent needle goniectomies as their choice of MIGS because it's a lower resource environment. And so they basically replicated the angiographic study I did, where they did this imaging in temporal, there's not a lot of signal, but after opening the TM on the temporal side, he could actually show rescue as well. And since temporal is usually low flow, maybe this is the poor man's way of just going after low flow regions. And so right now, we're just starting to get that clinical information. The paper's just been submitted. We have a series of patients, 15 and 15, surgery temporal, surgery nasal. Doesn't make a difference. There's a very little change in IOP, meaning low flow is a little bit lower. But listen, this isn't how we do these studies because people are on drops. We do these studies by defining success. And so partial success is going to be 20% reduction in IOP with drops. Full success is going to be 20% reduction in IOP with no drops. And total success is both. And so this is a measure here in a Kaplan-Meier analysis of total success, meaning that when surgery is done in a high flow region, there's more over here, there's more failure, and surgery in lower flow regions in a clinical cohort seem to do better. This is the first clinical window into the fact that surgical targeting may make a difference. Now, a couple slides real quick on drugs, because we do surgery, we can also study drugs as well. So this is work done in collaboration with Chris Gerken at UAB. I'm gonna make a quick statement right here. I know we're in Irvine, but I kind of feel like we're in Birmingham. I mean, we have Christine Curcio here. We have, Yuhua, who spoke earlier, made his bones with Christine Curcio at Birmingham, and here we got more data coming from Birmingham as well. So basically, at UAB, they've been performing this imaging as well. High flow regions, low flow, but no surgery. We add Myocol. Myocol is what you use during cataract surgery. Myocol is FDA-approved acetylcholine to bring the pupil down, but Myocol also opens the trabecular meshwork to lower IOP. So we see that you can actually get improvement. Here's the control. Now here's the funny part. While this makes sense, what's weird to me is why is the improvement only in these two spots? Myocol is a small molecule. It's everywhere. How come there wasn't improvement here? What does it mean to be rescuable? And what are the properties of being rescuable? These are the things that may lead to glaucoma and the things that we need to actually understand in terms of this spot versus this spot to really get at the disease. Now, having said that, listen, here's the real problem. After all this work, I'm telling you, the angiographic approach isn't going to work. It's too laborious, it's expensive, and it's invasive. We ultimately have to go back to something non-invasive, simpler, which is going to be OCT, particularly of the anterior segment. But in order to do that, we have to connect the meaning of flow to structure. That structure function concept that's always been in the back of the eye needs to be reopened now for the front of the eye. Now, what you guys already know is that posterior segment OCT is way more important than anterior segment OCT, both clinically and in research. And I don't know if you guys have ever wondered why, but if you understand why, then this allows for improvement as well. This is a totally different talk, but the point is that there are strategies to try to improve imaging in the front of the eye. And one strategy I'm taking with Dr. Hao Zhang, who's a bioengineer at Northwestern, is to do tissue or outflow specific OCT of the front, meaning optimized wavelength and then perpendicular or conformal imaging of just the outflow pathways. If we just bring it around the outflow pathways, we always maintain the shortest distance between the OCT to the tissue, and we can always maintain that perpendicular configuration and not image areas we don't need. And what that allows us to do is then create outflow reconstruction, circumferential 360 degrees around the eye to understand what's going on. Now, of course, this is mouse, and we need to transfer this to human. And real quick in terms of drugs, just like the data at UAB with Chris Gerken, we've seen similar results in mice, where if we add pilocarpine, there's actually an improvement of outflow areas in a very focal region. So in conclusion, multiple groups, multiple species, multiple tracers, enucleated and live eyes, outflow is segmental. Our TM, surgical, and pharmacological interventions influence that segmental outflow. The segmental outflow may even influence the success of the treatments. Ultimately, we're going to have to move to structure to be able to evaluate what's going on more easily, cheaply, and non-invasively. Thank you.
Thank you so much, Dr. Huang. Do we have any questions? Okay, I have a quick question. Oh,
I thought I was going to get away.
You almost got away. Well, Dr. Zode, you can go first if you'd like. Go ahead. Go ahead. Oh, I had a quick question. Do you think that the anesthesia had any effect on the results at all, as far as like aqueous outflow and the segmental nature of it or anything like that?
The answer is yes, and probably even more than just that. We all know that anesthesia can impact, intraocular pressure. In this case of, cataract surgery, we're typically talking about benzodiazepines and, a little bit of fentanyl. So that's going to be there. The other thing to keep in mind, though, is also dilation, because a lot of these patients are getting cataract surgery, right? So we're getting phenylephrine in tropicamide. Tropicamide is an anticholinergic. It can actually decrease outflow, right? And so a lot of the human results we're seeing is in the context of clinical surgery. So no doubt that the imaging itself is going to be impacted by some of the drugs they're getting.
Yeah, very interesting work. So my question was on your initial work. Yeah, okay. So in your experience, in your work, do you think the downstream of TM pathway, like the Schlemm's canal and the collector veins, how much resistance they provide in glaucoma especially? And is it correlated to low flow or high flow?
So it is absolutely important. I mean, we both know that, a lot of the seminal work that's been done has shown that TM is 50% or more, but, that sounds great, but that means that distal is actually 50% or less, meaning that distal actually still contributes, significantly. Now, we're trying to study that now. The reason being that, with the Strohmeyer work in terms of targeted surgery looked great, but there was variability in that data, right? Not everyone reacted the same way. And so what we're trying to understand now is whether or not distal pathway caused that variability. So one thing that we're doing, we talked about lipids and so forth, is we're trying to apply OCTA. I mean, how can you do this? We do OCTA to follow blood in the eye. Well, we can add things to the eye to create that contrast as well. The simple way is to go to Ralph's and buy some whole milk, but whole milk is not that reproducible. So we bought lipid emulsions. If you buy lipid emulsions, the lipid itself can act as an OCT contrast agent. And we can mix it with fluorescein to go in the eye, to use a fluorescein to tell us high flow, low flow, and then to use the OCTA to tell us about the features of those pathways, like the distal regions. And what we're seeing, at least right now, is that not all low flow regions are the same. Some of them have Schlemms, and that's it. Some of them have no Schlemms and no collectors. And the question then is that anatomy and the variation therein impacting the system and impacting the results that we get during the surgical evaluation? So that distal pathway may be important. Thank you
so much. And that will conclude our glaucoma session, and we'll turn the time over to Cornea.
All right, good afternoon, everybody. I'm not Marjan Farid. Unfortunately, she was unable to make it, so I will be functioning as a moderator and also be giving a presentation here. I think Dr. Vladimir and Lynn were very wise in selecting ocular surface disease and dry eye as the final topic, in that it's the most exciting. Probably knew that everybody was going to be kind of hypoglycemic at this point, and kind of to help wake everybody up. So that being said, we're kind of switching gears in that my presentation is more going to be bringing in clinical aspects of dry eye disease. So going from the bedside, and then we'll be moving on to lectures that will bring us back to the bench, but hopefully this will kind of help tie things together. So I have no financial disclosures. We all know dry eye is one of the most common diseases, and in the US, over 38 million people suffer from this. Many of us in this room, about 1 to 7 adults, have dry eye. And this is something that is not going away, and I think with our lifestyle, it's just continuing to increase. We're finding more about potential risk factors, and it's not just the single risk factors, but also we're finding that multiple things are causing this, which has become very important when it comes to the treatment of dry eye. I think we can all agree that dry eye is basically loss of the normal homeostasis of the tear film. We all know the important parts of the tear film, mucin coming from the goblet cells providing viscosity between blinks, the aqueous component having metabolites and various nutrients, and then the outer lipid component coming from the meibomian glands, kind of providing more so with the visual focusing of the ocular surface. Dry eye sounds like it's not a big deal, but clinically, you can kind of see here with moderate evaporative dry eye, this can really affect the patient's visual acuity. Kind of giving a real-life example, it's like driving a car when the windshield is dirty. So now put a dry eye patient in that windshield, and it probably should be illegal for that person to drive. But this really does impact our patients day to day. And we've come a long way in terms of how we're clinically approaching dry eye. Back in 2007, kind of breaking it up into aqueous, deficient, and evaporative. In 2017, moving on to now focusing on symptoms and clinical aspects of dry eye, and how we can bring that into how to treat dry eye. But kind of here, just want to highlight, it all comes down to bringing back that homeostasis, whether it's aqueous, deficient, evaporative, or a mixed picture. And this slide just kind of highlights right now where our treatment options that we have available and where we're focusing on, and kind of color coding the different options that are there. I think looking towards the future is how we can target 2 to 3 of these areas with one treatment option. So we're going to kind of get a little bit more into that, starting off with what is currently available when it comes to dry eye treatment. So in the early 2000s, we had groundbreaking FDA approval of topical cyclosporin, but not much real exciting was happening, until 2018. So there was a lot of movement from 2018 to actually last year's bench to bedside meeting. We had the availability and approval of basically 5 new treatments when it comes to treating dry eye. A cholinergic agonist, which basically is instilled into the nose twice daily and through the trigeminal parasympathetic response. We believe it increases tear production as well as tear stability. We did have a generic version of cyclosporin. I know this is mainly a research-oriented meeting, but when it comes to the real life, it's difficult for our patients to afford that treatment option. But that was a 0.05% formulation. There was a higher dose, a 0.09 formulation that became available of topical cyclosporin, which this we all know is targeting the T-cell mediated inflammatory response that can go along with dry eye. We also had kind of further up the pathway with lifitegrast targeting LFA1, that same kind of T-cell mediated immune response. And then we know with dry eye, it's a waxing, waning, kind of fluctuating disease process. And, early on, we didn't have great topical corticosteroid options, but we did have a nanoparticle loperidonol become available. The idea here is that we are kind of more focused in terms of the absorption, more superficially, not getting deeper, potentially avoiding complications such as increase in intraocular pressure and cataract formation. So from last year's meeting to today on the clinical side, there's actually been a lot of movement. So if you imagine in those 5 years, we had about 5 options. In the last 9 months, we've had 3 new options become available for us to use clinically in treating dry eye. The first being PFHO, which in the clinical trials was referred to as Novo 23. This is the first anhydrous lubricant that has become available. Additionally, another cyclosporin at a higher content, so 0.1%, coupled with a similar anhydrous lubricant. So this is kind of one of our first targeted treatments where we're focusing on inflammation, but then also on tear film stability, and Lottner ophthalmic solution, which is an addition to the antimicrobial treatments that we have for Demodex blepharitis and the MGD related to that. So with PFHO, this has a dual mechanism of action. Because it's lipophilic, it can interact with the tear film, improving stability, and then also it's believed to be able to absorb into the meibomian glands, potentially softening the meibum. So with this, we basically have a medical grade lubricant that's working on 2 ends. Here, just wanted to highlight, compared to our commercially available over-the-counter lubricants, this medication basically does last longer with a smaller amount instilled on the ocular surface. We've kind of hit this with the cyclosporin, but basically movements have been to higher percentages, and also with lifitegrast becoming available, but with a more recent the higher dose of 0.1%, coupling this with a lubricant. So once again, being able to target 2 different mechanisms of dry eye pathology. And I think for us using this clinically, noticing that patients are tolerating it, as we went up with the higher concentrations, patients were complaining about irritation with instilling the cyclosporin. So not seeing that as much with this. With the Lottner ophthalmic solution, this is basically a GABA chloride ion channel blocker, which paralyzes the insects, and in this case, the parasite Demodex. So this is basically a game changer for us because we can now specifically treat this type of blepharitis. And because it's lipophilic, it tends to get into the base of the lash and the meibomian glands and really eradicate this microorganism that sets sets up shop there. Also, it's tolerated pretty well. The current dosing is twice daily for 6 weeks. And then the patient, if they get recolonized, we can do it again. But on this end, basically, something to help on the antimicrobial side when it comes to meibomian gland dysfunction arising from this blepharitis. On the diagnostic side, clinically, we've had MMP9 tear film markers available for some time. It's helpful with identifying inflammatory dry eye. We've had osmolarity as well in the clinic. This has kind of lost favor just because depending on the time, who's doing the sample, the results kind of vary. But in this last year, there has been an IgE testing, a point-of-care testing that has become available. If you're wondering why does this kind of pertain to dry eye. In many cases, what we're seeing clinically is a mixed picture of allergic conjunctivitis with dry eye. So this kind of allows us not only to properly make a diagnosis, but follow these patients and ensure that we're properly treating them, whether their symptoms are coming from the dry or allergic conjunctivitis. On the procedure side, not much has changed in the last few years. We've had thermal pulsation available with this technique. We're basically applying heat, and this is followed commonly with pressure sequence to clear the meibomian glands and ultimately improve the tear film stability. I did see a poster about using SLT. That's kind of an interesting thing with Dr. Jester's group, but IPL as well, not much movement here. We've had this available. It became FDA approved a few years back for dry eye disease coming from meibomian gland dysfunction. The overall mechanism of how it's working is not fully understood, but a couple postulated ideas of what's happening there. So now focusing on what's coming down the pipeline for us in terms of clinical options when it comes to treating dry eye. So Reproxalap is a RAS inhibitor. We know that RAS are involved higher up in the inflammatory cascade. And so with this potential medication that's in clinical trials, it gives us a new option for treating inflammation on the ocular surface. Their clinical trial, they submitted to the FDA last year, and this was the response they got. So obviously still not available clinically, but there may be some movement with this option, which could be a game changer for us on the clinical side. Thymosin beta-4, this is kind of another proposed sterile, preservative-free eye drop. It is a regenerative peptide that's found all over the body. And their initial trials did not meet the primary endpoints, but they did find statistically significant improvement in grittiness after instilling it for 2 weeks in patients with dry eye. They have a fourth phase 3 clinical trial ongoing, so this may be one that we may be seeing on the clinical side. The transient receptor potential myostatin 8 agonist, the idea with this is stimulating the cold receptors on the cornea and the eyelid, potentially that way increasing tear production and maybe having a cooling sensation on the ocular surface. There are 2 phase 3 clinical trials with this option. AZR-MD-001, this is a keratolytic, so this is also kind of a new potential treatment option for treating MGD, but basically with this breaking down the abnormal bonds in keratin to soften the blockage of the meibomian glands, potentially also preventing future blockage. It's applied to the eyelids twice weekly, and the goal here is targeting the meibum, making it softer and better quality and more quantity. Visomitin is a cardiolipin peroxidation inhibitor. Kind of a few ideas of where this may be acting. There was a phase 3 clinical trial that did conclude, and there's another one that's ongoing, but they did find statistically significant improvement in dry eye staining. Glaukos, not a new medication, but a new formulation of a cream-based drug. We're basically applying pilocarpine transdermally to potentially get some of the dry eye benefits without the side effects. This is in a phase 2 clinical trial. So lots of excitement, lots of things coming down the line. I think just highlighting, we may potentially have a TNF alpha inhibitor. Surface Ophthalmics does have a combination clinical trial medication with mycophenolate and betamethasone. The other, utilizing topical nerve growth factor, Sanedgermin, with other dry eye treatments and kind of seeing where that goes and with dry eye treatment. And then other areas with androgen eye drops and also secretagogues. With this, obviously, a new technique of focusing on the mucin-producing cells, the goblet cells. So I think, what are we missing? We talked a lot about what's coming, what are we targeting, but really what I'm seeing on a day-to-day basis is behavior modification. We are just so dependent on screens. Many of you, I'm not trying to put anybody on blast, but we're on our cell phones today, on our computers, and so we know the longer we spend on screens, our blink rate decreases and our tear film just gets worse as we spend more time. So currently we don't have great treatment options when it comes to this. So counseling patients to take breaks, use more lubricating drops, or adjusting the brightness of their screens. I think we're kind of behind on this, and this is something where, using technology, whether it's AI, where we can provide our patients or us in general, reminders that, the screen can kind of detect our blink rate or our tear film osmolarity. The same way when you get in your car, you plug your car in to the Bluetooth, it reminds you that you're driving and kind of blocks you out from using it. This is an area, I think, in terms of potential treatments in the future that we can use technology to our advantage. So that being said, dry eye disease is growing fast. Obviously, we have two great presentations that are going to follow that will be focusing on both the age-related changes of the meibomian glands as well as tear film markers. Newer medications looking at 2 approaches or potentially targeting 3 would be great for us clinically, and then new novel techniques like biologics topically available would be a game changer for us. All right, thank you. All right, I guess we can move on with Dr. James Jester. He is a professor of ophthalmology here at UCI and also a professor of biomedical engineering. He's going to be sharing with us one of his research interests, specifically the effects of altered meibomian quantity and meibomian gland population dynamics.
Thank you. Let's see if I can figure this out. Yes. Before I start, I'd just like to mention my thankfulness for the committee who selected me to give a talk, particularly at this time recognizing Chris Kenny. I've known her my entire career, or at least known of her work, particularly in anterior segment before she turned to the dark side and went over to the retina and mitochondria. And that she did a lot of wonderful work in the cornea, particularly in keratoconus, and also recognize her for her work in the National Keratoconus Foundation, which has provided funds for those of us working in that particular area to continue on our investigations. Today I'm going to talk about, I'm not going to talk about age-related, I'm going to talk more about specifically our work looking at the effects of altered meibum quality on meibomian gland dysfunction and what happens to the cell population dynamics. This is a collaborative work with a group in biological chemistry, Dr. Bowie Anderson and his graduate students. Also, the animal model that I'll be talking about comes from collaboration with Brian Leonard at the veterinarian school at UC Davis and postdoc who identified the mouse that I'll be describing today. We just heard about dry eye disease, and I think what I'd like to mention is that somewhere around 80% of dry eye disease is associated with what's called meibomian gland dysfunction. And I've spent a greater part of my career trying to understand meibomian gland dysfunction. And what we do know from a clinical standpoint is there's 2 signs, basically. One is when you pull down the lower eyelid, you can see dropout of gland, and that's been known for some 40 years. The other thing is that going along with this dropout of gland, if you press on the eyelid, you can express lipid from the gland, and that comes in several different flavors. One, it can be normal, which you can hardly see, and some people say it looks like olive oil. or it can be more opaque, it comes out more difficultly and you have to press harder, or you have to press really hard and you get out this toothpaste-like stuff. And a lot of chemists have been working on what's the chemical reason for these changes. We really don't know. I think as we go forward, we'll learn more about that, but it's still sort of unknown. The basic mechanism, though, that's thought to involve the loss of gland and this altered change is an obstructive mechanism, but it doesn't really explain what happens with this altered meibum quality and what that does to the function of the gland. Recently, we published a paper, Shanggang Liu, who is a biomedical engineering graduate student in our lab, did a finite element model analysis. I recommend you might read this. That sort of explains how meibum is expressed and how obstruction might occur. And one of the first things we have to realize is that meibum is not a Newtonian fluid. Instead, it is a non-Newtonian fluid, so it's much more like ketchup, which means it has a resistance to flow. And as you tip it over a ketchup bottle, you have to give it a shake to overcome that resistance, and then it will flow. Which means that when our eyes are open, we're not secreting any meibum. Meibum is only secreted when you blink, which is about 0.2 seconds over a 5-second period from when you get blinked. So it's a very short period of time that lipid can flow. And when lipid does flow, it flows because of the contraction of the orbicularis muscle, exerting force on the eye, and then you have this blind end tube of the meibomian gland. This force then pushes meibum through this terminal duct, and of course the diameter of the terminal duct also plays a very important role on the flow of lipid out. But one of the key things is this resistance to flow as a non-Newtonian fluid, which depends upon really the temperature of the material that you're looking at and the melting point or the phase transition change of that. And for meibum, it's around that last phase transition is around 32, and you can see that the resistance goes up dramatically if that temperature changes or gets lower, and also the viscosity goes up much more greater. So you can get these different degrees of flow depending upon the pressure of the eyelid. If you have a high pressure, a good strong blink pressure, you get a lot of flow. Have a weak pressure, like when you get older, you have no flow, or you leave your eyes open the whole time, you get no flow. And we normally exist somewhere in here, but as this temperature shift phase changes, you get a marked change in the amount of flow that occurs. Now, why are we interested in wax esters? Well, there's sort of a growing consensus that the combination of wax esters, which represents about 40% of the lipid in the meibomian gland, with the cholesterol esters, which represent the other 40%, that together those define the phase transition of meibum, moving from a fluid liquid olive oil-like solution to a more opaque or inspissated meibum. So this wax ester plays a very important role in defining the temperature shift, as it has a much lower melting temperature than the cholesterol esters. Also, more recent work by Budovich at University of Texas has shown that there's a change, a decrease in the wax ester concentration, and also that's been shown in some of the cases of meibomian gland dysfunction, that there's less wax esters in the meibum. So we're interested in what these wax esters are and their synthesis. Budovich has worked out all the sort of enzyme chemistry. There are two key enzymes, FAR2, which produces fatty alcohols, and also the AWAT2, which produces the final wax esters. Interestingly, both of these enzymes have been knocked out, and they lead to plugging or obstruction of the meibomian gland that has been noted, and dramatic dilation of the meibomian gland duct, and a dry eye phenotype. So we had the opportunity, in collaboration with UC Davis and Brian Leonard, to look at the AWAT to knockout mouse which is a bit different than what's already been published by several different other investigators. But this is a mibographic image of the reflected light of the conjunctival side of a wild-type mouse, and these are the meibomian glands. The AWOT2 shows marked enlargement of the glands, plugging of the orifice, this little white stuff here. Here's the inspissated and non-melted meibum that's out on the lid margin of the mouse. Histologic sections comparing wild type, you can see a very small, narrow duct with very thin epithelium. In the knockout, this duct is markedly swollen and thickened and dilated. As you can see here too, thickening hyperplasia of the ductal epithelium, and then some, what appears to be essentially normal acini. So we wanted to ask the question, what does this dilation of the duct do to the population dynamics? And we performed some single-cell RNA-seq looking at both wild-type and AWOT2 knockout mouse. We performed some spatial transcriptomics, and what I'd like to propose today is sort of a potential mechanism for meibomian gland dysfunction and dry eye disease at the end of this talk. So we did single-cell transcriptomics. Basically, we cut out the tarsal plate from the mouse, which contained the meibomian gland, some conjunctiva, some of the root hair follicles of the cilia, some leading edge of the skin of the eyelid. And we digested that, collected single cells, and ran it through the 10x genomics. Also, we took cryostat sections, frozen, fresh, unfixed, placed them on glass slides and submitted them to Resolve Biosciences for doing spatial transcriptomics. They have a specialized proprietary way of labeling different probes. You can, in their particular systems, there's 100 transcripts you can look at. They're all individually labeled, so you can then spatially identify the expression of those transcripts in the tissue. The resolution in this system is about 0.2 microns, so we're well within a single cell, being able to measure one transcript within a single cell. The current systems are much higher number of transcripts that you can look at, but not much better in resolution. So what do we see? With the single-cell RNA-seq, we were able to identify 8 different types of cells in the eyelid. As you might expect, we got meibomian gland cells, hair follicle cells, conjunctiva, fibroblast, immune cells, pericytes, melanocytes, and endothelial cells, and they could all be identified by specific gene expression patterns that uniquely identify those cell populations. But what about the meibomian gland? And the meibomian gland, everybody talks about keratinized epithelium. So if we look at the epithelium, we have progenitor cycling cells and non-cycling cells. These are undifferentiated basal cells. It would be marked by K5. And then you also had differentiating ductal epithelial cells that were non-keratinized, identified by alternate non-K1, K10 expressing cells. And then you also had the differentiating ductal epithelial cells that were keratinized, meaning they expressed a marker for keratinization that was K1 or K10. Each different one of these groups can be seen here as expressing different patterns of genes. For the meibocyte population, or the acinar population, the cells that are making the lipid, you have cycling progenitor cells and progenitor cells that could be identified, and then differentiating meibocyte cells that were expressing specific genes of differentiating acinar cells. We're interested in also identifying stem cell populations, so we've looked at the progenitor cell populations. We regressed out the cycling gene so we could just look at the other genes. And we could see that these cycling cells, both from the duct and the meibocyte, had different gene expression patterns, which suggests to us that under homeostatic conditions, the cells giving rise to duct are not anywhere near similar to those that are acinar cells or meibocytes. This can also be seen in this trajectory analysis where they stay separate, and also we see the different cell types here that remain separate after we regress out the cycling gene. So we think this data is consistent with our lineage tracing data, which we've already published, and our label retaining cell experiments, which we published, which suggest that the stem cells, there's one stem cell per acinus giving rise to acinar cells and a separate cell that gives rise to duct. Looking at the sequential, at the normal wild-type animal, looking at the spatial transcriptomics, each dot represents a collection of RNA expression. This is the conjunctiva here. These are the meibomian glands cut through the duct and the acinus, hair follicle cells, muscle, skin would be down here. We could identify 4 different populations of fibroblasts, 3 or 4 different populations of meibomian glands type cells. Interestingly, if we look over here at AWAT2 expression, which is required for wax ester synthesis, versus SOAT, which is required for cholesterol ester synthesis, we can see there's a segregation, differentiation between the localization of these RNAs in the acinus. So this would be basal acinar area around here, or if you can see over here, if we look at FAR2 and AWAT2, and a basal cell marker, IGF-BP2, which is around the outside. So you have basal cells, you have beginning suprabasal cells, and then inside that, near where cells are beginning to disintegrate, you have the AWAT2 expression, but they're separate from the FAR2 expression. Remember, FAR2 is fatty alcohol. It's a precursor of the wax esters. So this suggests this is also paralleled in the protein. Here's AWAT2 protein. This is the K5 marker delimiting the basal cells. Here's AWAT2 expression in the center of the acinus, no expression out in the periphery, which would be the early differentiating cells. So we think this suggests that the acinus, the meibum synthesis, is segregated in its synthesis. It's stratified, much like skin is, where you have basal, spinous, granular cell, and then the cornified layer. The same would occur for the acinus differentiation. You have a basal compartment. You then have a early suprabasal compartment where the cells are making fatty alcohols and cholesterol esters, and then they later make the wax esters and perhaps elongate the lipids later that are being expressed. So this is sort of our revised idea of how you have stem cells that lead to progenitor cells and then transitional basal cells. Then they begin to differentiate and express genes that synthesize some lipids, and then later genes that finish the synthesis of the meibum. So that's the wild type. If we look at the AWAT2 knockout, we see similar types of meibocytes that are being present. We have ductal progenitor cells. The ductal progenitors are non-keratinized and keratinized duct. But when we look at the percentage of cells, we see this great increase in the ductal progenitor cells that are proliferating and non-proliferating compared to wild type. We see a marked decrease in the keratinized, representing here's keratinized ductal epithelium wild type. That's actually decreased, which is against what was published by previous papers. We have increased in the non-keratinized here compared to what's in wild type. And then also decreases meibocyte progenitor cells and differentiating meibocytes. So the meibocyte population seemed to be decreased in AWAT2, whereas the ductal population is increased. The trajectory analysis confirms this type of switch to more of a predominantly ductal. And if we look at the gene expression patterns between AWAT and normal meibocytes, we see a decrease. This is increase here in the meibocyte population, wild type, but not lost here. And those that are low, CNN2, which is an inflammatory marker, they're upregulated in the AWAT2 knockout. Same for duct. And also we can see in the duct there's an increase in the PERK endoplasmic reticulum unfolded protein, which is also a marker for inflammation. If we look at the eyelid populations, again, one of the main things that we see is a completely new population of neutrophils, which is localized over here and identified in this, that's not seen at all in the wild type. And we get marked increases in the T cell and lymphocytic cell populations and the dendritic cell populations. So there's an increase in inflammation around the gland. If we look at the specific cell types, we can see IL-23 is upregulated predominantly in the differentiated duct non-keratinized cells, maybe some also in the ductal progenitor cells. And then this LCN2 is increased in all of the cell types in the AWAT knockout mouse, indicating inflammation. The LCN2 is a lipocalin-2 associated with innate immune responses. Also, the IL-17A is upregulated in T cells. This expression pattern of IL-23, IL-17A, and these other inflammatory markers is very reminiscent of psoriasis, where you have upregulation of IL-23, recruits IL-17A T cells, and then the recruitment of PMNs, and a psoriasis-like syndrome. Other markers of psoriasis were also seen, S100A, A8, and A9. This is young and old wild type, young and old knockout. We can see the yellow for the S100A8, and it's localized here at the lid margin. This is K10. You can see K10 is not present at the orifice of the gland here, but is present there at the mucocutaneous junction of the wild type. So there's no K10 involvement, no keratinization involvement, upregulation of these inflammatory psoriasis markers. And you can also confirm this with staining for PMN infiltrates. So what we'd like to propose is that various types of conditions, environmental conditions, aging, cholesterol diets, androgen perhaps, changes the meibum viscosity, shifting the melting point. This leads to obstruction, dilation of the duct, and a mechanical strain similar to psoriasis, which results in the upregulation of IL-23, recruitment of PMNs and Thi-17 cells, and then you get this ductal hyperplasia and a vicious cycle that occurs. And with that, I'll take questions, and thank you for listening to me.
I should have gotten up. I thought that was a fascinating talk. I was wondering about the life cycle of those cells. You've got the progenitors and kind of a parade of cells towards the right-hand side of your diagram. Do they turn over? What is the time frame of turnover? Do you know? And is any of the obstruction due to sloughing of cells into the duct?
So there's been one study that really looked at the half-life of cells. So the meibomian gland is a holocrine cell, which means that the cells have to divide just like the skin. You have to have basal cells divide, and then they differentiate and then are desquamated. The same thing happens with sebaceous gland and meibomian gland. You have a basal cell proliferation, which then the cells differentiate, fill with lipid, and then disintegrate. And release the lipid into the duct. And that's a 9-day, in a rat, meibomian gland, it's a 9-day life cycle from the time it leaves the basal compartment. The proliferative rate's somewhere around 25%. So 25% of the cells are in cycle at any one time. So I imagine it's about 4 days. We don't know how long it takes for them to go through, but it seems that, if you do labeling, the cells from one acinus seem to come from a single cell. So there's only one progenitor cell under homeostatic conditions, only one stem cell that would lead to the transient amplifying cells like we see in the cornea. Thank you.
Thank you, Dr. Jester. So we have Dr. Sarah Hem Alvarez, who is a professor of ophthalmology and pharmacology at USC Keck. She is world-renowned in cellular membrane trafficking and high quality imaging. And we're going to be speaking to us about new biomarkers of the ocular surface.
So thank you. I am grateful to the organizers for inviting me today, and I'm very well aware that I'm what stands between you and a very nice reception, so I will try to be brief. I work on the lacrimal gland and ocular surface system, and we look at mechanisms of dry eye development, therapeutic development. We have a particular focus both on Sjogren's related dry eye and age-related dry eye. But today I'd like to talk about some directions that we've taken for identification of new biomarkers, and in particular working on Sjogren's syndrome. I have no relationships to disclose. So you heard a very nice presentation, several presentations on dry eye, and so you know that it's a highly prevalent condition, and those of you who do treat patients with dry eye will know, as it was mentioned, 1 in 7 adults has dry eye. Sjogren's is a form of dry eye that's characterized by a lymphocytic infiltration of the lacrimal gland as well as the salivary gland. Sjogren's is associated with reduced tear and salivary flow as well as damage to the ocular surface and to oral health. Sjogren's is also a systemic disease, and you get inflammation and loss of function of multiple internal organs, as well as increased risk of B-cell lymphoma. And so within that population of dry eye patients that are coming to the clinic, 1 in 10 of those patients will be a Sjogren's patient. So there have been, since I started in this field, there have been a number of challenges in Sjogren's diagnosis. When I first started, the time to diagnosis from manifestation of symptoms was over 6 years. It is now 3 years, but that's still too long for patients with this disease. So some of the challenges in diagnosis are due to cumbersome formal classification criteria. So for a patient to have Sjogren's, they must meet these 2016 ACR-EULAR criteria that require the presence of signs and symptoms that are both systemic, local, related to the lacrimal and salivary gland, some of them invasive, such as the performance of a labial salivary gland biopsy. So this isn't used as much in clinical practice because, unfortunately, diagnosis of Sjogren's often doesn't make as much a difference to the patient as it should. So there are currently no dedicated therapies or specific therapies for treatment of Sjogren's syndrome. Patients are typically treated with a constellation of immunomodulatory drugs, as well as the topical treatments for dry eye that were mentioned. And while Sjogren's patients may exhibit some relief from those symptoms due to actions on the ocular surface, there's very little evidence that any of those active agents can actually get back into the lacrimal gland and affect the fundamental autoimmune exochronopathy that is driving the ocular surface disease. So we think that some of the therapeutic therapeutic challenges may be remediated if we had better biomarkers that reflected the disease state of the lacrimal gland and that might allow us to assess different therapeutics and see, in fact, are we changing the inflammation within the lacrimal gland? So probably this audience has thought potentially that tears might be a good source of biomarkers. So tears are in fact an excellent source of biomarkers, and you have the source of biomarkers in your laboratory every time you conduct a Schirmer's test, because that's basically the material that we take away and we elute proteins or antibodies or microRNAs from it. So tears have easy and non-invasive access. They're highly enriched in proteins. You heard about extracellular vesicles earlier today. They're highly enriched in extracellular vesicles. They're cleaner relative to saliva. They also contain lipids, as you heard from Jamie, as well as nucleic acids. And most importantly, for ocular surface diagnosis of diseases of the eye, they're actually produced by the affected tissues of the ocular surface system, and they contain information related to disease pathogenesis. So for years, we've been actually working on identification of tear biomarkers that can be identified that will distinguish Sjogren's patients. And I'm going to talk about three different types of constituents of the tear fluid. So you heard already a very nice description of the tear fluid, and you're aware that there are multiple contributors. I draw the lacrimal gland bigger than Jamie does. Jamie draws the meibomian gland bigger than I do. In any event, we know that there's lipid, many proteins and nutrients in a mucoaqueous layer, and then there's a glycocalyx with protective mucins. So a lot of the focus on biomarkers has focused on the presence of proteins that are differentially expressed in the tear film. We have spent about maybe 10 years looking at a particular protein called cathepsin S that we thought might represent the definitive biomarker for Sjogren's patients. And actually, in two observational studies of patients with Sjogren's syndrome as compared to patients with other autoimmune or ocular surface disease, we found that cathepsin S activity showed incredibly high specificity and sensitivity. Unfortunately, as we continued to work in this area, it appeared that cathepsin S, one of the reasons that it's elevated is that it's increased in the inflamed tissue and secreted because it's increased, but also its endogenous inhibitor, cystatin C, is decreased significantly. And so that creates the elevated activity that's present in the tear film. So although cystatin C serves as a surrogate for increased cathepsin S activity in the tears, it's very difficult to advance a biomarker for some reason if it decreases in disease. People want to see that a biomarker increases in disease. So we were left with a protein that we think actually is also involved in ocular surface inflammation, but that doesn't work very well as a candidate biomarker. So we thought, let's look at other constituents of the tear film. And so in today's talk, I'm going to talk a little bit about some of our published work on microRNAs, as well as some new work on antibodies. And I do recognize that an antibody is a protein, but I'm going to be talking about autoantibodies, so I've grouped them a little differently. So just an introduction to our model. We use two different mice. We use the male non-obese diabetic mouse and also a knock-in to the NOD mouse that knocks in a region of the C57 mouse locus that removes the diabetes from this non-obese diabetic strain, and this one's called NOR. Both of these mice develop a spontaneous Sjogren's-like autoimmune exochronopathy in the eye. They also show decreased extracellular matrix, increased MMPs, increased cytokines, decreased secretion, all occurring spontaneously as early as 8 weeks. It is the females of the strain that actually express the sialoadenitis, or the salivary gland dysfunction that you see in disease. And despite the fact that Sjogren's is 90% prevalent in women rather than men, this model still remains one of the most popular models and has been very effective in generating leads that have actually been validated in human populations. So we thought that we would look at nucleic acid expression in the tears, and you know that nucleic acids are rich in the tears, and you've heard that many of the nucleic acids that are actually shed from cells are shed because they play important signaling functions and are exchanged utilizing extracellular vesicles, such as exosomes and other types, which enable them to be protected from degradation and also to be secreted from cells and to exert effects on target cells, some of them very distal. So we are particularly interested in microRNAs. MicroRNAs are present in tears, both free and in extracellular vesicles. And these are species that are largely regulatory in nature. So when I took biochemistry as a graduate student, we had messenger RNA, transfer RNA, and ribosomal RNA. So now we have many, many different kinds of RNAs, many kinds of small non-coding RNAs, and this is one of the major categories. MicroRNAs are 19 to 22 base pairs long. They participate in gene silencing and post-transcriptional regulation of approximately 60% of mRNAs that are actually transcribed in mammals. They're highly conserved in mammals and between mouse and human, and increasingly dysregulated microRNAs are implicated in a number of diseases. So I apologize for this busy slide. This is just meant to illustrate our workflow for the small RNA-seq that we conducted. We utilized male NOD mice as our disease control. As our disease model, and we had 2 control strains, age-matched male BALB/C mice, which were disease-free, and female NOD mice, which served as a strain control and were disease-free in the lacrimal gland. This takes you through our workflow. And what we identified, and I should say that we were conducting this small RNA-seq in tears from the mice. So we identified over 550 distinct microRNA species, which is really a lot. We found that through the unbiased hierarchical clustering that both the BALB/C and the female NOD were, the differentially expressed genes clustered these strains away from the male NOD. PCA analysis showed that there were both strain and sex effects, and we used pretty stringent criteria to select our hits, which we identified as 14 in total. And again, very busy. These just show the upregulated microRNAs relative to 2 control microRNAs, and these are those that are downregulated. So of those 14 hits, we were able to validate 8 by qPCR, and no surprise that the IPA analysis revealed that the major pathways that were affected were related to inflammation, cytokines, chemokines, and immune cell development. What we've been excited by is subsequent work that we've done in tears from Sjogren's patients. And so we used the tear biorepository at the Roski Eye Center that we've launched that basically makes use of Alzheimer strips that are collected in the laboratory and banks them for analysis. And what we did was analyze the upregulated microRNAs that we had identified in the Sjogren's model, and we tested their abundance in both tears from identified Sjogren's patients and tears from patients with meibomian gland disease. And we know from Dr. Jester's talk just now that meibomian gland disease is not an autoimmune disease process. It's an inflammatory process associated with dysfunction of the meibomian gland, and so distinct from Sjogren's. And what we were able to see is that all 3 of the microRNAs were significantly increased in the tears of the Sjogren's patient. This is also the first demonstration that miR-181A5P and 203A5P are involved in Sjogren's, period. So I'd like to conclude by talking about autoantibodies for a moment. So in the tears are mucosal surface, and plenty of antibodies are present in tears. One of the most abundant antibodies is secretory IgA, which is basically the terminal fragment of the polymeric immunoglobulin A receptor responsible for transporting IgA and IgM from the basal surface to the apical surface. And this provides a lot of protection from pathogens and other agents. So we also have IgG in the tears that gets there through paracellular transport. One of the things that we had thought about in Sjogren's is the fact that the inflammatory infiltrates in the gland may produce their own autoantibodies. So autoantibodies are a characteristic of autoimmune diseases. Autoantibodies are characterized when the immune processes become dysregulated and start to recognize self-proteins as foreign. They develop autoantibodies, and as part of the disease mechanism in many autoimmune diseases, autoantibody deposition at the tissue sites that are affected is associated with tissue destruction. In Sjogren's, the major autoantibodies that are associated with the disease are two called SSA and SSB, and they have been reported in biofluids from Sjogren's patients, most notably in the serum, but also in the tears. But we speculated that, so the germinal center structures responsible for supporting B cells that have been primed with antigen to differentiate and to produce antibodies, these germinal centers are normally organized in professional lymphoid tissue. But in Sjogren's, it's known that ectopic germinal centers can form in the affected glands. So this depicts our model of what it might look like if an ectopic germinal center formed in the lacrimal gland. And actually, we have confirmed that these structures are present in mice in the lacrimal gland of the affected mice. But I'm not showing the pictures today, but we've confirmed ectopic germinal center expression. So we thought, well, maybe there are definitive autoantibodies that are in tears, and perhaps may they be diagnostic, but may they also help us understand the degree of disease in the lacrimal gland as an additional measure. So we used an autoantigen array of 95 known autoantigens. And we screened in two cohorts, both in young and old, BALB/C and NOD and NOR mice with disease. We looked at the effects of disease and age, and these are results that have been gleaned from the IgG levels from this study. And so we see a couple of autoantibodies that are present and elevated both in tears and serum. One of these is the SSB antibody that has been extensively linked to Sjogren's syndrome. If we look at IgG antibodies that are present in the serum but are not present in the tears, we identify a subset of the total. You note the presence of multiple complement antibodies. But we also see IgG autoantibodies that are uniquely elevated in tears of the Sjogren's model mice. The Jo1 autoantibody is the most prominent and promising, but again, 10 different autoantibodies that are present in the tears of these different Sjogren's model mouse strains that may reflect local disease processes in the gland in a different way than the serum autoantibodies. So I've used up my time. I can tell that the reception is calling. I'll leave you with the conclusion that changes in tear composition of microRNAs and autoantibodies may reflect disease status in the lacrimal gland in both Sjogren's model mice and in patients. These autoantibodies and tear microRNAs may serve as possible diagnostics to identify these Sjogren's patients who come in the clinic from patients with dry eye due to other causes. It's notable that autoantibodies in Sjogren's patients may appear a decade before any symptoms, so these may be early diagnostics of disease. It's notable that tear autoantibody and microRNA differences in Sjogren's may be different than in serum, and we believe that use of tear biomarkers may enable the extent of lacrimal gland disease and potentially provide a companion diagnostic if new drugs are developed for this condition. And again, I'd like to thank my lab, my collaborators, and supporters. Thank you.
Any questions?
Hi, thank you for your talk. That was really interesting. I had a question about when you validated your small RNA sequencing with RT-qPCR. What did you select for your housekeeping genes, and how did you know that those were like appropriate to normalize your data to?
That's a really good question, and I would actually have to scroll back to see the specific tier microRNAs, but that's, we have two specific housekeeping reporters. One is the MIR25-3P, and one is the MIR93-5P. Again, we're working with tier biofluids, so there's very little published, and we weren't really sure what the reporters should be, but these are two that are commonly used, and so we basically defined hits as elevated when we normalize to either. Okay.
Hi, thank you for sharing your knowledge with us. I have one quick question. Did you try to collect the tears with microcapillary tubes or any other technique other than Schirmer? What is your experience with that?
Yeah, and so there's actually, in the field of tier biomarkers, there's a lot of controversy about trying to provide a standard method for tier collection, because sometimes the findings that you obtain may depend on the mode of collection. So we're starting to come to some consensus that Schirmer's strips are good for protein analysis and also for nucleic acid analysis. Schirmer strips are terrible for collection of or analysis of lipids. And so for analysis of anything where you would be looking at a lipid, you would want to use a microcapillary. I think there's more resistance to microcapillary collection in the clinic, just because I think that, you know, particularly dry eye patients with a very sensitive ocular surface may not like a sharp object next to their eye. And so we get a little bit more pushback from the clinicians when we've tried to push the microcapillaries, but actually people use Schirmers, they use microcapillaries, and they also use special sponges that can be wiped gently across the surface and then eluted. And they also, you can use impression cytology, but of course, you know, you'll have cellular debris there. And some individuals actually use tears collected through irrigation, and that is sometimes something we do even in the mice, because mouse tear volume is so small, and when you're working in a dry eye mouse, it can be even smaller, a fraction of a microliter. So sometimes even just bathing the ocular surface with a defined volume of PBS and collecting through capillary. Did I answer your question? Okay.
Thank you. Thank you. Just a few parting words from me. I've learned a lot. It's been a great day, and I would like to think that Chris Kenny would have appreciated and would have been pleased by the diversity of presentations we had today. We started with neuro-ophthalmology and psychophysics, went through age-related macular degeneration, retinovasculature, glaucoma, all the way to ocular surface and dry eye. And it's been particularly gratifying to see the increasing participation from of colleagues from outside of the Gavin Herbert Eye Institute. We had several talks from colleagues from UC Irvine, and we also had presentations from Duhini Eye Institute, Stein Eye Institute, USC, UC San Diego. So if you enjoyed the day today, and if you, if you've learned something and you've had a good time, we hope to see you again next year. Please tell your colleagues, please bring your trainees and have them also participate and present posters. So the last order of business, in addition to the reception, but no dinner, is that, Dana, is that we have the poster presentation, which we will do in about 5 to 10 minutes. So thank you all for being here and spending your Saturday with us. Please join me in thanking all of the speakers and all of the moderators for an exciting day, and enjoy a drink and a bite while we are getting ready for the posters. Thank you.